morning, everyone, and welcome to the day 4th of the 27th iOS PG convention hosted by Tena Dental College, NAVI Mumbai. We will start with the sessions. The first speaker for today is Colonel Dr. Manu Krishnan. Sir is a postgraduate in orthodontics from the Armed Force Medical College, Pune, commissioned into the Indian Army in 1999 with 25 years of experience in serving different dental units of Army and commanding two units. He was awarded with several military honors for distinguished services. His research areas are
Novel
novel
biomaterials,
biomaterials,
in
in
vitro
vitro
and
and
in
in
vivo
vivo
biocompatibility,
biocompatibility,
animal
animal
experiments,
experiments, stem
stem cells,
cells,
craniofacial
craniofacial
bone
bone
tissue
tissue
engineering,
engineering,
microarray,
microarray, gene
gene
sequencing,
sequencing,
radiation
radiation
biosciences,
biosciences,
and
and
oral
oral
cancer.
cancer.
He
He's
is
a
a
life
life
member
member
of
of
Material
Material
Research
Research
Society
Society
in
India, Electron Microscope Society India, and Society of Biomaterials and Artificial Organs India. At present, he is the senior specialist at the Department of Orthodontics, Army Dental College, Research and Referral, Delhi, and is involved in several high end interdisciplinary clinical and research projects. The topic for his current presentation is orthodontic biomaterials. We welcome you here, sir. To chair this session, we have Dr. Saugat Ray sir, who's a post-graduation from Armed Force Medical College, Pune, 21 years in Indian Navy, and presently working as an associate professor in Department of Dental Surgery and Oral Sciences, AFMC, Pune. Welcome, sir. Over to you, sir. Do you want the pointer? So mic.
Dr. Anisha, no?
Dr. Alisha.
Anisha.
Alisha.
Anisha.
Met you in the morning.
Anisha, no?
Alisha.
Ajisha. Thank you, Dr. Anisha. Thank Morning, Chairperson. Good morning, everybody. As she said, my topic is orthodontic biomaterials. I bring you greetings to all of you from the Army Dental Center Research and Referral. This is the main building of our institute, and this is the side block, annex block, where our department is housed. Well, all of you might have seen this book, two books, uh, dental materials books in our PDS graduation and orthodontic materials in our postgraduate curriculum. But have you noticed the nomenclature is like orthodontic materials or dental materials? But I would like to call it as orthodontic biomaterials. This is because of the type of work which we were doing for past 2 decades. I noticed that materials are not just materials like automobile materials or industrial materials. They've got a lot of interaction interface with the biologic systems. So a right terminology would be to call them biomaterials rather than materials. And if you see any medical surgical material which is inserted into the body like a cardiac pacemaker, splint or anything is known as biomaterial. What is the importance of orthodontic biomaterials? Like we might have seen a lot of clinical cases in the last 2 days and in the succeeding sessions also when they correct malocclusions like this and get the result. So the hardware which works over there is the orthodontic biomaterial. And why to study orthodontic biomaterials? To give the clinician evidence-based support for the selection of appliance for scientific and not empirical practice. The quality control in delivery of standard orthodontic care for our patients. You know, we get brackets for 550 rupees, 350 rupees also. We need to check it whether they have got the properties. Know of the iatrogenic effects and safety to self, patients and to the environment. We are concerned about not only our safety, to the environment also. To learn methods and tests to critically evaluate the biomaterials, that's also a part of our postgraduate curriculum, and to contribute the Make in India policy of Government of India and achieve natural self-reliance. Yesterday, we all might have listened to that inspiring speech of Sudha Chandran madam, and she said that she got corrected her limb amputation because of the Jaipur limb which she has got. So that's because of that was first time when India was making efforts for to get self-reliance in biomaterials technology. We have a flourishing domestic and an established international biomarket material market outside. This is an image from the stalls. You get so many products. So how do you make a right selection of your products? However, the subject is not very sought out by the students and clinicians. It's like a dull subject or a boring topic. It's generally felt like that due to the physico-chemical and biological aspects and the complexities involved in that. So this presentation is an attempt and an overview of this complex discipline and to generate interest and passion for the young orthodontist. So how do you approach the subject? To make it easier, I said we will study it as per the evolution of the specialty. Now precious metal alloys are not used. Like gold are not used as archways, or they are only used as a bonding attachment in the lingual appliances. Other two important alloys which we use regularly have come from the industry, like stainless steel and watch spring, cobalt spring. Third one has come from again from an aircraft. It has come from the reconnaissance aircraft of US. From there we got the beta titanium, and of course many of you may be knowing that nickel titanium has come from Naval Space Research Laboratory of US. Have you also contributed to the biomedical science? Yes, there is Indian contribution to that. We all may be knowing that iron pillar in Delhi in Qutub Minar, it has withstood rust made in 402. So we also know about a little bit about metallurgy and science of that. So my humble work on this from our research and development on biomaterials. This is from the Indian Missile Program, and the line shows the protection which Indian DRDO gives to the country in terms of different levels of missiles which can reach up to our adversaries and far off places. Now, another approach to study this orthodontic biometals would be like to see it in the global perspectives. If you see US, starting from EH Angle, Tweed, Murrayfield, Kessling, they are all there. And then here, this side in Australia, you have Bex, and our functional appliances all come from Europe. And then Southeast Asia with India, China making up the large number of orthodontists all over, and the market and the patients and everything. And we have the Make in India concept initiated by the current government in 2012. So if you see these are the appliances, you may be knowing it in terms of in chronological I will put it. If you see, it has got the tryst with biometals. You can see it's a mix of like stainless steel and precious metal alloys. And now we have the removable appliances, the traditional appliances from the Europe side, and the new appliances which are a mix of Europe and US. And there are several type of appliances. I'm sure that most of you might have studied comparative evaluation of the studies in their PG program. And this is a recent work done by our resident, Major Garima. She has also compared two things. Yesterday, Sir was telling about the comparison of lot of class 2 corrections. So the mainstay for appliance is stainless steel. It is the main appliance, major appliance which we are using it. What is stainless steel? It is an alloy of base metal alloys, iron, thallium, zinc, nickel. If you can see that pie chart, the iron is the maximum content of 79, and there is an 8% nickel. And it also got phases like austenitic, ferritic, magnetic, and a mix of that duplex. So little bit should know about the atomic structures also, like how is FCC and body-centered cubic, etc. As I said, stainless steel brackets form the mainstay of the brackets. They are made from metal injection molding, casting, 3D printing. There are a lot of research aspects involved in that, like multiple prescriptions, designs, bond strength, mesh, best metal leach, all these are important. Another type of stainless steel brackets are self-ligating brackets. They are of different size. They have got a latch or spring or something. In fact, our paper was the first paper which categorized self-ligating brackets into active and passive when we published it in American Journal of Orthodontics in 2009. And with SEM we showed that how the clips adapts with the wire, and how do they express the torque and other features of the pre-adjust appliance. However, current evidences are in respect of self-ligating brackets are not in favor of reducing chair side time. The advantages which were cited in the past have not come to so far. So we have a dissertation done in our institute by Major Manu Pressure. So it showed that it doesn't increase the transverse dimensions as such. Another type of bracket which we are using, I mentioned about 8% of nickel in the orthodontic brackets. So that can cause an allergy to some patients, or patients who are on MRI therapy and all. You will have to substitute the stainless steel. In that case, you can use grade 2 commercial pure titanium alloys or titanium 6Al4 and 1ADM alloy. Intuitively customized brackets, as with artificial intelligence coming up, we'll have more of insignia systems and others. In that case, the plan is like scan, design, build, and bond. Lingual appliances are there with the coming of the other type of aesthetic appliances like clear aligners. It has gone into a lit of backside, but at the same time, from a metal perspective, you should know that the pads contain precious metal alloys, and the Win appliance is a 3D. Again, it's a proprietary. I think it's somewhat of a precious metal combination only. However, the aesthetic concerns of metal brackets have led to the development of plastic and ceramic brackets. This was a development in the 1980s when you had a lot of plastic brackets and ceramic brackets. Both came in 1980s, but it didn't sustain time. Plastic brackets have gone into oblivion, and ceramic brackets are there with two formats of the aluminum single and polycrystalline brackets. they give transparency. And ceramics are, they're glass, they're brittle. So they probably have got a chemical interaction with the wire. They try to put a metal slot in between to avoid this interaction with the wire. And there are issues of breakages, opposing to the enamel abrasion, etc. The molecular structure of these ceramic brackets are got a thick array of atoms, and they have got a high crystal density, high bonding energy, directional characteristics, sharp edges and corners. They create a bond. This is a structure which looks like this: crystalline and non-crystalline structures. The crack is impeded by the grain. For monocrystalline, you will have so many grains, and one single grain, and polycrystalline, you have so many grains. So the traversing those grains will determine the crack characteristic. And when it is a wet atmosphere, that is at formula, the sigma critical, which tells about the breakage, at what point it will sustain a fracture. So to give a general appraisal of what is the strength of the brackets, like ceramic is at the top, and your titanium and stainless steel coming almost same, and plastic in the last. So I said about our research and development of some biomaterials from the Indian missile program. So we were the first to report on a transparent ceramic that was from the missile, which can be fabricated, and we are in the final step of converting it into a bracket so that it's likely to look like a transparent one, better than a ceramic. This is to give us some idea of the in vitro physicochemical characterization which we do, and you do a lot of characterization, then only you can get it into the category of the accepted biomaterials. And some amount of cytotoxicity studies which we do, you can see that those are the MTT assays, apoptosis can be determined by the propidium apoptosis, genotoxicity by micronuclei, etc. And after the in vitro test, normally you have to do the in vivo biochemical evaluation. It has got different levels like skin irritation, intracutaneous reactivity, intradermal reactivity, and then systemic toxicity is done by several levels. These are all ISO standards are there. If you are interested, you can note down. So this is the way you undertake studies and implantation studies, and then it is done again in a different set of ISO categories. So these are some of the images which shows when we are, since our area of interest is like cranial area, so we have to do it in the cranial bone, because in molecular level the cranial bone may differ from the other long bones. These are the experiments on the mandible, and these are experiments with the PET-CT. When we connect the bone formation, to evaluate the bone formation, you connect it with the tracers and the fluoride, 18 fluoride, etc., and you can show the glow signs which show the bone activation. That's in the mandible. This is histology in cranium, histology in mandible, and the general systemic biocompatibility. And then the same material which I am mentioning about, that we were making it into a transparent bracket, we made it into a porous structure to activate bone formation with a stem cell mediated mechanism. In fact, we were first to report it. We reported it in a very high impact paper in Biomaterials in Ceramics paper. And this is a concept like the material will become into a porous like structure, and you will impregnate stem cells into it, and both will be inserted into a cranial defect, and then the material will disintegrate or something. Only that the tissue will remain. That's the beauty of it. You don't have to do a second surgery. Because in armed forces we have a requirement of the bone, all the data with us is like Indian Army soldiers sustain injuries, and whenever there is a whole body trauma, 75% will be whole body, 25% inevitably will be craniofacial area, and that's the data which is coming from the ongoing Russian-Ukraine conflict and the Israel-Palestinian conflicts also. The army really regularly collects this data, and so there is craniofacial bone loss is there. So this is the particular scaffold which we made from that ceramic, the same ceramic I mentioned before, so that's patented now. So, and in fact, we were the first to report the stem cells from the pulp, orthodontically extracted premolar teeth. We were the first to report in the country, and we established cell line nodes on that and integrated the cells, the stem cells, into the scaffold which we developed. So the interaction, if you see like this, this is the material, and the green are the cells. So that's why I told it's better terminology would be not material and biomaterial. Several other tests were done on seeing the efficacy of the perpetuation of the cells inside the scaffold, and this is a microarray data. It shows the gene heat maps, which explains what all genes are activated in that. These are the interactions between genes which we evaluate the protein-protein interactions. Like these are all done through bioinformatics, and then we validated the genes which are coming through acute RT-PCR, and the fold changes are established. And this schematic picture will explain you how the material has worked in towards initiating an osteogenesis. Like the scaffold, you can see the cells inside, and the cell scaffold interaction will activate several cascades of biomolecular pathways, which will cause several pathways which will hit the nucleus for initiating osteogenesis. These are other skin irritation intracutaneous tests in New Zealand white rabbits. These are the scaffolds are gone into this thing, and you can see that's very the interaction in this SEM level and special high-end stains. and this is a bone interaction. Can you see that on 6 months the material which I integrated into the defect has totally disintegrated? There is no material over there. It's a host bone which has formed over there. The stem cells have initiated a very positive host response. So what would be the clinical translation of this thing? We'll be doing the craniofacial regeneration of the bone. The concept would be like not to replace but to regenerate. And in orthodontic perspective, you will have secondary grafts being done with this metal. We all know that how difficult it is to get grafts integrated into the clefts area. So it would be possible. That is what we are aiming for. And currently we are activating the next generation sequencing work, like that will unravel more about the biometal cell interaction. And most of you may be aware of next generation sequencing. Otherwise you can read and understand an exciting world of gene and interaction. And it is like this. So a missile is there, and the missile can become a bracket, and missile can become a material to fill a bond defect. See the beauty of the biomaterials and how we can research and change the material into different formats of our use. Now coming to our archways, they are the main stay of our practice. You have got mainly 4 alloys: stainless steel and cobalt, common explained. Beta titanium. The numbers are difficult to understand, but I'll take it as a mnemonics. You can say that stainless steel and cobalt will have a modulus of velocity somewhere around 200, and others are in between. You can think like that. And this, how do these archways generate force? It's because of their elastic properties. Otherwise, the bracket cannot generate force, and the wire cannot generate force unless it has got elastic properties. So those elastic properties are represented by the force deflection curves. Suppose you all of you are supposed to know all these variables which are there: strength, stiffness, range, all these things. And how do these elastic properties are explained? It's explained on the basis of a beam geometry. It can be an unsupported thing, just supported thing, or fully integrated thing. So the formation or configurations will make the differences in force according to the mode of attachment. See, a clinical example would be a finger spring, look at cantilever and improved spring with the loops, multi-stranded bias. And the same, the hook, deflection. If you see for nickel titanium wire, it has got very flat curves because you know the phase transition. I mentioned about in stainless steel before. It happens in different temperatures, but in our case, it happens in the intraorally in the same temperature. That is what you got a flat deflection curve. And every time when you retie a nickel titanium wire, the deflection or the coming back is in a different direction. There is energy difference also. So you can use the same nickel titanium wire for some more time. What are the structures? It has got a martensite stability structure, a martensite active or thermoactive nitride. This is the nitride which is known as copper nitride or something, which will have, when we heat it and put it, it change. And the nitride which you normally buy from the brown market is austenitic nitride. How do you evaluate all these changes? How do you evaluate it? You can study it to a deflection curve, doing a physical experiment. Otherwise, you can do an X-ray diffraction study or differential scanning calorimeter. I'll explain it. How is it done? Before that, again, I mentioned about the structure of the stainless steel, but at the same time, you see austenitic martensitic structure of the nickel titanium wire also. You can read about it in detail. The uniqueness of nitride is like that the transition between two structures reversible occur at intraoral temperatures. Much of our understanding of the phase transition metals goes to these two people, Adolf Martens, that is why it is martensitic phase, and Sir William Chandler from Austin, from English metallurgist, to which austenitic phase is. We are indebted to them for their phenomenal work. Yesterday, Dr. Krishna Naik was asking about the same cross-section of the wire. They don't give the same torque. You know why? Because of the material properties. You see that graph, nickel titanium wire, irrespective of increasing the size from 1622 to 2125, it cannot go beyond 5. The stiffness values remains below 5. But at the same time, stainless steel goes high. That's the difference in the metal properties. Now we come to aesthetic or modified archways. Previously, a decade before, it was a fashion, or with more ceramic bracket or something coming up, we may be requiring ceramic wires which are aesthetic or which sustain corrosion. In fact, we did a lot of work on this nickel titanium wires and its characterization, very voluminous work, comprehensive work, and studied about its corrosion also. I will just show some SEM images. How do the nickel titanium wires and other wires look like? They've got a lot of defects, surface roughness. All these parameters are very important, considering the fact they are expressed in terms of root mean square surface values and interaction, friction, biofilm adhesion, retrieval analysis. A lot of biometal concepts are there related to this. And how do you estimate the composition? For example, you buy a nickel titanium on the market, you want to know whether it contains the requisite amount of nickel titanium. You can do an energy dispersive analysis. So you can do a Raman spectroscopy. It's from the CV Raman, and your Rayleigh line and other lines, you know how to determine the exact configuration. You all can study about the surface analysis using atomic force microscopy and profilometry. This will give an idea of about how the structures, biometal structures look like. This is important when they go inside the body. And we did a lot of corrosion studies also, because oral cavity is a very highly challenging atmosphere. So the material undergo corrosion. So this is open circuit potential and corresponding breakdown potentials. You apply it and you can determine it. It was published in Anglo Orthodontist. And we did a lot of work on the shape memory also. This came in a very high impact journal in Bullet of Material Science. How we evaluated phase transition. I mentioned about that in differential scanning calorimetry, you can estimate. You give some heat energy into that thing, and depending on the energy changes, like in terms of enthalpy values, you can determine at what point of time this material undergoes a crystallographic change from the martensitic to austenitic or austenitic to martensic. These are the values, and then you can find out those the phases which are generated during this heat heat giving. You can see, can you see that peaks, austenitic peaks, and these are determined by JCPD files. That's Joint Committee on Powder Diffraction Status. The International Chemistry Society studies peaks. So they give us the data. You can compare it to the library and understand what sort of phases are there. We have got archways for fixed retention. And to give an overview of this thing, what all sizes are there in archways? You should know about it. And previously, R.P. Kusi and others have brought about the nomogram. Now that is not much discussed. So on fingertips, you can say that stainless steel has got a stiffness value related to 1, and others will be compared against this. We cannot live without temporary anchorage devices these days, because it has enhanced our anchorage capacities. What are the materials? Stainless steel and titanium. Most of you may be aware of this stability graph. If not, what are the material advantages or additions which we are planning to get in this thing is like the abutments may change, new metals may come there to facilitate tooth movement. These are plates, that is basically from stainless steel and titanium only. But if you have more other components in this thing, you may have nickel, titanium, etc. getting joined on that thing. Elastomers are also very important, and we cannot live without E-chains to move tooth fast. They are all made from polymeric materials with a varying composition, molecular weights, and degree of cross-linking. But the main problem is with the elastomers is that the force decay after some time. And we in fact did a very extensive study on this elastomer force degradation. That was the world first reporting on that thing. Like we connected the molecular structure of the polymers with the glass transition temperature or storage modulus, through which we could say that understand how the molecular structure of the particular elastomeric chain would be determining the force decay. Aligners is the topic of the day. What are the three main characters? You have got biomaterials, attachments, and software. Yesterday, Dr. Abhishek was explained about it in detail. The parameters from a biometric perspective is high modulus of elasticity, high hardness, high degree of transcriptance, transparency, and high creep resistance are the parameters which we were looking for. Invisalign had improved upon their product. They have got now force decay values which are very sustaining and not rapidly decaying. Variations in aligner properties are mostly because of the biomaterials or polymers which are used, like polyethylene, polypropylene, PETG, etc., and thermoforming procedures also. In fact, you can see that the niter-waves have got a range of action of several millimeters. Aligners would be like 0.2 mm. That's why you change the material quite often. But still, you can determine the modulus of elasticity from that. And what are the intraoral alterations of orthodontic aligners? They undergo visible changes, surface roughness, deterioration of two-phase, and loss of strength and force. All this happens. These are the current conventional attachments, optimized attachments. So like orthodontic adhesives are another major addition in our biomaterial category. 50 years profession is into reliable orthodontic bonding after the Bonoco's acid etching and Bowen's resin. Comfortably, we are able to get about 5 to 20 megapascals of bond strength. We are living in the 8th generation or universal adhesives for dentine bonding. Where the other categories are there: moisture sensitive primers, self-fetching primers. What does it contain? Mainly, most of the resins which are used are RMGS, resin modified glass ionomers. There is a deprotonization method which was said. There are new lights which have come up, LEDs, and like bonding to artificial tooth surfaces, like how do you bond to porcelain, zirconia, amalgam, composites. All have been given with a specific new primers have come for this material, and inevitably there will be an alumina sandblasting at the rate power of 7 kilogram per centimeter squared. There are adhesives for special adhesives for bonding, indirect bonding also, like your special adhesive. You can use any type of thing, and fixed rigidness, they say that we should be using fiber reinforced composites to have more retention. Blue light is the LED light which is generated from semiconductors. That is what being used for heat active polymerization. But a lot of new reports coming about the blue light. It can have disturbing mitochondrial function and activating reactive oxygen species, etc. But this is very good in the terms of it synergizes very well with the camphoroquinone, the photoinitiator molecule in that. White surface lesions are another potential problem in orthodontic treatment due to demineralization or monomer leach. But this mostly happens because of the filler content is less in orthodontic adhesives. So there is a lot of leach, and you can have this problem. Then the preventive fluoride program is there. You have a CPP, ACCPF or sodium fluoride, etc. If the white lesions are established, then you should wait for some time and then only activate this thing. These are the structures. Most of the resins contain bis-GMA or the resin. That molecule contains another molecule known as bisphenol A. But now it is now suggested advised as an endocrine disrupting chemical. So this is a structure. It has got two phenyl rings. This is the culprit, and this is said to be can be a cancerogenic molecule. The other structures are like ormoses with our linear SOSNs and silyranes with SOSNs. So we recently developed a new filling material, absolutely new filling material, which does not contain. It's patented now. It does not contain bis-GMA. It does not has got any polymerization shrinkage. It's almost equal to 0. It doesn't require a primer. It releases fluoride. So it's an all-in-one material. It will be scheduled for a technology transfer by Government of India shortly. The animal experiments have also been completed. Now, in the backdrop of COVID, aerosol is an issue. It happened when we boned and when deboned, and that's the solid particulates 100 micrometers in diameters. They have the potential. Again, the problem bisphenol is there, and plus the material, the particulate per se can be a problem. At the same time, it can be a problem for transmitting infective agents. The facts, something about we have got better understanding of like inhalable fraction, thoracic fraction, respirable fraction, workplace exposure limits, and methods to mitigate that. And the last of our recent research and innovation was like we use a lot of CBCT radiation assisted investigations. The doctor is also not wearing a thyroid collar, and the patient is also not wearing a thyroid collar. Thyroid gland is the most radiosensitive gland in the human body. It is constantly exposed in orthodontic practice in other ways also. So lightweight, lead-free, human and environment-friendly material has been recently patented and approved by the government. So we will be doing with a lead assisted radiation blocking, and will be going into lead-free material. So this is one radiation image of the thing. Can you see that it shows the radiation attenuation of this material? It performs as equal to a lead thyroid collar. All these things works with patents and has been approved and acknowledged by Government of India in patents and awards. And we have humbly contributed to the Make in India to have quality products in our country also. So by learning and investing in orthodontic materials, we have better understanding what are the benefits, better understanding of the hardware we practice. We can have predictable results. We can do good to our patients. We are safe, we are ethical, and most important, we can innovate. Are you ready for the scientific practice of orthodontic biometals is the question which I like to leave here. And this is my mail, you can contact, and this is my department, my faculty, students. Jai Hind, thanks.
Thank you, sir. Over to the chairperson.
Thank you very much, sir. And it was quite a lucid and a quick recap of almost all the biomaterials in half an hour, and quite informative for
most of the audience, especially the students. So, any questions from the audience? Especially the students, the first year MDS, the PG students. Sir, there are no questions, but if you permit, sir, can you liked it? Sir. So there are no questions for the audience, but I have one small question, sir. I'm very inquisitive to ask you, sir, because I don't know how many people know, sir, has substantially worked with in collaboration with the DRDO, and numerous research, sir, has done. Among those, sir, regarding that mesenchymal stem cell extraction and creating the scaffold, what are your experiences in rehabilitating the defects of the cleft? One, and second thing, does this also have any correlation with the volume of the defects, sir, or any human studies, especially the mesenchymal stem cell from the dental pulp, sir?
Yeah, like the mesenchymal stem cells from the pulp was our objective of saying initially our control material was the mesenchymal stem cell from the bone marrow.
Sir.
Okay, as you know, bone marrow contains two components of the HSC cells, the hematopoietic stem cells, were the cells which are used for transplanting for other blood discretions and all. And here this is the mesenchymal stem cells. That was a control. It worked well. In fact, our pulp cells performed better than that thing. The defect size another thing, and we have not worked out. It has no human experiments have been done. We have now taken up a FMRC project for that to go into then in association with the RR hospital.
So the what was the approximate, like in animal studies, what was the approximate of the mean, the volume you could able to rehabilitate, sir?
We the sections which we cut were 5 into 5 5 mm into 5 mm defects.
Sir.
In multiple places, because you know in cranial bone we have to see it in the rat cranium and the mandible, so the bone size is very less. And when we came into the New Zealand white rabbits in long bones, it was 15 into 15 mm sizes.
Thank you very much, sir. Thank you, sir.
I would request the chairperson to please felicitate Manu Krishnan, sir. I would request Dr. Prabhakar Krishnan, sir, to please come on stage and felicitate our chairperson. Thank you, sir. Our next speaker for today is Dr. Prabhakar Krishnan, sir, Professor and Head, Vinayaka's Mission Shankaracharya Dental College, Tamil Nadu. He has specialized in surgical orthodontics, cleft orthodontics, and evidence-based clinical practice, along with 5 publications in PubMed Index journals. He's currently pursuing PhD in smile aesthetics. He has won the Best Paper Research at the first International Orthodontics Symposium, Indian Orthodontic Society with Eurasian Association of Orthodontics at Kurk Institute of Dental Sciences. He has been a guest lecturer at various institutes, national and international conferences. Today, he will be briefing us about genomic insights in orthodontics. We welcome you here, sir. The chair for this session is Dr. Shantanu Khatri, sir. Sir is the professor and a PG guide, Department of Orthodontics, Career PG Institute of Dental Sciences, Lucknow, with an academic experience of more than 15 years with various national and international journal publications. Sir, I would request you to come on stage, please. Over to you, sir.
Good morning, everyone. Am I audible? Yeah. Yes. No. So thank you for the very nice introduction. Thanks a lot. So at the outset, I would like to thank IOS 27th IOS PG Student Convention, the organizing committee, Shanava sir, Prakash sir, Dr. Robin, Dr. Prachi. All of you have done a wonderful job, and it's been an honor here, honor to be present here and giving a lecture on genomics insights in orthodontics. Now, it's basically a conceptual presentation. So I hail from this institute, Vinayaka Mission Shankaracharya Dental College from the city of Salem, Tamil Nadu, under the ambit of VMRF DU, which is deemed to be university. So H.L. Mencken, he was a satirist. He was a very famous journalist. He passed away in 1950s, but he's known for his very intellectually brilliant comments. And he once said that for every complex problem there is an answer that is clear, simple, and wrong. So all of us know that the malocclusion, what we deal in day-to-day orthodontics, is definitely complex. So malocclusion, that it arises from combined interaction of genetic and environmental, not always a summative, not always a summation, but a different kind of interactions are there. So there are two important key considerations for us: identify the cause of the problem before attempting treatment, and identify factors that can influence the treatment outcome. So for you students, you should always be able to relate treatment outcomes with Jackson's triad, if you put it in a very simple words. So Human Genome Project, it was finished in 2003. We know it was done because it was done basically to identify, map, and sequence the entire human genome from a physical and a structural standpoint. So now we know where human genome contains about 25,000 genes, and an average gene is about 3,000 nucleotide base pairs in length. So DNA, double helix structure, 3.2 billion, approximately 3.2 billion chemical nucleotide base pairs, and we all know that A pairs with T and C pairs with G. Chromosome, approximately 50 to 250 million base pairs, and the DNA is packed into these chromosomes because of the influence of histones and nucleosomes, and the histones actually regulate when and where the cells and how they will use the portions of the DNA. So we know 46 chromosomes, 22 homologous pairs, which are called as autosomes. They are homologous because they are similar in size and structure. The autosomes are actually numbered from 1 to 22 based on the size. I mean, the first one is the largest, second one is the smallest subsequently, and when you go to 20, 21, 22, they are the smallest of they are the smallest. Now we also have one pair of sex chromosomes, which are homologous in females, XX, and partly homologous in males, XY. Now when I spoke about structure, the basically similar in structure, autosomes, the autosomal pairs are similar in structure, means the position of the centromere, the long arm, and the short arm. The short arm is called a P arm. It is derived from a French word called petit, that meaning small, and Q arm is a longer arm, which means tail or Q. So a gene is a smallest physical and functional unit of inheritance, the complete DNA sequence that codes for synthesis of a specific polypeptide. Now, genes at the same locus on a pair of homologous chromosomes are called alleles. So assume there's a locus D, multiple locus this picture shows. Each chromosome which forms an autosomal pair, one is there from the maternal origin and another one is of paternal origin. So the gene which is present, let's say at a locus D from the maternal origin, is allele 1 and allele 2 from the paternal origin. So general structure of the gene, coming to general structure of gene, complete DNA sequence that codes for synthesis of a specific polypeptide. Genes can also code for, they can also act as a code for formation of messenger RNA, synthesis of and a synthesis of specific RNA molecules like tRNA, rRNA, and non-coding regulatory RNA. Now, basically, a gene has three important parts. There is a white region, which is a non-coding area. Then you have the exons, and in between exons, there are introns. Now, only the exon portion of the genes code for a particular polypeptide. So, coming to the genotype. So, genotype is a combination of alleles at a given locus within the gene. We are not talking about phenotype or trait, or observable characteristics, which are outcome of a gene. We are just talking about the gene, basically. So they can be homozygous or heterozygous. If the alleles are same, they're A, capital AA. If the alleles are different, A and small a, and if the alleles are similar again, but has a recessive inheritance, then represented by a small h. So there can be, of course, gene can undergo mutations. So if there is only one single nucleotide base pair change, like from, when you see, if you can see that G when you compare gene allele 2 and allele 1, the G is replaced by A in the allele 1, which is of maternal origin. So such kind of mutations are called as single nucleotide polymorphisms. They're also the pronunciation is SNPs rather than SNP. So chromosomal abnormalities, single nucleotide polymorphism. Large abnormalities can be there: deletions, insertions, translocations, and duplications. The best method to study them is karyotyping or genomic hybridization. Small scale variations can be studied by linkage and association analysis within families, and in those members who have similar phenotypic expression or similar observable characteristics. Within large populations, if you want to study the genotype, you have to use association analysis. We'll come to little later what you mean by linkage and association analysis. So what's a phenotype? Phenotype is observable properties, measurable features, physical characteristics of an individual. Now, it's a summation of effects of genotype plus environment. For let's say, for example, disease susceptibility. A blood type is a phenotype, but blood group A or B is a trait. So, what are human physical traits? A particular aspect or characteristics of an phenotype that can be inherited. For example, eye color, height, behavioral traits, mandibular jaw size can be a trait, and these traits can be monogenic or polygenic depending on what basis they are inherited. So a simple example, a melanocortin 1 receptor gene. These examples are from your textbooks, Graeber-Wenestal. So a gene can be expressed in various tissues. So depending on which tissue it is getting expressed in, that the traits will also change. For example, if this particular gene is getting expressed in hair, it gives rise to a red hair, whereas in the skin, it gives rise to freckles. Freckles are small brownish pigments, especially more common in Caucasians, especially when they are exposed to sun. So this particular trait of red hair is the inheritance pattern is autosomal recessive, whereas freckles have an inheritance pattern of autosomal dominant. So it's not the gene which is we're talking about here; it's a trait when we're talking about inheritance. Similarly, the same gene we are just discussing about monogenic inheritance pattern of MCR1, MC1R. So it can also give rise to a complex trait. Now, this particular study from Journal of American Dental Association has found out that MC1R gene variants and their phenotype, red hair especially, color is associated with increased dental care related anxiety and fear. So coming to modes of inheritance, can be monogenic traits or which follows about Mendelian pattern of inheritance. Now these traits are usually discrete, they're qualitative, they're very dichotomous, either it's present or no, but traits can still be variable. Examples are amelogenesis imperfecta and cleidocranial dysplasia. If you want to go and do a further study on these monogenic traits, you can visit your website called omim.org. Now, in monogenic or Mendelian traits, the predominantly of single gene affects the trait. But even then, a small number of environmental factors and even a small number of other modifying genes can also affect the expression of a particular trait. So it's not always exactly a monogenic. Even then, there are influenced by few number of other genes and environmental factors. So these traits can be dominant versus dominant or recessive. Trait is present only when one copy of a particular allele is present. They called as a heterozygous allele pair is a dominant type of inheritance. Trait is only present when both alleles at a locus are the same, are homozygous or at autosomal recessive. So pedigree charts. Pedigree charts are basically family-based charts, family trees. The indication for constructing a pedigree chart is when patient's medical history indicates then when there is more than one member of immediate family is affected. Now, when you record a pedigree chart, you have to make sure that you record family history in first-degree relatives, that is siblings and parents, and second-degree relatives, half siblings, aunts, uncles, and grandparents, and third-degree relatives, first cousins. So basically, why we do that is to approximate the likelihood that a patient or a sibling may develop a trait. So pedigree charts, how do they help us? They help us in identifying patterns of inheritance. They help us in assessing risk. They help us in referring patients to physician and for genetic counseling. Genetic studies and gene mapping can be done by linkage analysis and association analysis, clinical diagnosis and treatment planning, and formulating personalized treatment strategies. They can help us to refer the patients to physician or clinical genetist. Now, these are some of the common conditions where you are required to actually refer a patient to a clinical genetist or a physician. Dental agenesis with features of an ectodermal dysplasia, taurodontism, or other radiographic findings, midface or malar hypoplasia, basically a class 3 jaw discrepancy, especially in patients who are having other medical findings, and if there is a developmental delay. So coming to autosomal dominant again, so trait, when you look at the family chart, a pedigree chart representing autosomal dominant trait, it's 50% of the offsprings will have the trait. Each offspring will have a 50% chance of inheriting that particular trait. If the parents are negative for the trait, offsprings will also be negative for the trait. So you can see in this picture, the middle generation, out of 4 children, 2 children are affected, and whenever the parent has a trait, there is a 50% chance that one of the children will also have a trait. So expressivity and penetrance. Now this particular concept is very important for postgraduate students, because look at the first line. I mean, all the individuals, there are about 10 individuals there. All the individuals will have the same genome, but there is an incomplete penetrance. That even if it's autosomal dominant, only 6 of them have that phenotype or observable characteristics. The rest of them do not have. So such kind of inheritance pattern, when you see, it's called incomplete penetrance, and it is a non-penetrance. It's called non-penetrance for that particular individual who has a genotype, but the phenotype is not expressed. Apart from incomplete penetrance, it can also have variable expressivity. Variable expressivity means degree of expression and severity of a particular trait. So the second line, you can see the dark blue circles are actually representing penetrance, or the particular individual is affected, whereas the light blue circle is decrease in the degree of expression of a particular trait. So expressivity and penetrance is you have to keep in mind when you take up any research projects concerning genetics. So variable expressivity vary in severity and expression of the same gene. Examples are class 3 hypodontia, osteonic imperfecta with type 1 collagen, craniosynostosis syndromes. So basically, monogenic traits, environments, and modifying genes affect the expression of genes, but to a very minor extent. So this is the first one is the pedigree chart of autosomal dominant with incomplete penetrance. You can see only one male child is affected in the second generation. Autosomal dominant trait with variable expressivity. Again, the light blue colors show you that the degree of expression of a particular phenotype is toned down. So coming to autosomal recessive traits, 2 causal allele copies of observable phenotype. It should be homozygous. The parents are usually carriers, and the children can express the traits. Now coming to X-linked recessive female carriers, males are typically affected, females can be carriers. Transmission can be from affected fathers to daughters, and transmission will be from the carriers to son. So coming to nature versus nurture, the debate which we ponder in day-to-day life. Growth and development, we know both genetic and environmental influence are there. Genetic factors are basically the DNA which we inherit. Environmental factors can be diet, stress, living conditions, or epigenetic landscape. When we talk about epigenetic landscape, what does it mean? It means that heritable changes which can occur which can cause changes in the structure of the chromatin. Now, these changes can directly influence how the genes and when the genes and where the genes are turned on or off. Epigenetics will determine what, when, and where the polypeptides will be made. Environmental factors can cause changes in DNA sequence through mutations, but can also alter gene expression through epigenetic regulation. So same diagram again, same picture again, example of a monogenic trait. Just extrapolate it, multiply this picture into many times, and you will get a picture which is representative of a complex trait. So you can see here there's single phenotype, single observable characteristics, but there are 4 genes involved here. Environmental factors affects the expression of or the synthesis of proteins from all of these genes, and each protein generated can affect expression of remaining genes also. And ultimately, the environmental factors as well as the proteins which are synthesized out of genetic code can also affect the expression of a phenotype. So the interactions are complex. So form follows function. The two articles, excellent articles by Melvin Moss. So what he says is neither genetic or epigenetic factors are sufficient. Only their integrated activities, interactive activities, provide necessary and sufficient influence on growth and development. Another example which will help you understand these forms versus function. These skeletal muscle cells, they can have type 1 or type 2 muscle fibers. Type 2 muscle fibers are fast contracting muscle fibers, whereas type 1 are slow contracting muscle fibers. So these muscle cells, skeletal muscle cells, have something called as myosin heavy chain protein isoforms, different forms of a particular gene. So how does this example, let us consider this example, masseter muscle fiber composition affecting a vertical dimension. In mandibular asymmetry, if there is a significant increase in type 2 muscle fiber area and area and frequency, the masseter muscle tissue in the masseter muscle tissue on the side, in the side of the, on the same side as deviation, you can have long faced and open bite phenotypes, inversely related to the size and proportion of type 2 muscle fibers and greater type 1 muscle fibers. Type 1 muscle fibers, remember, are slow contracting fibers, whereas type 2 are fast. So whenever there are greater area and frequency of type 2 muscle fibers, you will have a short face and deep bite phenotypes can be seen. And that is why deep bite corrections, especially in younger patients, are difficult to maintain. Sagittal, these in contrast, when you talking about sagittal dimension, muscle fiber composition, they vary to a lesser degree in class 3 mandibular prognathism, with the exception of type 2 and the other types of type 2. Class 3 deep bites, again increase of type 1 and 2 hybrid muscle fibers are also increased in class 3 deep bite cases, fast contracting fibers. So what are the methods to study genetic expressions, genotype, phenotypes and all? Twin studies is a very common method you must have come across, very frequently asked question in your exams. So it was given by Sir Francis Galton, basically a part of population genetics. It can be used to study influence of genes and environment on phenotypes. Comparison of monozygotic and dizygotic twins. It is used to partition variance of quantitative traits into environmental and genetic factors. Another method very similar to twin study is a twin half sib method, where basically step brothers and step sisters. So all these things, monozygotic. What are monozygotic? They originate from one fertilized egg that divides on later. It's identical in genetic makeup and sex, and any differences observed are due to environmental factors. Dizygotic, on the other hand, they originate from two separately fertilized eggs. They are different in genetic makeup. Half the pairs are also differently sexed, but observed differences can be due to genetic as well as environmental. So what are the problems or shortcomings of these twin studies? Assumptions of equal environment. Monozygotic twins may experience more similar environments or social environments than a dizygotic twin because of their difference in appearance among the dizygotic twins. Occurrence of twinning can itself be influenced by genetic and environmental factors. So there can always be biases when you are estimating heritability and environmental influences derived from the twin studies. Limited generalizability. Unique dynamics of twin relationship and shared experiences cannot be extrapolated to singleton individuals or a larger population. Difficulty in disentangling or separating your genetic and environmental factors also can be a shortcoming in such studies. Non-shared environmental influences may be difficult to measure accurately, and obviously in any twin studies, sample size is always a limitation. So some basic outcomes of monozygotic twin studies over the years are variable and frequently insignificant variance of overbite, overjet, molar relation, posterior crossbite and rotations. Now genetics, and we also learned out of these twin studies is genetics definitely has some role to play in arch width, arch length and crown dimensions. So coming to the next type of study, which is linkage analysis versus association analysis. Now linkage refers to the physical proximity of a loci. For example, let's assume locus D and locus C are very closely placed. So whenever during gamete formation by the process of meiosis, recombination will not take place. These two locuses, the genes present at these two locuses will usually be inherited together. So probability of chromosomal recombination between two loci which are very close together is small. So recombination fraction is the probability that two DNA markers will appear in a new combination on a chromosome not seen in the parental generation. So recombination fraction for a closely linked genes will be less than 0.5. Two distant genes will have a high chances of getting recombined in during the process of meiosis. So that will have a recombination fraction of close to 0.5. Linkage analysis looks at the relationship between transmission of a locus and trait within families. Linkage analysis is basically done in families where there are multiple individuals, individuals showing a particular trait or a phenotype. So it can also be done in large extended families with multiple affected individuals, but it will be difficult for very rare diseases. On the other hand, association analysis. These are the methods. These are the methods which can be used to determine if a particular allele is more frequent in a group of individuals with diseases compared to a control group. So very similar to a case control study. Again, the requirement will be that you have to closely match the cases and controls to minimize the false positives. Can be done in large extended families also. Apart from linkage analysis, large extended families, you can do a association analysis as well. So out of linkage and association analysis, you will get something called as a heritability estimates. Now the problem what we face is that we often what you call expect too much out of heritability estimates. The question will be, do they actually influence our decision making in day-to-day orthodontics? How we treat a case orthodontically? Can they influence our treatment options? Will it define limits of tooth movement? Does it help us to predict jaw growth, and does it help us to predict growth modulation outcomes? So what are heritability estimates? These are the quality of a characteristics being transmissible from parent to offspring. It's a measure of how well differences in people's genes account for differences in the traits or phenotypes. Now but we know that the ability of the patient to respond to changes in environment, in our case it's the treatment which we provide, will define these limits. Now H square is a heritability estimate. It's a collective parameter. It's actually meaningless to discuss about a trait in an individual. Only it's a population. It's an outcome of a population study. You can say that out of this population you have heritability of your particular trait has got a certain heritability. So if you put it in a mathematical equation, heritable estimate can be equated to G divided by G plus E. G is your variation of trait size among the individuals because of different genotypes, whereas E is your environmental variations caused by the environment. So G plus E will be the total phenotypic variation. So how much the genotype is responsible for a particular trait divided by the total summation of genetics and environment factors. There are two important concepts which actually affect these things. One is dominance. We already saw allele interactions. So suppose if you have two alleles, let's say A enhances the trait size. Consider height. So A is responsible for increased height, whereas B does not enhance height. So ideally, if the influence was summation, just like A plus B, the intermediate genotype of AB should have a height which is exactly in the middle between a person having a genotype AA and a person having genotype BB. But in nature, it is not so. AB will be closer to a person who is having a genotype of AA because of dominant expression, inheritance of a particular trait. Epistasis is gene-gene interactions at two or more loci. So aggregate of all gene-gene interactions can be represented by a letter I. Dominance is D. So actually the original equation where the phenotype was a summation of G plus E can be extrapolated because of complex inheritance patterns. So the equation can be like A plus D plus I plus E plus E. The small e represents your errors in your statistics. Then D is dominance, and E is your environmental effects. So what are environmental covariates? Now siblings, and the question is, siblings and twins, do they share environment? When the siblings live together, obviously they have similar environments, so the phenotypes can converge. So environmental covariation can lead to positive and substantial heritability estimates in the absence of any genetic contribution at all. Now, environment can affect genotypes in two ways. So, genotype environment correlations and genotype environment interactions. For example, let's assume one way you can be smart is to have a smart parents. If your parents are intellectually very intellectually able, they had good disposable incomes in their families, good education, so the chances are high that the children will also have similar environment. Uh, so this kind of correlations are your genetic environment correlations. Genotype environment interactions. Now here a particular genotype can perform well in particular environment. For example, let's say if your alleles, if your genotype is such that you are indifferent to pollen allergy, or you don't develop very easily chronic allergic rhinitis, there are high chances that you will not have enlarged adenoids. There are high chances that you will not develop a mouth breathing habit. So these type of interactions are genotype environment interactions. Also assume that, let's say, you have genes which make you susceptible to hypertension. So a bad diet of high cholesterol and high fat diet will make you hypertensive. So growth differences during puberty. So pubertal growth spurt, we know it's affected by sex steroids, growth hormones, insulin-like growth factor, and other endocrine, paracrine, or autocrine factors. One important parameter called TE ratio, that is testosterone estrogen ratio. This is very critical in sex-indexed male facial characteristics. So when the T is high, you will have a good male facial characteristics like growth of cheekbones, mandible, chin, strong jawline, lengthening of the face. Now, aromatase is an enzyme which actually is a key enzyme in aromatase synthesis. It catalyzes the final rate limiting step in conversion of testosterone to androstenedione to estradiol and estrone. Now, a particular gene which affects the aromatase is CYP19A1. Now, certain alleles of these gene are associated with increased growth velocity. This particular study, which was done by Shushu et al., James Hartsfield, actually a person who is very well known in for genetic studies in orthodontics. Now they found out then when persons who males especially if they have inherited certain alleles of CYP19A1 gene, the increments of growth in maxilla it was 1.5 millimeter per year more than the controls, whereas in mandible it was 2.5 millimeter per year more than the controls. Other genes, other proteins or genes which can affect your growth is growth hormone receptor gene. It's very important in difference, causing difference in adult mandibular ramus length, and also P56 IT allele. Significant correlation between shorter ramus height in Japanese women were found. So can these things affect treatment outcomes? Definitely. So individuals which have a particular allele of that the gene CYP19A1 gene can have a better response to myofunctional therapy or orthopedic treatment. So coming to skeletal variations and malocclusions, class 3 we know short maxilla, long mandible, or both. Usually they are autosomal dominant. They have variable expressivity. That means difference in severity of expression or severity of severity and expression of mandibular excess or maxillary deficiency, and of course incomplete penetrance can be seen. So one way how can we arrive at day-to-day treatment guidelines out of genetics is we should do phenotypic characterization of jaw discrepancies. Now, a particular article published in Angle Orthodontics in 2006. What they do is basically they do a population study. They divide the clusters in a particular population into various clusters, and they try to find genetic linkages to the phenotypes, particular types of class 3s. Now, whichever particular type of class 3s is showing better responses to better skeletal correction, better responses to orthopedic treatment. Now, we will know that if that particular associated gene is present in other individuals, there can be a predictive advantage of such genetic studies. So gene linkage studies to identify gene loci for specific traits. Clinical importance, those subtypes can be treated differently or different outcomes of or retention protocols. So genetic polymorphisms underlying the skeletal class 3 phenotype by Cruz et al. Polymorphism in myosin heavy isoforms could be used as a marker for genetic susceptibility to class 3 malocclusion with mandibular prognathism. polymorphisms in growth hormone receptor as well as fibroblast growth factor were associated with maxillomandibular deficiencies. Now, coming to class 2 division 2, twin study has shown us that all the most of the monozygotic twins are concordant for class 2 division 2 against very small percentage, that's about 10.8 percentage. So strong association of class 2 division 2 with dental developmental anomalies, statistically significant reduction in mesodistal width of maxillary central incisor was found in patients having class 2 division 2. Coming to external apical,
almost finished.
Coming to external apical root resorption, we know the commonest reason is excessive force, but root resorption is also seen in non-orthodontic patients, patients with missing teeth, increased periodontal probing depths, reduced crestal bone heights, bruxism, and other habits. It's always important to learn the disease pathways to actually to understand genetic influences or environmental influence on the genes. So clearly, I'm very confused. Maybe you could explain it to me. So what are the insights finally? Identify early warning signs of aberrant conditions in patients with malocclusion, personalized, rational, effective treatment programs. You can formulate individualized treatment strategies for the patients, orthopedic treatment of borderline discrepancies to avoid later surgery, and you can also avoid early orthopedics in case where relapse is certain. So that means you can plan out surgery. So these are some of the advantages of studying genetic influences and epigenetic influence on malocclusion. Thanks a lot for your patient hearing.
Thank you. I'm sure everybody would have brushed up with their basics.
Yeah.
Any questions, anyone?
Thank you, sir. I would request our chairperson to please felicitate sir. I would request Shrikant Shindre sir to please felicitate our chairperson. Our next speaker for today is Dr. Shrikant Shinde sir. Sir is the professor, Department of Orthodontics, Oxford Dental College, Bangalore, Karnataka. He's a postgraduate student from the prestigious Institute of SDM Dental College, Dharwad. Sir is also the M.Orth RCPS from London and M.Orth RCS Edinburgh. We welcome you here, sir. The session is going to be chaired by Dr. Trilokya Bharali, sir. He's a postgraduate from A.B. Shetty Dental College, Mangalore, and presently working as a professor in Department of Orthodontics in Regional Dental College, Guwahati. Also, Dr. Akhil Agarwal, sir. He's a life member of Indian Orthodontic Society and the World Federation of Orthodontics. He has been awarded the International Observership by the University of Australia in association with the IOS. I would request our chairpersons to please come on stage. Over to you, sir.
Good morning to one and all present over here. The title for my presentation today is Class 2 Non-Extraction Treatment, which is simplified with the passive self-ligation, utilizing the power of the transverse development. So when you are talking about the Class 2 correction, if you go through the Graeber textbook, you can find probably 25 to 30 methods by which you can correct Class 2s. And there is nothing called a right or a wrong method. But the most important thing when you talk about the class 2 correction is you need to control the labiolingual inclination of both the upper and lower anteriors for the best aesthetics and retention. Well, I started my journey with the blessing of my teacher, my mentor, Dr. V.P. Jaide. Sir is a living legend of Indian Orthodontic Society, and he continues to inspire us. So before I start to speak about the passive self-ligation, I'm going to just show you. Primarily, I was taught in beg appliance. I'm trained in beg appliance. So this is a case. I'm just trying to show you one simple case treated with a beg appliance. A 14-year-old girl. Typical features of bimaxillary protrusion. You can see the bimaxillary protrusion over there with a bit of a crowding in both the arches. So it's a simple bite opening bends given, which is a typical of a beg stage 1, and then there is a controlled tipping happening with the help of some braided rectangular wires, which is giving you torquing, and there are some uprighting springs on the canine. So you're trying to do a controlled retraction during your stage 2, so that you don't want your stage 3 to be doing a lot of uprighting and torquing. And this is how she finished. This is the comparison. So when you can, why am I showing this case is just to tell you the fact that it is a non-programmed appliance. There is no tip built into it. There is no torque, no in out. But look at, she's got a stunning smile. At the end of the day, what difference does it make whether you are done a patient with a aligner or a passive self-ligation? What stays with this girl for the rest of her life is the beautiful smile she has. And I think that is where we all need to take a credit for the fact that we are orthodontists and we are great at our skills. We have the ability to design smiles like this, and I think that is the tribute to all the orthodontists who are here. So when we talk about class 2 malocclusion, most of the time we spend a lot of time during our clinical examination over the sagittal and the vertical dimensions, but somehow the transverse dimension doesn't get the same due which needs to be given during the clinical examination. So just to support this article, a beautiful article written by Dr. Samir Bishara in 2013 AGO. So if you look at what is it trying to say is, suppose you have a constriction in the dental arches during deciduous to mixed dentition or mixed to adult dentition, the constriction doesn't self-correct. That means if there is a constriction of the maxillary arch, it is going to remain the same way. It is not going to self-correct. And this constriction of the maxillary arch can have an impact on your mandibular arch as well. So it is very, very important that you pay attention to the transverse dimension. And I think that is where the passive self-ligation comes into play, that it starts to correct your transverse right from the word go. Probably this is the most important slide of my presentation. So if you're trying to get the best out of your passive self-ligation system, so you have three components. You have a passive self-ligation bracket, which is like a series of mini tubes bonded onto the teeth, wherein the wire can express itself. There is hardly any friction. There is enough freedom for the wire to move around. Then you have copper nitride wires. So because of the low load deflection rate, they are going to give you very less force for any given degree of deformation of the arch wire. So that is the beautiful thing about copper nitride arch wires. But the heart of passive self-ligation is going to be the disarticulation of the arches. Most of the time you don't get best of the passive self-ligation simply because you fail to do the disarticulation. The reason I am saying it is the heart of PSL for the simple reason is, until unless you don't do disarticulation, you are not able to reduce the masticatory forces which are acting on the dentition. Then only the copper nitride arch wires can give you low forces. Otherwise, if you fail to do the disarticulation, the masticatory forces are so high that they overpower your arch wire forces which are generated with the copper nitride arch wire. And the best thing about the disarticulation is it is going to give you the transverse development, which is not going to happen otherwise. So like I was telling you, there are 22 methods given in a a textbook regarding the class 2 correction. Most of my patients, what I'm going to show in upcoming slides are all adolescent patients, wherein I have simply used only the class 2 elastics, huge overjets, no spaces. So I came across a beautiful statement by Dr. Alan Bagdon that tats and elastics will replace the surgical scalpel in 21st century. And if you feel this statement has to be right, as a clinician, it is your responsibility to motivate and educate your patients to use the elastics. If you are looking at correcting big overjets, just with the help of class 2 elastics, and patients are not going to use the elastics, you are not going to get the best of it. So in the subsequent cases, I am going to show you the kind of a cooperation I got from my patients, which helped me to correct such great malocclusions. So we all know about this foot and shoe mechanism, that a wider shoe will allow the foot to assume its normal relationship. In the same way, if you have a maxilla which is wider, it is going to allow the mandible also to express in a more forward direction. So I call this as a foot and shoe mechanism revisited. That is, you're going to have a bracket which is generating a low friction. You have copper nitride arch wires which are generating low force. You are doing disarticulation, and then you are adding class 2 elastics in that environment. So what am I doing with my disarticulation, low force, low friction? I'm slowly allowing my upper arch to expand in a transverse direction, which is called as transverse development. They don't use the word expansion. We are talking about transverse development. And when you are using class 2 elastics, it is creating a favorable atmosphere for the mandible to assume a more forward position. So like I told you, most of my patients, what I'm going to use in this presentation are adolescent patients, somewhere between 13 to 15-year-old age. So you need to have certain strategies. What are the strategies you're going to have in order to be treating the adolescent patient? Support or augment the soft tissues, not reduce it. If you're treating a 13 or a 14-year-old patient, your whole focus should be to do a non-extraction treatment, because I know if I do a non-extraction treatment, I'm going to augment my soft tissues. If I'm going to do an extraction treatment, I'm going to reduce the soft tissue drape. Second, you need to understand the principles of growth and development and long-term changes expected in the both hard and soft tissues. If you're treating a 13 or a 14-year-old patient, we all know that a lot of nose growth and the chin growth is left. So if you're going to be planning an extraction treatment, you're going to be very, very careful about, you need to know how much the nose is going to grow, how much the chin is going to grow, how the patient is going to look with the extraction at the age of 20 years, at the age of 30 years, at the age of 40 years. Because we are trying to talk about treating a patient for the lifetime. So I need to be very, very sure about what I'm doing. Third is, you need to maintain or improve the incisor display. Probably this is the most important time. We know that as the age advances, the lip is lengthening. The incisor exposure is reducing. So if you are treating a 13 or a 14-year-old patient, don't use a mechanic which is going to intrude the maxillary incisors to a great extent. She may end up with no incisor display by the time she's 20. She's 20 and she might look like 40. So that is the concepts I'm talking about. So this is little bit about the arch wire sequencing. Very simple. You are going to start with the oven 4 copper nitride if you're doing PSL. Leave it for 10 weeks. 10 weeks of 14 by 25 copper nitride. So when you're on a 14 nitride, you are using a 5 by 16 class 2 elastic, and if you are on a 1425, you are using 3 by 16 class 2 elastic force. And these two sequence of arch wires in a non-extraction treatment will finish 70% of your treatment. And of course, you have major mechanics wherein you are going to use 1925 stainless steel, which is probably for the extraction cases. In a non-extraction case, PSL, you can as well finish with 1925 TMA. So with this background, I come to the case presentation. So this is the typical 14-year-old boy presenting with a bimaxillary protrusion. He's complaining of a forwardly placed teeth. He doesn't like his smile. He's got acute nasolabial angle, deep mentolabial sulcus, a convex profile. If you see, he's got a relationship on the right side, which is end-on, canine end-on. Left side, he's got almost class 2 molar canine, increased overjet of 7 to 8 millimeters, which is a typical of a class 2 division 1. Cephalometrically, his skeletal class 2. His upper incisors are proclined, lower incisors are also proclined, compensating for the skeletal class 2. His IMP is 106. This is his OPG, and his both the parents are MD radiologist, and so I had various plans in my mind whether I need to extract upper 4, finish the molar into class 2 relationship, or can I use a fixed functional appliance and try to do non-extraction treatment. But when I looked at his IMP, was 106. I was not too sure about how much the lower incisors will get proclined with the form of functional appliance I'm going to use. So I just started to use a non-extraction treatment for him to begin with the PSL. So we went ahead and used the standard talk prescription for him. And this is just 5 months into treatment. If you carefully see now, from a class 2 molar, I have a super class 1 molar relationship. So I have not done any IPR, nothing, just simple archwire changes, 10 weeks of ON4 copper nitride, 10 weeks of 1425, simple class 2 elastics. Look at the changes in the premolar area, little bit of a transverse development in the premolar area, leading to a wonderful change for this patient. These are his pre-finishing photographs showing very good overall changes. The soft tissue looks more relaxed, and you can see everything is started to soak in. I removed all the blue bite what I had used. This is his pre-finishing lateral ceph showing good inclinations of the teeth, and OPG showing good root parallelism, and this is how he finished. So you can see the change. Look at the, you get a good class 1 canine, class 1 molar, occlusion is socked in, see the change in the arch form, and this is what I call it as a Frankel effect of self-ligating appliance. Look at the profile. I'm not saying that everything has happened because of my treatment. Of course, nature also has helped me, but I have created an atmosphere wherein his mandible could express. So very happy with the change. So this is the overall changes. This is the changes in the arch form, which probably resulted in mandible posturing forward, comparison, beautiful change. And this is the superimposition showing a little bit of a mandibular relocation. See, in spite of the fact that the lower incisors are proclined, it is not showing on the soft tissue profile. That is the beauty of PSL. Things happen at such a low pace, at such a low force, your lower incisors don't get dumped. I'm not saying the teeth have not proclined. Lower incisors have proclined, but it is not manifesting in the soft tissue profile. I think that is the best thing to have happened to PSL. So when you look at a case like this, the decision to extract should not be based on your cephalometric readings. Okay, my cephalometric showing teeth are proclined, I'm going to extract. Not like that. How is the soft tissue drape? How this patient is going to look at the age of 20 years, 30 years? I think those are the things which overrides the cephalometric measurements. So there is another girl, 13-year-old girl presenting with a forwardly placed teeth. She doesn't like her smile. Typical of a class 2, you can see acute nasolabial angle, deep mentolabial sulcus. She has a convex profile. And if you look at, she's a typical of a class 2 division 1 subdivision. So everything on the left hand side is class 1, and right side, if you see, everything is class 2. Her lower dental midline is shifted to the class 2 side. So if you look at the etiology of class 2 division 1 subdivision, it's all about the occlusal prematurities which take place because of a constricted maxillary dentition, which does not allow the mandible to express. So that is where PSL comes into play. So having got the great results for the first patient, I just simply started with a non-extraction treatment. This is her cephalometrics, just showing that she is a skeletal class 2 with a vertical growth pattern. Upper incisors are proclined, so as lower incisors, compensating for the skeletal class 2. This is her OPG, and we just simply started with the non-extraction treatment. So this is a particular slide I got from Dr. David Sarwar's textbook. I'm a huge fan of David Sarwar. So we talk about taking a lot of records for our patients. You know, everything is becoming digital. People are talking about CBCT today, all those things. But I think the clinical examination trumps all the records we take in our profession, because nothing substitutes a good clinical examination. Many times we are doing a good clinical examination and history. So then you're forming a problem list, right? And to this problem list, you have a solutions. We all write treatment objectives and then corrections of those treatment objectives. And finally, you have a treatment goal. Many times we do that. But many times we do not write about the positive attributes which are there in the patient. Let us say a patient has a good incisal exposure at rest. Let's say a patient has a good smile arc. We don't make a note of it and write it somewhere in our treatment plan that yes, this is a good attribute and I'm supposed to be maintaining it throughout the course of the treatment. So I think this is what Dr. Sarva talks about, that not only problem list, pay attention to the good features which patient is already presenting to you. So same thing we did here. This particular patient has a deep bite and she has an incisal exposure which is normal. So I can't be intruding the upper incisors here. So as a result of that, I decided to place the upper incisor brackets very much gingival so as to maintain the smile arc. The whole of a deep bite is going to be corrected in this particular patient with the incisor intrusion and lower incisor intrusion and lower molar extrusion. So these are called as the aesthetic considerations you have to have during your diagnosis and treatment planning. Just because you have a deep bite, you can't be intruding both your upper and lower incisors. Which group of incisors you are going to intrude and why? We need to be clear about it. So simply we used a class 2 elastics. I am still on a 1425 copper niti. I am using a diagonal anterior elastic because the patient midline had shifted to the right side. These are her pre-finishing photographs. Her smile arc started to improve. The soft tissue profile is starting to show more relaxed. These are her intraorals, midline corrected, both the side, good interdigitation of the teeth. You can see the changes in the arch form in the premolar area. And this is how she finished. A wonderful consonant smile arc, good soft tissue profile, competency of the lips, and look at the occlusion. So, and at the same time, look at the soft tissues. The soft tissues are looking so very healthy, beautiful, and stable. So, see, nothing much has changed in orthodontics over the last 20-25 years. 25 years back when I was a PG student, I was attending, everybody still talked about getting midlines matching for your patient, get a good class 1 canine, get a good class 1 molar, good interdigitation of the teeth. Nothing much has changed. Even you talk about cases today, you talk about this. But what has changed is this. Today, the maxillary incisors drive the treatment plan. There was a time we spoke about mandibular incisors being placed upright over the basal bone. And the entire diagnosis and treatment planning was revolving around that. You know, first you need to find out how am I going to place my mandibular incisors upright over the basal bone, and then everything else revolves around it. But now I think this is what is going to be called as phase-driven orthodontics. That means I need to have definitely a good occlusion for my patient, and it should be coupled with a great aesthetics like this, and they are going to be the test of the time. So this is the comparison. You can see the comparison, midlines corrected beautifully, deep bite corrected. This is her OPG showing good root parallelism. And you can see some amount of mandibular relocation which happened because of the transverse development. So father is very happy patient at the end of the day. So here's the next patient who's 13-year-old boy. Is presenting with the typical features of a skeletal class 2 division 1 malocclusion, increased overjet of 7 to 8 millimeters, absolutely no spaces present anywhere. You have a deep bite, constricted arches. The moment you say case like this, first thing will come to your mind, maybe I need to do some extractions or a fixed functional appliance. I haven't done anything of that sort. He's a skeletal class 2. You can see the IMP is at 100. Lower incisors are procline, compensating for skeletal class 2. So similar plan. So what we did here was I used the high torque brackets for him in the upper arch and low torque in the lower, because I know that the class 2 elastics are going to dump the upper incisors. So to prevent that, I used high torque brackets, and the low torque is going to give me the control on the lower incisors. So this is just arrows showing how you use your high torque and low torque brackets. This is the best form of prescription you can use if you are doing class 2 corrections, high torque for the upper and low torque for the lowers. So simple class 2 elastic, similar mechanics, all the cases, just to show you, matter of 3-4 months, and this is how we started to show changes. These are its pre-finishing photographs. All there, you must be wondering, where did the overjet go? How did the molar become class 1? This is the power of transverse development of the passive self-ligating appliances, which just allows you to treat cases in a very smooth fashion. And this is how he finished. Look at the smile arc. It's a beautiful smile arc. He's a smart boy now. And I think this is the power of PSL in treating patients non-extraction. Wonderful. Same thing I was talking about. This is phase-driven orthodontics. You want to get a best possible aesthetics for your patient coupled with a beautiful occlusion like this, which can last lifetime. This is the comparison in the arch forms. And again, I would call this as a Frankel effect. I'm not saying that everything happens because of your treatment. This is the beauty, meaning you start non-extraction, the nature tends to help you, and you get a mandible which is so very good. This is the overall comparison. Another girl, my favorite patient, her name is Shreya. She is a 13-year-old girl presenting with a huge proclination of the teeth, typical class 2 patient, and increased overjet of 5 to 6 millimeters, Andonish relationship of molars and canines, and she's got some amount of crowding too. And if you look at a picture like this, you don't even need any cephalometric analysis to tell you that how much the teeth are proclined here. I don't have spaces. So when you see a cephalometrics like that, you're first tempted to think about extraction, maybe upper 4 alone, upper 4, lower 5, or a fixed functional appliance. So typical features, all again, we went ahead and used the standard torque prescription. So many a times, if you look at in PA, what is happening is we are standardizing the bracket placement from the incisal edges. You are taking a chart, bonding brackets for all your patient. I think that is one of the reason why the smile arcs get flattened with the conventional appliances. I think your bracket placement should be based on, you need to see the patient's incisal exposure at rest, see the patient's smiling picture, how much incisors are visible. Based on that, these two parameters decide as to where to place your brackets. Otherwise, if you keep placing the bracket as per the MBT chart, most often than not, you'll end up flattening the smile arcs. So these are her mid-treatment pictures, similar mechanics, simple class 2 elastics, 4 to 5 months into treatment, disarticulation of the arches, and look at the smile, wonderful smile. And I'll show you how much the mandible has grown. And many a times, adolescent patients, we talk about E-line. I think there is no role of E-line in a young patient because both the references what I'm taking, nose as well as chin, they are continuously growing. So in an adolescent patient, I think the role of E-line is very little. Don't take into consideration the E-line because the nose is also growing forward, the chin is growing forward. So look at the profile. These are the changes. What I wanted to show here, look at the amount of growth the mandible has undergone in a matter of 1 year. So that goes to show that we need to be very careful in treating a young and adolescent patients like this. This is a comparison. And look at the buccal carotid space, the role of, now you want to have a narrow buccal carotid spaces because for the greater aesthetics, you want to have a nice and a wider smile. So this is an article given by Jackrison. He stated that lingually inclined posterior teeth would increase the negative carotid and subsequently decrease the smilefulness. So narrower the buccal carotid, great is the smile arc. So this is the comparison of the arches, good overall changes, good occlusion, class 1 canine, class 1 molar, midline matching, and this is her smile. So you can just see the amount of mandible which has grown in a matter of years time. So this is a slide I got from a Chris Chang presentation. We are talking about a 10-year-old mandible here to 25-year-old mandible there. So if you are looking at the 75 to 80% of the patients what we are treating, they fall under this age group. And if this is the transformation the mandible can undergo from 10 to 25 years, I think we need to be careful about uh, uh, taking just cephalometric compilation and start extracting our patients, because look at this mandibular growth is affecting your class 2, class 3 cases. It is affecting extraction, non-extraction, deep bite, open bite, asymmetry. When you compile everything, it forms orthodontics. We are talking about entire thing what we do on our patients that starts to get affected if you don't pay attention to these details. So coming to a last case presentation, compared to all other patients, he is a 17-year-old boy. So I was not too sure whether whatever I did for the other 4 patients would work for him. I thought, let me just go ahead and see. He was a typical class 2 with a teethy smile. His lips are potentially competent. He had a acute nasolabial angle, a deep mentolabial sulcus. If you look at his intraoral pictures, everything is endonish, which is tending towards class 2, an increased overjet of 6 to 7 millimeters, bit of a constriction of the arches. This is his cephalometrics. He is a skeletal class 2. Both the upper and lower incisors are proclined. And he is a 17-year-old boy. This is his OPG, nothing great, showing erupting third molars. So we went ahead and used the torque prescription, which was standard in the upper and low torque in the lower, because I know that I am going to be using a lot of class 2 elastics. I don't want to procline my lower incisors. So in short, when you're talking about low torque, it's nothing but a labial root torque. So we went ahead. We used a simple mechanics, again, class 2 elastics throughout the course of a treatment. 10 weeks of 014 copper nitride, 10 weeks of 1425, and of course there is a disarticulation, which is so very important when it comes to PSL. And these are his pre-finishing extraorals. He started to show some changes. You can see already the endonish relationships have gone. You started to get more of a class 1 relationships, and this is how he finished. He got a wonderful smile arc. The lips are competent. He lost some amount of weight also during the treatment. As a result, he is looking like a different guy altogether. Wonderful aesthetics. That's what I'm talking about, that nothing much has changed. You want to do for all your patients, class 1 canine, class 1 molar, midlines matching. Look at the soft tissues. They have improved a lot. And this is the overall comparison. Look at the chin throat angle, This has happened probably because he lost weight, and he's looking like a smart boy now. This is his comparison. So I would say that the proportionality of maxillary incisors, the teeth were looking big here because the arches were constricted. The moment you expand the arches, the same teeth start to look smaller in size. That is the concept of transverse development. What can it do to the teeth? So this is the change in the arch form. This is his post-treatment ceph, showing good inclinations, good angulations on the OPGs, and superimposition changes. Yes, we have been able to procline his lower teeth. I'm not saying that teeth are not proclined, but it doesn't show on the soft tissues. That is the best thing with PSL. Your lower lip doesn't become procumbent. So that is the great advantage. So when I look at this, I was just asking myself what could have happened for all this correction. So you have a PSL bracket which is giving you low friction, copper nitride low force, disarticulation is giving you low masticatory forces. Combination of all this creates a muzzle deprogramming effect, leading to transverse development, which allows the tongue to posture more superiorly from an inferior position, because you have a constricted arch, tongue goes down. And all these things creates a second chance. It provides patient a second chance to develop a new neuromuscular balance, and that is what has happened in all the patients. The new neuromuscular balance was able to be developed. So when I did all these things, I was wondering, I wanted to go through a literature and check whatever I have done, what the literature has to say. Based on this is a systematic review on class 2 elastics. So based on this, you can see that the class 2 elastics and the fixed functional appliances, they have a similar effects. I just take 30 seconds more. And I wanted to check what is the difference between advancing and class 2 elastics. So advancing and intermaxillary elastics both are effective in normalizing class 2 malocclusion in comprehensive orthodontics. So in conclusion, I would like to say that the amount of proclination and crowding are not the primary determinants of the extraction modality. You need to understand the principles of growth and development and the long-term changes like the chin growth, the nose growth, and the goal of the treatment should be face-driven orthodontics. Yes, we want to get a great occlusion for our patients, but at the end of the day, we want to have patient flashing their smiles like this. Thank you, ladies and gentlemen.
Thank you, sir. Over to the chairpersons.
So, Dr. Shrikant, well, you presented your case very nicely, and you presented the concept of phase-driven orthodontics in a nice manner, and how we can clinically examine a case with a lot of patience and keeping in mind the potential of growth.
Exactly.
And keeping in mind the basic principles of orthodontics. So I hope the audience was able to pick some key points from here. Any questions, please? So that's fine, I think.
Thank you for a nice presentation, and it was very informative.
Thank you.
I would request our chairpersons to please felicitate sir. I would request Dr. Anupama ma'am to please come on stage and felicitate our chairpersons. Our next speaker for today is Dr. Anupama ma'am. She is a postgraduate from A.B. Shetty Dental College, Mangalore. Ma'am has a clinical practice experience of 13 years with Ayush Dental Care, Mumbai. Currently, ma'am is working as an associate professor at the SDM College of Dental Sciences and a consultant orthodontist with many clinics and hospitals in Dharwad, Hubli, and Bangalore. Ma'am has authored many publications in index journals, both national and international, with many book chapters. Presented scientific research papers at various national conferences and symposiums, and has won the Best Research Paper Award in the World Laser Conference in 2023. Ma'am specializes in use of photobiomodulation for accelerated orthodontics. Ma'am is also a certified Invisalign provider and the aligner provider and planner. We welcome you here, ma'am. Our chairpersons for this session are Dr. Subramanya Shetty, sir. Sir is a postgraduate from NTR University of Health Sciences and is currently a professor at Yanapoya University, Mangalore. Sir also has many publications in renowned journals. Our second chairperson for today is Dr. Sanju Somaya, sir. Dr. Sanju Somaya, sir, is a consultant orthodontist and a professor, PG Guide, Department of Orthodontics, Kurk Institute of Dental Sciences, Virajpet. I would request them to please come on stage. Over to you, ma'am.
So good morning to everyone present here at the 27th IOS PG convention at Tena Dental College. It's a beautiful morning after yesterday's gala banquet in the beautiful flamingo city of Navi Mumbai. I don't know how many of you know that Navi Mumbai is called as the flamingo city because of these migratory guests we get in the winter season, especially in the December and Jan, who come to the Airoli Creek nearby. So the topic for my today's presentation is accelerated orthodontics, and I will be trying to put accelerated orthodontics into the perspective of, is it just a means to an end, the end being finishing our orthodontic treatment quickly, or is there more to it? And if there is more to it, how do we incorporate it into our clinical practices and not just leave it in the pages of the textbooks for academic purposes? So what is this? This is a bullseye, right? And in our practices, what is the bullseye? The patient's chief complaint should be our bullseye. It should be, even Dr. Shrikant mentioned, it should be patient-centric care. So to do well in our clinical practices, it's very important that we plan our treatment around the patient's desires. What the patient says is his chief complaint is what we should be planning our treatment around. So to resolve the patient's chief complaint and to resolve it in a reasonably short time should be our goal. So when the patient comes to us with their problems, we do an examination and we arrive at different treatment options, explain it to the patient, tell them the cost of the treatment, and what they ask is, how long is it going to take, doctor? And then they go like this. Why? Because we say 2.5, 3 years, we drop the time bomb, right? And most of them don't have time, including us. All of us would want to finish off treatments quickly because time is money. So we should pay attention to finishing our treatments quicker. Of course, biomechanics and treatment planning and the mechanical aspects of our treatment is very important, but even biology can play a part, and we can modulate biology to finish our treatments quicker. So patient-centric care with comfort of the patient, the aesthetics of the patient, of the treatments which we deliver, the lifestyle of the patient, cost, and treatment care, it being safety and speed is important. They are the important parameters for a patient-centric care. I would be concentrating on the speed of treatment. Now, what are the factors which affect speed or duration of treatment time? The first one being customizing appliances. This is majorly biomechanics. We can't treat all cases in a similar manner. We have to do customized treatment planning, and we also have options like incognito and your aligners, which are customized plans, can be used, and they help in finishing our treatments quicker. Also, by modulating the physiology around the supporting structure, the PDL and the alveolar bone, which is done in our dental practices, we can quicken the treatment, the tooth movement, and reduce the treatment time. Also, the third option is by giving auxiliary devices to the patients themselves, we can speeden up the treatment to have a satisfied patient. Now, what is the rationale for accelerated orthodontics? It comes back, we have to go back to the biology of tooth movement. So when we are trying to figure out, like Van Diesel and his Fast and Furious team, where do we apply that extra gear so that we increase the rate of tooth movement? We have to go back to our basics, which is the biology of tooth movement. We know that there are various cells and various tissues involved, the bone tissues, the bone cells, the periodontal ligament involved, which is in the tooth movement. So once we understand the entire cascade of orthodontic tooth movement which happens, the interaction between the cells and the molecules, the first messenger, second messenger, and the entire orchestra which happens there, then we can decide where we want to intervene so that we enhance the body's response for the orthodontic force we are applying. By intervening at the right areas and at the right time, we can modulate the body's response to the same force which we are applying. So this is the cascade. I would not go through it, everybody knows it already, or you can go back and read. Now I will give an example to explain this. When we apply orthodontic force, there is always hyalinization. We try to reduce hyalinization by keeping the blood vessels intact, by reducing the occlusion of the blood vessels by PSL as an example, and also by applying light continuous force. But however, there is some amount of hyalinization that does definitely happen. So what removes the hyalinization is the osteoclasts. So we want to stimulate the osteoclasts, increase the number of osteoclasts, and also increase each osteoclast activity, that is stimulating the osteoclastic activity, so that the hyalinized zone is removed quickly. So that is how the tooth would accelerate into the next stage of movement, right? So osteoclastic activity needs to be increased. Now, is it sufficient only if we increase osteoclastic activity? No. Why? Because osteoclasts need osteoblasts for their differentiation. The pre-osteoclasts have the rank receptor onto which the Rankel ligand, which is released by the osteoblast, comes and attaches. That Rankel ligand is what causes differentiation of the pre-osteoclast into osteoclast, which brings about removal of the hyalinized zone. So osteoclasts and osteoblasts both need to be stimulated and activated. It is just that between our activation cycles, appliance activation cycles, the population of the cells at different times is different. Initially there are osteoclasts, then there is a plateau where we see osteoclast, osteoblast population being equal, and then the osteoblasts increase. So we have to stimulate at each stage, each cell. With that background, I come to the most popular methods which we use to accelerate tooth movement, and all these we should not be leaving it in the textbooks. This can be incorporated very easily into our practices. I would like to divide this into doctor controlled techniques, which are of two types: invasive techniques and non-invasive techniques, and patient controlled techniques. So doctor controlled techniques, the first one, three main ones. The first one being corticotomy, which was introduced in 1959 by Henry Cole. The rationale behind the corticotomy is RAP, which is regional accelerated phenomenon, as given by Robert Frost, wherein there is a trauma or an insult done on the teeth. We do a cut on the alveolar bone in the interradicular area. Multiple cuts are done between the teeth, whichever segment we want to move, and the teeth is moved and the force is applied. Now this, as gross as it looks, is an invasive procedure. There is time, it consumes a lot of time. There is patient recovery time involved. There will be costs and training involved, and other specialists who have to come in for treatment. So and also with corticotomy, there was adverse effects like root resorption and uncontrolled osteoclastic activity which goes on, which leads to root resorptions and reduced bone density and tooth mobility, which was seen. So to overcome that, there was a technique given by the Wilco brothers, Thomas and William Wilco, perio and orthodontists, in 2001, wherein in the same corticotomy cuts, on top of that they placed a autogenous bone graft, so that there would be osteogenic potential which is induced and there would be quicker healing. So we can control the continuous osteoclastic activity and break it at some point in the future. That is called as PAO, periodontally accelerated osteogenic orthodontics. Still, it is still invasive, right? To reduce the invasiveness, a technique called as piezo-scission or dissection was used, wherein mostly flaps are not raised, or if required, tunneling is done, and the osteotomy, the cuts, corticotomy cuts are given without raising a flap, again in the same position interradicularly between the teeth. So this again requires a bit of training, and there is still anxiety and pain caused to the patient. So these three are mainly the invasive procedures which are used. Now doctor controlled minimally invasive or non-invasive techniques. The first one is micro-osteoperforations or MOP, as we call it, MOP, MOP. So in this, instead of cuts, of course there is no flap raised, and instead of cuts there are small perforations which are done along the roots in between the interradicular area. The advantages are, it is almost non-invasive, it is micro-invasive. There is no patient recovery time. We just do the perforations and then start off with the procedure and patient goes back. There is no pack or anything given. So there is no pain or very less pain, and there is no anxiousness to the patient about the procedure. It does not require any extra time and no extra cost. Once we have invested in the equipment, it is done. We can use, except for the disposable tips, we can use it for all patients. The other procedure is low level laser therapy, which is completely non-invasive, used by Kohara and Cruz in 2004 in their clinical practices. Here we use photobiomodulation lasers, which is nothing but, when we say low level lasers, it is nothing but the photobiomodulation range of the lasers which we are going to use. It is used and it is completely non-invasive, no patient recovery time. It takes around 5 to 6 minutes time, and the training is like 5 minutes. We can learn how to use the laser machine. Now so that completes the doctor control techniques, invasive and non-invasive. The patient control techniques are of course non-invasive. First one being the LED, the light activated device. So it comes like a mouthpiece with the switch on and off button. It is a rechargeable device. It requires that the patient uses it for just 20 minutes a day, and of course it being a patient being using it, it needs patient compliance. That is the only disadvantage. There is a cost factor because it's a device which is given once for the patient. So there is, in the US, this device which I have shown here is OrthoPulse. In the US it costs around $1,000. This other device is a vibratory device which uses high frequency vibration. The device in the picture is vPro 5. Here high frequency vibrations of around 120 Hertz are used, and again the device is given to the patient. They use it from their houses and they use it for around 5 minutes a day, which is sufficient. Both these devices, like all remote devices which the patient carries to their home, have an app through which the doctor comes to know how the patient is using the device, whether they are overusing it, underusing it, and we can communicate with the patient. So I will delve a bit deeper into each of these, especially the non-invasive techniques. First one being low-level laser therapy. This can easily be used in our practices. If we have some device which is very portable and which is like our light cure gun and which can be just switched off, switch on, and we know how to use it. Many times we don't use a device, one because of accessibility or procurement problems with the device, cost for the device, cost of investment, and third thing is we don't know how to use it, or if it is too complex we don't want to get into it, right? But this is a very simple device, costs around 60,000 bucks, made in India. It's called as NLASE, made by a company in Hosur who are into optical lasers already, big time into optical lasers. So inside this there is a gallium aluminum arsenic semiconductor diode, which is the source of the laser light. Einstein he invented photons, and since the time of invention of laser photons, it has been used in various industries, including health sciences. In dentistry also there is hard tissue laser, there is soft tissue laser. The laser I am talking about here is a cold laser. Cold laser meaning it does not increase the tissue temperature. Laser has this effect. Surgical lasers which are used by our periodontists and other fraternity people are all heat producing lasers. When it is used on the tissue, there is heat produced. But this is cold laser with a lesser wavelength in the initial part of that infrared light. So the parameters we should be familiar with while using the laser device. First one is the laser source. As I said, it is gallium aluminum arsenic diode laser which is used. Now how is it used? It is used in continuous mode. Continuous meaning once we switch on a device, we don't put it off till the procedure is over, till the irradiation is done. It can be used in continuous mode. Why? Because there is no heating of the tissue which happens. It is a cold laser, right? So the tissue on surface on which you put the laser, there is no heat produced. Whereas in surgical lasers, we have to use it in pulsed mode. We have to allow the tissue temperature which has gone up while using the laser to reduce. So that is why we use it in a pulse mode, whereas this being a cold laser, we can use it in continuous mode. Hence the time required for the procedure is less. That is also the advantage. It is used in contact mode. You don't have to keep it away from the tissue because there is no damage caused. It is used in contact mode, and it should be used in contact mode so that we have good penetration. Now till where should the laser light penetrate? Where we need the stimulation of osteoclasts and osteoblasts in the periodontal ligament area and in the adjacent alveolar bone, right? So it should penetrate through the cortical bone into the medullary bone. So penetration needs to be good. So next point talks about it, which is wavelength. The laser wavelength we use is around 630 to 810 nanometer, which is the photobiomodulation range. When we say photobiomodulation laser, it is not some different laser, it is the range, it is the wavelength we are talking about. So 630 to 800 nanometers. 630 as the laser light, if you see the laser light, I have kept this on at around 630 nanometer wavelength. That is why we see that redness. If we reduce, if I change the settings to 810, it will be a very narrow beam. Only if we focus on it, we can see. And if we go towards 1100 nanometers, we can't see that there is a laser light which is there. That is why we need to be careful with our eyes, and eyewear protection is required when we are using lasers in a room. So the penetration is inverse. If the wavelength is less, the penetration is more deeper. So we use ideally this wavelength of 630 to 810 nanometer. Most of the scientific evidence also falls within that range. Penetration, of course, is also a factor of, is it a hard tissue laser, is it an argon helium laser which we are using, the power output and everything. The third parameter is the power output. It being an electric device definitely requires power. So power settings will be either 10 to 500 milliwatts or 0.01 to 0.5 watts. We can change it as per our requirement. Now the fourth setting, now all three first parameters which I mentioned are they all come with the device. We cannot change it. A parameter which is in our control is irradiation time. We can control whether we want to irradiate the tissue for 5 seconds, for 10, 15, 20 seconds or a minute even. So that is where we can control how much energy we deliver to the underlying tissue. So based on playing with these factors, we can decide how much energy density or how much energy dose we give to a particular tissue. And for acceleration, there is a particular dosage which we need to give. That is, I'll explain that. So the energy delivered, what is the formula? Many of us don't know the formula, so you can take this because it's not there in our, it's there in one of the current recent publications of mine, but it is, I had a great difficulty time finding it when I started my doctoral research. So energy delivered is given by power output, which is the third point there, and the exposure time. There is no wavelength in the formula. We keep searching for wavelength. There is no wavelength in the formula. So there is power output, which is the third point, and so we set our device to a particular power output and we decide how many seconds we want to expose it to. So basically it's done inverse, it's done reverse, the reverse manner. We decide what is the energy we want to deliver to the tissue. According to that, we decide how much is the time we want to expose it to. Okay. So this is a laser irradiation process for en masse retraction of the anterior segment in an extraction case. So how is the laser irradiation done? It is done for all the 6 anterior teeth from canine to canine. Each tooth, we have 2 points of exposure on the buccal side and 2 points on the palatal side. On the buccal side in the cervical and the apical half, and even on the palatal side in the cervical and apical half there. So we have 4 points per teeth and totally 24 points for all the 6 teeth. So the energy dose calculation, you see, I used a setting of 200 milliwatts. In the formula, it has to be used in watts and not milliwatts. So it is 0.2 watts multiplied by, I used it for 8 seconds per point. So 8 seconds and 4 points per tooth. Hence, we delivered around 6.5 to 7 joules of energy per tooth. The evidence tells us between 6 to 10 joules is what is required for stimulation of the cellular activity beneath. So that is why we decided on 8-second exposure. It can go up to around 10 seconds. The total energy dose which was used for all the 6 teeth together was around 40 joules. Now next point, after we decide how much energy we need to deliver to the tissue, is how frequently should we do the laser irradiation. I told that between 2 appliance activation appointments, there is that tooth movement phase, we know the lag phase and all those which happen. Now we need to understand the cellular activity which happens between these 2 appointments. So as I said, initially hyalinization, then the osteoclasts come in from the bloodstream. Osteoclasts start removing the hyalinized area. The number of osteoclasts increases, gradually increases from 24 hours till around the 7th day. They peak at around the 7th day. From the 7th day, the osteoblastic activity also increases, and at around the 14th day, osteoclasts and osteoblasts are almost at the same level, then the osteoclast activity reduces. Why? Because osteoblasts along with Rankel, which stimulates the osteoclast, also release a decoy enzyme which is OPG, osteoprotegerin. Osteoprotegerin competes with Rankel and goes and attaches to the rank which is on the pre-osteoclast. So it blocks the Rankel by bonding to the rank. That is why the pre-osteoblasts cannot further differentiate into osteoclasts. So if we stimulate the osteoblasts, what that does, it acts like a braking mechanism. It breaks the continuous osteoclastic activity. We want osteoclastogenesis in the beginning, but we want it to stop also at a point of time. That is why stimulation of osteoblasts is also important. The way lasers act, I'll not go too much into it, but we should know that lasers act on the powerhouse of the cell. It is energy, right? So the laser photon is absorbed by the photoreceptor, which is there in the respiratory chain molecules in the with mainly the CCO, cytochrome C oxidase, that is CCO, which is there in the mitochondria. This leads to increase in ATP production and further increase in DNA synthesis and transcription factor production, which leads to increased cellular activity. The evidence, most of the studies here show that there is increase in activity with around 6 to 10 joules of energy being delivered. There is one study in red where there was no effect or inhibitory effect. If you observe, sorry, some of this is not working. So if you observe the dose which was delivered, it was 25 joules per point, which is too high. So this is called as the biphasic effect of laser. Biphasic effect is also called as by the name famously called Arnold-Schunck's rule. Now, if we use laser dose, if it goes beyond a certain point, then it is inhibitory. So we should keep it between 6 to 10 joules. Many Indian studies which have been done. Now, the only limiting factor I found was most studies were either canine retraction studies. Now, this is contradictory, right? One side we are trying to accelerate the entire treatment time and reduce the treatment time, and if we do canine retraction and then again do anterior incisor retraction, it counters the entire logic. So, and canine retraction we usually do in academics and don't do it in clinical practices, especially with the invent of TADS, which is used for anchorage control. Hence, NMAS retraction is what makes sense. Most of the studies have done canine retraction studies. They are single tooth studies, and they are around 3 months, 2 months observation period studies, whereas we have observed the tooth movement of the entire anterior segment, so NMAS retraction for the entire duration of space closure, and there was around 40% increase in the rate of tooth movement with the use of lasers. So that completes lasers, and we move on to micro-osteoperforations quickly. It's easy to learn, but it is misunderstood because most of them think it is just a hole punctured into the bone. It's not so. What is required is the rationale, as I said, is RAP, bringing in RAP, regional accelerated phenomenon. The rationale is similar to corticotomy, wherein it is the dismantling of the cortical bone which is required, or reducing the resistance of the cortical bone which is required is the rationale which is used. So when you do an MOP, there would be compressive stress in the cortical bone developed, and there would be micro cracks which develop. So that is important to be done. Not okay, sorry. So that is the micro cracks which develop and compressive stress, and there is change in the local bone physiology and chemistry. The device comes in two forms. One is a handheld manual driver with disposable tips which come, and the motorized version. Motorized version is nothing but a handpiece, contra-angled handpiece with the disposable tips. We can adjust speed, torque, and access is improved because of the contra-angle tip. The latest avatar of the device is accelerator PT. Now, is this available in India? Because I will always come back to, can we use it? It's not just for textbooks. So this is not available, marketed in India. 5 years back, it was quite difficult because it had to be bought in the US and then shipped privately to India. But now, Dentsply Sirona, SureSmile has bought Propel in 2021, and we can order these devices. So the technique is nothing but Wilcodontics simplified. RAP, cortical bone disruption, is what is required. Where and how many and how often? It is done between the roots of the teeth. Depending upon the root length, we can either do 2 or 3 perforations. If it is short rooted like premolars, we can use 2. If it is canines, we require around 3 perforations. Anteriors, again 1 perforation. How often? For an extraction case, we can do at least 2 or 3 times during the leveling stage, and during the space closure time, 2 times can be done. At the depth of the penetration, we have to just touch the medullary bone. So in the anterior region, it is around 3 millimeters. In the upper posterior region, around 3, and in the lower posterior region, 5 to 7 millimeters. More dense bone and more thick cortical plate. The technique is very simple. We keep the handpiece perpendicular to the targeted tissue area, press the on button. There is an indication of how deep the tip has gone. So the minute it goes to 3 to 4 millimeters, we stop and then reverse it back. So micro-osteoperforation, many people, most of us, even we think that we can replace the propel nahi hai to kya hua, we use our TAD, but there is a difference in the design. To achieve the same 1.5 millimeter thickness of perforation, we have to go deeper if we are going to use a TAD. Why? Because it is tapered, right, to go more wider. It is important to grow wide because the focus is on creating micro cracks in the cortical bone than go deeper. So that is how the perforations are done in an extraction region. And there is enough evidence, more than 600 peer-reviewed journals are there which support the effect of corticotomy. Micro-osteoperforation, sorry. Now, also, it need not be only for accelerated orthodontics. In cases like this, where we have an old extraction site with dense bone, and where the buccal and the palatal lingual cortical plate are close together, also we can use micro-osteoperforations to enhance or speeden up the protraction of the molar. So the take home is they are an effective procedure and well tolerated. Again, the literature says around greater than 60% of tooth movement happens. Mechanical vibrations started off with low vibrations, which is less than 30 to 40 hertz. Then NASA came in. Again, they did a lot of research on how to maintain the bone density of the astronauts who go into space. So they were already using vibratory machines there. So they found out that high frequency vibrations are much better than low frequency vibrations. So we shifted even in health sciences and medical sciences too, from low to high frequency vibrations. High frequency would be anything above 100 hertz. So in orthodontics, in 2013, we were using low frequency vibrations, whereas now it's high frequency vibrations. So this is a device which is specifically meant for high frequency. There are 2 devices. One is Acceledent, and then there is VPro 5, which is by Propel, now by Dentsply Sirona. So it's a simple device with a mouthpiece which has to be kept between the 2 upper and lower arch and pressed on, can be easily used by the patient. Mechanism of action is it enhances the rank-rankle Dawkins, the nuclear factor kappa B ligand, which is rankle, and rank activation is done and osteoclastogenesis is stimulated. And there are growth factors also which increase with the use of mechanical vibrations. So the evidence compared to low frequency, high frequency is better. There is faster tooth movement compared to fixed orthodontic braces. The other factors, we have been talking only about increase in speed of tooth movement. Though there are other advantages of doing these mechanics of accelerated orthodontics, which is it is found that root resorption is reduced if we do even CBCT studies with acceleratory devices which are being used. There is extended pain relief in patients, especially with the use of low-level lasers, and there is reduced need for refinements. When I say refinements, aligners is where we need refinements, right? So because VPro 5 was marketed initially as only to be used with aligners, they were given as a package by SureSmile and other aligner companies, or even Invisalign, along with their aligners, so that the patients can use them. That is why reduced refinements. Most of the research is on aligners. So take home is it significantly increases the bone remodeling. there is increased in the cellular messengers like cytokines and all. There is reduced pain and reduced discomfort to the patient, and it can improve accuracy. So the other benefits of using AO, accelerated orthodontics, is effective pain control. It is found that there is hypoalgesia, that is reduced pain. Why? Because there is this theory called as the gate control theory, which talks about the neurons, the big neurons, which are the large dimension neurons, and the small dimension neurons. With the use of lasers and also MOP, it is found that we can block the input to the input or the stimulus which comes to the small dimension neurons. That is how that gate is closed and there is hypoalgesia. So with the use of lasers, we can have hypoalgesia. And there is also studies on retention. After our RME and expansion being done in the mid-palatal suture, if we use lasers, low-level lasers, there is better remodeling, there is faster remodeling which is seen, there is better fiber, collagen fiber realignment quicker. So hence it can be used even for retention protocols. Root resorption has found to be lesser with the use of lasers and also with the use of high frequency vibration. However, there's a bit of controversy in this area because this requires long duration studies. If we use, like in that study, only 28-day period, it's not sufficient to study this. So we can overcome all the disadvantages of fixed orthodontic therapy alone, if it is done alone, if we use these accelerated techniques. So it's a no-brainer and it's quite simple to use, right? So I conclude by saying, irrespective of whatever method we use in accelerating our tooth movement, the differentiator we can influence, which is patient experience and practice efficiency, can be influenced better with the use of accelerated orthodontics. And it is not just a means to an end of just quickening the treatment. We can also provide better quality care and also reduce our own burden of rendering treatment as clinicians. Thank you.
Over to the chairpersons.
Thank you, Dr. Anupama, for your wonderful insights on the methods to increase accelerated orthodontic treatment. Any questions from the audience?
Yes, sir. Yes, sir. Yes, sir. I would say lasers are better.
Hello.
One is, it is non-invasive, definitely. So we don't like invasive techniques as clinicians. Patients definitely don't like it because of the pain and the anxiety. So lasers are more easier to use and we can do it ourselves. We don't have to call any specialist to come in. And with devices like the one I showed, which are available at around 50,000-60,000 with no maintenance at all. I've been using it for 5-6 years now. There's no maintenance. So definitely lasers.
I mean, that case, that means in every extraction cases, if you want to hasten the treatment, you will prefer laser for the closure of spaces, or you need some specific case-based treatment indicated only for laser.
We can use it pan orthodontics. We can use it for all our cases. There is no restriction that we can't use it. But in a more challenging case, wherein I feel the anchorage demand is more, wherein there is more proclination, I would want lesser resistance to tooth movement. I would use lasers because I would want to accelerate the bone remodeling in the anterior segment, tax the posteriors less, tax the anchorage less. So in more complex cases, I would use lasers.
I mean, bone remodeling. If bone remodeling is happening so frequently with the exposure of laser, that means what will be the treatment interval, the duration of this exposure, ma'am?
Currently, all my cases, I do the same 4 to 6 weeks appliance activation protocol. But seeing that the bone remodeling is quicker, we can reduce it to 3 weeks of appliance, between appliance activation.
Thank you.
Dr. Jain, very good presentation. It's really a treat to watch your presentation. And there is a lot of scientific insight to take home. Adding to your presentation, your main skeleton of your presentation is speed. And I've seen many board besides roads, speed thrills but kills.
Yeah, there's one here with the car crashed.
So just add on to that, I would like to ask you, what is the tissue death percentage when you use a laser for 1 joule of energy? Is there any ethical clearance or any studies done on that?
The tissue depth penetration in vivo is not studied because there is no way of studying it, but definitely ethical clearance is required. But what we understand is there are no adverse effects. If we even see the periodontal index, gingival index, there are studies which have measured all that. The tissue doesn't seem to have any adverse effects. So from that we can say that there is no adverse effects of lasers and we can use it safely for at least between that energy level of 6 to 10 joules.
What about medically compromised patient like hemophilic, you know?
Maybe we would have to, yeah.
And one more is that
lasers, I will complete. Lasers have beneficial effect even on the blood cells. Lasers do cause neoangiogenesis also. So that is how the wound healing is quicker with photobiomodulation lasers and across medical fraternity where it is used. So I think hemophilia should not be a contraindication for the use of lasers.
Okay, we have covered almost all in accelerator orthodontics. What is your take on injecting prostaglandin and calcium sometime?
Yeah, injecting prostaglandin, calcium, vitamin D, all these, even IPRF, because they will come and mostly under the invasive techniques. I didn't go into the depth because of the time constraint. But yes, prostaglandin injections are the adverse effect of pain and again uncontrolled osteoclastic activity which goes on. That is why in the early 2000s they were used, but they were stopped. So with the non-invasive techniques being more popular, the invasive techniques are being less and less used. We don't like calling specialists to our clinics. That's why.
Altogether, it is very well put in. Very wonderful presentation.
Thank you, sir. Thank
you. I would request the chairpersons to please felicitate Anupama ma'am.
Thank you. Thank you. Thank you, sir.
I would request Dr. Soumya ma'am to please felicitate our chairpersons.
Who's coming with me? Please come. Thank you.
Our next guest speaker for today is Dr. Soumya ma'am. She has done her post-graduation from Savita University, Chennai, and a fellowship in craniofacial and special care needs orthodontics from the University of Michigan. She had a clinical attachment with Manchester Regional Cleft Unit, Royal Manchester Children's Hospital, Manchester, United Kingdom. Ma'am has won the Best Cleft and Craniofacial Orthodontist Award by the World Dental Conference held at Dubai. Ma'am has been honored with Savitribai Phule Award on the occasion of International Women's Day celebration at Gandhi Bhavan, Karnataka. Currently, ma'am is the head of Department of Dental and Oral Surgery Unit at Christian Medical College. We welcome you here, ma'am.
Thank you.
The session is going to be chaired by Lieutenant Colonel Dr. Amrit Thapa sir. Sir is a life member of the Indian Orthodontic Society and has attended various conferences and has publications in renowned journals. Sir's area of keen interest has been OSA, surgical orthodontics, rapid orthodontics, interdisciplinary orthodontics, and surgery first approach. I would request sir to please come on stage. Over to you, Soumya ma'am.
Good morning, all. It gives me immense pleasure to stand before all of you to talk about my presentation today, Contemporary Cleft Orthodontics. To begin, I would thank PG Convention, all the organizing committee for giving me this platform, and a special thank you to Dr. Shailesh sir and the organizing team. So I begin by a simple question. So today you're just going to understand the contemporary aspects of cleft lip. I'm sure you all know a lot about treating cleft patients. So what is a cleft? A cleft is just a gap or a split in the upper lip, palate, or sometimes both. The cleft can be incomplete or complete, and maybe unilateral or bilateral. I'm sure you all know that. And there are many classifications, but the classification that we follow is Dr. Balakrishnan, Professor Balakrishnan's classification at CMC Vellore. And he's a plastic surgeon, Indian plastic surgeon. It's a Nagpur's classification, which goes like group 1, group 2, group 3. And please read it. So the implication of cleft first. We see there's an altered facial appearance. We see feeding difficulties, speech and hearing problems, altered facial growth, dental development is altered, psychological well-being is also disturbed, and learning issues may be a concern. So very important is when you're treating any patient is to understand what are the problems and what are you treating. That is the most important, point to actually start or to begin with. So in a unilateral deformity, we would see there are 6 points that you'll have to see that is a problem list. One is a wide nostril base. There is separated lip segment on the cleft side. The affected lower lateral nasal cartilage is displaced laterally and inferiorly. And there's a depressed dome. There's also appearance of the increased ala rim. There's an oblique columella. There's an overhanging nostril apex. I don't know if I can. I'm sorry. I think way too fast. All right. If there is a cleft palate, then nasal septum will be deviated to the non-cleft side with an associated shift of the nasal base. I don't have a pointer here, so I'm not able to make point it out. I hope you'll be able to make out. The bilateral deformity, we see a procumbent or a rotated premaxilla. There's an increased ala base width. The lip segment are widely separated, severely deficient or absent columella that you can see. The flattened nasal tip appears tethered directly to the probolabium. The lower lateral alar cartilage are either flared and concave, but actually it has to be convex. So there are certain dental features which we see. There's hypodontia for sure. There's supernumerary teeth, ectopic teeth, malformed teeth. There's also the patients they have a high dental decay risk. So aim of this presentation for today is to provide an overview of contemporary cleft lip palate, and we'll also see the management and importance of the MDT approach. The principle of any cleft care team is to achieve relaxed speech, hearing, facial appearance so that the patient is integrated into the society with a minimal morbidity. So the best possible result, concentrated interventions, minimal burden of care, multidisciplinary team treatment from birth to adulthood should be our principle of care. Now, there are multiple management pathways. The pathway that we are going to talk about is the MDT team. So the American Cleft Palate Craniofacial Association, which was formed in the response to complexity of treatment and need to coordinate care. It was started in 1943 by Dr. Herbert, and this is Dr. Rusty, who is the grandson of Dr. Herbert, who started this MDT approach to cleft care, and he is also associated with the Michigan team. So the MDT team itself means a team which has clinical nurse, you have a speech and language therapist, you have a cleft coordinator or a social worker, you have a neonate surgeon, an orthodontist, a maxillofacial surgeon, a plastic surgeon, a pediatrician, ENT specialist, a genetic counselor, and a psychologist. So this team is either headed by an oral surgeon or a plastic surgeon. So no single treatment concept has been identified, especially for a cleft palate patient. So the timing of individual procedure also varies in different centers with different specialists. Now, this is the MDT team that I attended at Michigan. Now, again, everybody has a different protocol. The first point as an orthodontist is learn all the mechanics, be your own innovator. Do not stick to books. Please learn all the different types of mechanics, all the knowledge that orthodontics has to offer, but always be patient-driven and always be an innovator as an orthodontist. So to begin with, I will talk about UMICH protocol and at Royal Manchester Children's Hospital at UK, and then I'll go on to say about how in CMC we have innovated our appliances based on the patient need. So the following is the most common treatment protocol, which is currently used in most of the cleft centers and also at UMED. Usually the patient seen at newborn. At newborn, you actually diagnose, you examine the patient for general counseling of the parent is done, feeding instructions are given, palatal obturator or NAM if necessary is planned, genetic evaluation and specification of diagnosis, whether it is syndrome related or non-syndrome related cleft, empiric risk of recurrence of this cleft is calculated, whether it is, the next generation would get it or not was also spoken to the parent. Recommendation of a protocol for the prevention of a cleft recurrence in the family is counseled to the parent and age appropriately to the treated cleft patient, the adult. So at age 3, there's a repair of the cleft lip that happens, and usually the early surgery allows the child to establish a good orofacial function. That's very important for speech. But again, surgery usually causes scarring in maxillary, and that causes, protrusion of maxilla. That's another part for us to deal with. At age 6 months, we continue pre-surgical orthodontics, that is if necessary. The first speech evaluation happens at age 6 months. At age 9 months, speech therapy begins. 9 to 12 months, there's repair of the cleft palate. And if there is any tube that is required for the ear, it is done at that time. Age 1 to 7 years, again, orthodontist follows the patient for any intervention if it is required, 7 to 8 years. Or at 10 years, evaluation for ABG or the alveolar bone graft is done. It depends which tooth is erupting into the cleft, whether it is a lateral incisor or the canine. It is not just the canine. So it is very important to evaluate the patient between 7 to 8 years or 10 years based on which tooth is going to erupt there. So older than 10 years, an orthodontist always evaluates for growth plan, whether, based on whether you need a face mask therapy or bolted plates or fixed orthodontics, what exactly is needed is planned based on that. At 14 years, again, you're evaluating for your basic orthodontics and also rhinoplasty if patient is very concerned about how the nose looks, or you can always push it to after 17 or 18 years with your main surgery. At 17 or 18 years, a surgical consult is done to plan whether you need surgery for function requirement or aesthetic requirement, or so along with that, we normally do our pre-surgical orthodontics and and then post-surgery a little bit of orthodontics for occlusion settlement. So this is at Royal Manchester Children's Hospital. Again, their team has a different protocol. They do not believe in NAM. They go up and here the head is the oral surgeon, and they aim to repair the cleft lip by about 5 months and palate by 12 months. The unilateral cleft lip palate cases, the lip and anterior palate, they repair around 12 to 20 weeks. And the second operation for the soft palate or any remaining hard palate is done at 32 to 40 weeks. Bilateral cleft lip palate cases are repaired in stages according to severity of cleft there. And cleft palate is a one-stage surgery at 24 or 40 weeks. Submucous cleft is required, if clinical evidence of feeding or speech problem is there, then the oral surgeon intervenes. That's Dr. Victoria Belle, who is the oral surgeon heading the team at Royal Manchester Children's Hospital, UK. Now, what we do at CMC is when there's a newborn and the initial years of treatment, so again, I try to emphasize that an orthodontist, as an orthodontist, we all have different perspectives. We all have different idea of molding the maxilla or the maxilla in the cleft lip palate patient, because we do understand that further retroots the maxilla, right? But then the debate becomes very, very less important when you understand that the growth disturbance is it because of the surgical insult, or actually there is an intrinsic genetic defect. So there's nothing much we can do either. So if you treat the patient, there are a lot of benefits. So again, burden of care should be the most important thing that you have to decide when you're treating your cleft patients. So nasal molding is not a universal rule for all cleft patients, and you also have to be an architect to plan why, what, how you would design the alveolar segment so that they have the best possible position in the oral cavity, and keep in mind the burden of care the patient parent also would have to go through. So there are many centers that do molding technique, either due to some don't do it, either because they don't have experience, or either just the lack of acceptance in their team members. Like certain people just don't accept nasoalveolar molding. That's all. It depends on your team, depends on your surgeon, depends on your plastic or oral surgeons, and your innate want to do something for your patient. So what basically does this nasoalveolar molding segment do? Basically, this molding plate is worn for 24 hours, 7 days a week, including when they're feeding. And parent changes this tape. There's an extra oral tape and all that that's been put. And it is cleaned every, before, feeding and all that. And after that, like each adjustment is very small. It takes about 20 to 60 minutes, that's it. And by the time the surgery, when the patient is prepped for surgery, the nose has been lifted and narrowed, the gap of the gums have smaller, and the lips are closer together. The smaller gap definitely means there is less tension when the surgeon is closing it. So basically, you're trying to help the surgeon get a better, finish. So nasoalveolar segment molding is not the end of treatment. We should all remember that. For an orthodontist, it's just the beginning in cleft care. Now, what we do, we first start with an extraoral traction. So this I go into the NIC ICU and I use just an elastopore to close the to start with, and then I would go with a Dynaplast, and this is again using Dynaplast. There are certain parents who are very innovative. There could be parents who are coming from hilly region, so they have a cap, and he has used this extraoral traction, which is very important to begin with. So he's innovated this, and then we gotten an elastic force into that. So the next step we would do is the nasoalveolar plate, and this is how I would go into the OR along with the plastic surgeon to make the impressions. And once the model is come in, I follow Dr. Barry Grayson's technique. I don't know if this will play, but I can always play it later. I also take the impressions in dirty OR or minor OT along with the plastic surgeons being present there. So once we make the plate, we also see whether the feeding is good, and it is taught to the parents. So I'll play it, maybe because of short of time, I'll play it a little later. So these are feeding plates that we do. And again, there's a lot of innovation that we do with even the plates. We kind of use springs sometimes to get the premaxilla backwards. We've also used extraoral traction along with nasal stent kind. So this is one patient which we begin with. So this is very important. Positioning the patient when you're examining a cleft child is very important. Lap to lap technique is what is followed. And you always see how much the gap is, record the gap, and then you try to go ahead. So what we normally use is the Tegaderm or the Duoderm, like, Tegaderm tapes on the cheeks, and then use the Dynaplast. We also make the plates as required. More forward. This is another kind of innovation which we use when the premaxilla posteriorly is to tilted off. We have used a kind of orthodontics involving kind of appliances what we have made there. Again, a modifying molding plate. So like I said, please be an innovator. Do not stick to books. Learn all your get all your knowledge. Be a lifelong student, but always be an innovator. Patient care is at most important. So next, this is another patient that we've treated. So third step, we have just introduced after we've done the cleft lip, after we've done the nasoalveolar molding, we go in for the nasal stent. Again, it depends. Again, this is another kind of an innovation that we've done. One is a normal, the soft liner nasal stent that we use. We use another triangle based on the compliance, because otherwise it's touching the patient's nose and the eye and all that. So we kind of innovated into a triangular format. So again, like I said, this appliance is worn 24 across a day. Actually, the amount of force that's applied is about 100 grams. Again, it depends. As an orthodontist, we all know how we stretch our bands, right? So we kind of know that. And so like I said, we are going to use Dynaplast or Tegaderms on the cheek to reduce the irritation on the cheeks. So this is another patient. We used an expansion. Along with expansion, we wanted to get the premaxilla back. So we used some kind of E-chains, like we used some bands on, we used hooks on either premaxilla, and we did that. This was another patient who was in mixed dentition stage, and all this appliance innovation always depends on your patient, who are mostly sometimes not very compliant. So you just try to do something that at least they wear it, at least something happens. So that's how it is. So then there's some also with cleft patients, because it's an MDT team, it's very important to understand speech and language is always also a problem. So also these patients have hearing difficulties. So those with soft palate also, they're very prone to middle ear infections. So again, because of the muscle attachment, so they're very prone to serous otitis media or chronic otitis media, which threatens their hearing, which is very important. And at that time, you have to make sure that the team meets them at the regular intervals. So we will be a part of the like a moderator or a mediator to be a part, to make sure this patient sees the required team at the required age, because we are the one who are actually following the patient from newborn till the patient is done surgery till 18 years of age. So make sure you're vigilant on that. So during mixed dentition, again, you assess for the required of ABG age appropriately, again, based on the tooth that you're concerned, and bony defect in the alveolar alveolus associated with instability and mobility of premaxilla, that is what is checked that time. If there's any collapse of arch, compromised support at the base of the nose, teeth in the region unable to erupt, these are all the conditions that we're looking for at the mixed dentition. If there's any adjacent teeth which is unable to erupt, whether it's tipped, it's rotated, and any oronasal fistula is also checked during this time. So ABG is, again, if you see, we do iliac bony crest. Iliac bone is taken from the hip bone, and then that's when they augment it. Usually, typically, when the canine root, you actually see the x-rays and you time it according to once the 2/3 of the root is formed from the canine, and the same goes with the lateral incisor. And pre-OBG, normally expansion is done so that they get better access to the graft site. Excellent levels of oral hygiene is mandate, just not doing a treatment, but mandate is the oral hygiene for the patient is very, very important. So this is one of the procedure, like how they do the alveolar bone graft. Now, when assessing the ABG, very important, what we use is the Berglund scale. That's the gold standard of assessment. We also have Chelsea scale and the Kindelian scale. You can use either scales, but we use the Berglund. And definitive orthodontics, that's your during just about when the patient is about a teenager. And again, that depends on your cleft type, the facial growth. If there's any anomalies, then the cleft lip cases, the more conventional orthodontics that you do, if they're just cleft lip. And you always monitor the canine eruption, because after ABG also monitoring is very important. And you also look for if there's any missing teeth, and you either have to open or close spaces if there's any impaired maxillary growth. This is all done during this time. This is basically the assessment. And you also look in during this growth phase, you always try to get in functional appliances. Based on the compliance of the patient, you started a little early, like any kind of face mask. Bolloid plates are something that is in, which is surgical option, which can also be used around 14 to 16 years of age. When you think you can't use functional appliance, you can always go in for bolloid plates. Facial growth, we all know that in cleft palate patients, usually there is a retrograde maxilla, so you kind of use our functional appliance to get them forward. That's a maxillary protracting appliances. And normally the orthodontic issues that we face here is definitely an unfavorable growth. There's an abnormal crown and root anatomy. There is definitely a Bolton discrepancy also. There's asymmetry. Bonding problems because of hyperplastic teeth is also very evident in our clinical practice. So once the growth is completed, so what are you going to do? Either at that time you analyze whether you need to go in for orthognathic surgery based on patient's functional requirement or aesthetic requirements. So whether it's orthognathic surgery or distraction osteogenesis is decided based on the overjet. How much of overjet is there? Like we all know, the envelope of discrepancy. If it is more than 10 millimeter, you cannot do with surgery. Then you go in for distraction osteogenesis. So other surgical revisions also will be done during that time. Any scar revision, patient basically for aesthetic requirement, if there's any VPI consideration is done that time, and nasal revision or rhinoplasty can also be done during surgery. This again is all a discussion which is done with the plastic surgeon or the oral surgeon. So this is one of our patients which we had done distraction osteogenesis UMIT, but this we did it for Apert syndrome. Usually distraction osteogenesis, what I have assisted is for Apert or Cruson syndrome patient. I haven't done any cleft patient distraction osteogenesis, but hope so. I mean, if there is such bad retrograde maxilla, maybe we would. This is Dr. Bill. There is a book which is the Shaw Bill. I think most of our orthodontists know about it, and they've done lots of work. This cleft is, I don't know if you all understand, there is beyond knowledge, there's something called passion and something called service, which is very important for each one of us as doctors to give back something to our society. It is very important to be an empathetic orthodontist also, and make sure if you see a problem, kindly address it. In my tertiary care center, I see a lot of cleft patients, and that is where I have involved myself in cleft care. If I didn't have these patients, I would have done what was required for my patient. I would have gone for a quest of knowledge would be towards that. So since I had this, because of cleft patients were very much prevalent in my patient need, So that's how I developed this passion. So it is very important, as all you orthodontists, please develop your passion based on the treatment requirement of your patient at that socioeconomic status or wherever you are practicing. So that's important. So, and yeah, so I think Dr. Berkowitz, Dr. Grayson, Barry Grayson, there are many cleft orthodontists, and here in India, Dr. O.P. Karbanda sir, there are many people who've done a lot of work on cleft. And if you're all in, if anybody is interested, because I have time, if anybody is interested in cleft orthodontics, please reach out to me. I'll definitely help you out in any kind of I did a fellowship at UMich, Michigan. If there's anything that I can help you out with that, or even kind of if you want to visit centers at different places to have a global understanding of your cleft care or anything else, please reach out. Thank you. Thank you for a great time, and thank you for patient listening. Thank you.
Very nice presentation, ma'am. Any question from the audience, please?
Thank you, ma'am, for the wonderful presentation. Thank you. I have two questions, if the chairperson allows. First is about what is your opinion about the preschool rhinoplasty or early LR correction during the lip surgery itself? And my second question, your experiences with AMD, how much impact does it create on the VPD vis-a-vis your standard conventional distraction osteogenesis with RED?
Okay, so I'll first address your first question. Rhinoplasty, again, like we know, depends on the growth, right? Maxilla, we see the nasal growth. We normally judge it around 14 years. That is the, otherwise you'll be, see, the only thing about surgery is you don't need to repeat surgeries. Like even ABG, if you see, we had primary ABG and then we came up to secondary. So usually recurrent surgeries causes more scarring. So if it is really important to have a rhinoplasty at that age, that's a surgeon's call, I guess.
The contemporary protocol are being followed by the most of the reconstructive surgeon is the preschool rhinoplasty at the 4 to 5 years of age.
Okay.
Primarily because of the social impact, the social effect. So, but there are no long-term effects being studied on the mid-phase growth or on the premaxillary growth. So that's why I was asking about, because few of the reconstructive surgeons, even to the LR correction at the age during the lip surgery itself, the defects is not large. So your opinion about specifically about the preschool rhinoplasty, not that conventional what we do at the end of complete orthogonathy.
Yeah, see, that's what I'm saying. Like it all depends. That's why I'm saying it's perceptive. It's your the surgeon's perception, and also depends on the patient if they're very aesthetically driven. That's why when I'm doing cleft orthodontics, for me it's very important to have a global understanding of cleft orthodontics. I'm not so when you get a US background, it's very aesthetically derived. You know, if it is Dr. Barry Grayson, he does even a minute correction, because the patients are coming from New York, they don't want their child to even have like a minute defect. So they're so driven, aesthetically driven, whether the child needs it or not, the parents want it. But if you go to Europe, they're functionally driven. They do not believe in NAM. They do not believe in many concepts. So I will also be going to Australia in October, and I will get you the concepts from there. So again, to address your point, it's very, every treatment is driven by that protocol followed by the particular team. So if you ask me, again, for my personal thing would be the growth. Like we know growth, we know that there is a growth center here, and I would definitely see if you talk about the, if it is aesthetically driven by the patient, yeah, you can go ahead and do a rhinoplasty primarily, but I would always say, wait till, because Berkowitz knowledge saying that cleft untreated is also a good cleft treatment because you allow the growth to happen, but then again, you come back. So there's a juggle between treatment protocols, doctor. So I would just say all treatments is all right as long as your patient care is what that is important. I mean, whether there is aesthetically driven or functionally driven, or a doctor takes a call based on that.
The second doubt on the AMD, ma'am, your experiences on AMDs. Anterior maxillary distraction.
Anterior maxillary distraction, we did not done a lot of distraction, and just segmental distractions not yet. Like I have assisted distraction osteogenesis only for Apert and Cruzone syndrome, wherein the entire sutures are, and with that we have formulated different techniques for those patients. Distraction osteogenesis in cleft care, I haven't been a part of. So like I said, if the overjet, distraction osteogenesis also, like there's a lot of burden of care, like when you're using whether an internal distractor or external distractor. Again, at what age are you using that appliances? Are you using distraction during at 14 to 17 years, or you're using after 17 years? So yeah, so it depends.
Thank you.
Thank you.
Ma'am, in that case, if the severity, cleft cases, if the overjet is very large,
yes,
in that cases, how you're considering the distractions?
I would say distraction is the gold standard if you really want, because the envelope of discrepancy, like we've seen, if there's more to achieve because of relapse, you need distraction. Surgical, we know there's only that much we can achieve, right? So, but like I said, in cleft, we haven't done any distraction because the overjet was not that much. The overjet I see would be about 3 to 4 millimeters, because we've already done nasoalveolar molding. We already do expansion. We do face masks. We do bollet plates. We do so much so by surgery, there are times that they don't need surgery at all.
Ma'am, you have gained so much of experience by exposing yourself with the Europeans, and
yes,
my own, there are certain doubts are there, like what is your take-home message for the NAM treatment, ma'am?
NAM, see, personally, if you ask me, because I do NAM, it is a tedious process, because the child is not very cooperative, obviously, because when the parent comes back, she says the child takes out the appliance, the child does all this, and I'm like, the child is a baby, it's on the parent. The burden of care is more on the parents, because the parent has to be there like 24/7 to actually do, like you have to apply the tegaderm tapes, you have to put the, one is impression. When you're making an impression, you should be very careful. You're not doing it by yourself. However experienced you are as an orthodontist, you should not be doing on your dental clinical chair. You should be doing it under GA. And because I'm in a hospital setup, I get plastic surgeons or the minor OT, the dirty OR, I get the oral surgeons to be there to make the impression, because even if a little bit segment of your impression material, the patient, aspirates, your career is gone and cleft. So I feel it's very important. See, again, be very cautious when you're doing anything. When you're having protocols, understand, because there are many things can go wrong. When you just want one thing to go right, but many things can go wrong. And if you lose one patient in your whole journey, it's not worth it. So you have to be very stringent on your protocols. Like in your, if you're taking an impression for NAM, please do it under protocol. But just because I want to do NAM, I think NAM is great, and then doing it in my clinical, I really don't think that's a way to go. If you have a good support, if you have an MDT team, that's why I'm propagating an MDT team. Do not do it on your own. It's a complex treatment. You need to have a multidisciplinary team. You need to discuss. Patient care is very important. The child is very important, because you're seeing from day 1 to 18 years, there's 18 years of your involvement with that child. So if you have that passion, please go through.
Second is, ma'am, what about your experience regarding the secondary alveolar grafting? If the cleft size or the occult size is very large, in that case, how you go about it?
So that's actually a challenge. So first of all, it again depends on the surgeon by himself. Again, there's a lot of research that's happening with stem cell research and all. I hope that all comes in. Yeah.
Ma'am, other than the surgeons, it is the role of the orthodontist. That's why in that cases, we should be a proponent of the NAM. If NAM is there, in that cases, we will reduce the overall size of the occult defect from the beginning only.
Yes.
That's why NAM should be one of your treatment protocols.
See, that's what I'm saying. NAM is very important. That's the whole thing. So have a global concept again, I would say, because US believes completely in NAM. Europe does not believe in NAM. So when I went in Royal Manchester, when I saw, it was an eye-opener for me, because I also was, very NAM driven and all. Again, NAM, if required, please go ahead. If it's not required, like you said, if the gap, the cleft gap is more, please use NAM. If it is not, don't just do it because it's a cleft patient. You take a call on it. Yeah.
Thank you, ma'am.
All right. Thank you. Great. Thank you. See, again, a nasal stent when you're using, right? It again depends. Yeah. So normally they do it with the plate. If you have seen the plate, nasoalveolar plate, you've seen the nasoalveolar plate. With that plate, they use that 45-degree angulation, and they put the plate, and there's an angulation. But when you're lifting when you're lifting or trying to do, we look at when you're placing your domes. You see, for blanching, if you go beyond that, don't do it. Again, it's very personalized with every child. Don't go with 10-degree, 20-degree, 30-degree. But just imagine when you're lifting up, if you're going beyond blanching, don't go beyond that. Just, do that way. That's how we do it. I wouldn't say there's a particular force that is required. See, because the amount of active force that you're applying, the force is more to do with this, trying to get this. This lifting is more about how the soft liner you're adding. So I'm just going to look at how much I'm going to see about the blanching, and that's how we lift. That's the protocol that we have followed.
Thank you, ma'am.
Thank you.
Sorry, sir. Can I?
Yeah,
some of these questions are surgical. Do you mind if I?
Yeah, please. Share.
This question which you asked about preschool, can I? The so-called preschool, which you describe at 4 years and 5 years, right?
Yeah.
I think the key is for the surgeon to dissect the ala deformity without hampering the growth. So the orthodontist concept, the problem is that the dissection will create growth hampering, which will create maxillary hyperplasia. So the key for the surgeon is to make sure that only the ala is dissected.
Yes, sir.
So that because the appearance improves quite a bit.
Yes, sir. They avoid doing any undermining of the septal cartilage.
Correct.
They primarily deal with the LR correction.
Correct.
So that's what the philosophy, what the recon.
Yeah. So because the nasal septum is one of the growth centers and that hampers. So the surgeon usually dissects only the ala, just replace the ala. The appearance becomes drastically different, and you're not touching the septum. But the problem is a lot of these cleft cases have a severely deviated nasal septum, which is a functional problem.
Yes.
So aesthetically, they look good, minimal dissection and close. Now, alternatively, a lot of cleft surgeons, when they do primarily, either do NAM to improve the ala defect. It looks good, but it relapses, so you need retention. Alternatively, some people dissect the cartilage in a closed fashion. So you're not opening it up, you're closing small incision, you dissect the cartilage, free it up, and hitch it to the other cartilage with non-absorbable sutures. So that the ala is released, cartilage is released, it goes and hitches against other cartilages, so the appearance is good. So these are the things which is accepted, can improve without hampering the growth.
Yes.
So that is what is usually done. About I think you asked about the AMD.
AMD, sir, AMD.
The AMD moves the anterior maxilla forward. So it is believed that the posterior part of the palate, where the speech is dictated, is not moved. Means you will have to put your cut in front of the usual disjunction, which is between the pterygoid plate and the tuberosity. You put the cuts in front of that, wherever you want, and distract the anterior segment. So the posterior segment remains the same. Speech technically is not touched. Versus
theoretically, it doesn't have any effect on the VPD. So just want to know about the clinical impact. Does it cause, because I didn't find any study which is focusing on the VPD, especially in cases of AMD, sir?
No, AMD, there are a couple of studies.
No, about VPD only, sir. The correlation with the VPS.
It is there, it is there. I can give it to you. There are studies, because one of the major, there is a group between AMD versus the conventional distraction kind of. So the AMD group makes their argument strong by saying that the speech is not affected, and they have evidence to do that. So that's why. So I am a non-AMD person, conventional. I always get brickbats from the other group who can who do AMD. So, but that's one of the bigger argument they say that speech is not affected. But there are a lot of things to look at, because it's not just the maxilla which is dictating that.
Yes.
So a lot of things, but that is a major argument at this moment of time.
Thank you, sir.
Thank you, Sherry, sir.
Thank you.
I would request the chairperson to please felicitate Soumya ma'am. I would request Dr. Vineet sir to please come on stage and felicitate our chairperson. Our next guest speaker for today is Dr. Vineet Tundukatil. Sir is a postgraduate from Rajiv Gandhi University of Health Sciences, Karnataka. Sir has been a guest lecturer at various international conferences of the World Federation of Orthodontics, the IOC Japan, IOC Sri Lanka, and the national conferences at Jaipur, Kochi, and Bhubaneswar. Sir has publications in journals including AJODO, Angle Orthodontics, and APO Strengths. He has won the APO Strengths Award for the Best Clinical Pearl Innovation at Yokohama, Japan. Currently, Sir is a professor at Azizia College of Dental Sciences and Research, Kollam, Kerala. We welcome you here, sir. The session is going to be chaired by Dr. Ravindranath, sir. Sir is a professor in Department of Orthodontics and a PG teacher with an extensive clinical experience of 25 years and a teaching experience of more than 20 years. Currently, sir is working as a professor and head of department at MGM Dental College, NAVI Mumbai. Over to you, sir.
Thank you. Okay. Very good morning to one and all. Respected teachers, fellow orthodontists, and most important, my dear students. Before I get into the topic, I have to tell you a small story. A story of a newborn. The sister walks into the ward with the baby in the hand, hands it over to the mother. All the people in the room gathers around, and suddenly the baby flashes a smile. And everybody in the room are into a smile, or they are into laughter. See how contagious the smile is. Smile is the language of love. It's the language of peace, and it is universal. Various studies that have been done or surveys that have been conducted across the globe have shown that smile and facial aesthetics occupy a prime position. And in fact, in this selfie-driven generation, that is the me generation, wherein everybody wants to take a beautiful selfie, they need to have a beautiful smile. And what better can I tell you? Every single day when you open up your social media, including WhatsApp or Facebook or Instagram, whatever, you see many of these forwards which mostly has this particular word smile in it. And what better can I tell you than showing this picture? Now let's understand what a smile is. It's a facial expression characterized by the upward curvature of the corners of the mouth, indicating pleasure or amusement. A smile communicates a person's level of happiness as judged by the outside world. And this particular beautiful smile is marred with the excessive display of gingiva, which we refer to as high smile, the gingival smile, or a horse's smile. And that is what my presentation for today is, the checklist in the management of dummy smile, an etiology-driven, age-limited approach. And I am Dr. Vineet, and that's from where I am. Now let's understand what dummy smile is. Let's look at a qualitative definition of dummy smile. It's a condition characterized by the excessive exposure of the maxillary gingiva during smiling. It's a non-pathological condition causing aesthetic disharmony in which more than 3 to 4 millimeters of gingiva is displayed during smiling. According to Kokich, let's note it, according to Kokich, Any gingival display more than 4 millimeters has been identified by lay persons as anesthetic. And 2 millimeters, we orthodontists are more specific, 2 millimeters and more is anesthetic. And please note that smile attractiveness and gingival display has been studied across specialities, not just orthodontists, but prosthodontists, endodontists, everybody has evaluated this. And it is a controversy in quantifying this gingival smile. So for our understanding, let's keep it at the maximum, that is 4 millimeters or beyond. Let's keep it as gummy smile. Coming to the development of a gummy smile, the vertical dimension of the face, as we all know, it continues throughout life. It could be because of growth, which is favorable or unfavorable. It could be because of the growth of the maxilla per se. It could be because of the surrounding tissue or the musculature. It could be because of environmental influence. It could be because of orthodontic treatment, wherein we do intrusion, extrusion mechanics. Where it could be because of growth modulation or orthognathic surgical procedures, which results in an alteration in the position of the maxilla, the maxillary dentition, influencing the mandible to rotate either favorably or unfavorably, thus altering the maxillomandibular relationship, the orofacial musculature, and in turn the smile aesthetics. Anytime before we plan our treatment, we have to look into diagnosis. We have to do an extensive diagnosis of how this particular gummy smile is. First, we have to take a detailed family history, because as we all know, the gummy smile is usually inherited. Medical history, including the patient's age and overall health and medication history, has to be taken. The facial analysis, the rule of thirds, the lip analysis, both in the static as well as in the dynamic fashion. Then the dental analysis, which includes your crown length and have to look at the incisal margins. The periodontal examination, including the probing depth, the clinical attachment levels, and the gingival recession. The skeletal assessment has to be done using your cephalometrics, conventional cephalometrics, as well as the 3D volumetric analysis. And most important, at the end of the day, we have to do a phonetic analysis also. This is for the postgraduates. Now, what are the values or angular and the linear values that one has to look at when we are assessing the vertical dimension? It's mainly the mandibular plane angle. Whichever mandibular plane angle you like, you can take it. You have to look at the Downs y-axis, the Rickett's facial axis, the growth axis. You have to look at the C-axis, G-axis, and the new MSG angle that is formed between sorry. This is formed between the point M, the sella, and the G point. This particular angle is more important basically because it considers the landmarks that are in the core of the bone, which undergoes less of remodeling. Looking at the linear ratios, you have to look at the total anterior facial height, the lower anterior facial height, the upper anterior facial height, and the ratio, that is the Jarabach's ratio. One can also use the COGS analysis, especially in an adult patient, wherein we have both the linear as well as the angular measurements one needs to use. And this is the newer assessment tool that was proposed by Dahlia L. Bouclé in 2022, wherein she uses two angles. One is the maxillary angle, that is of Rickett's, and the position of the maxillary incisor, which speaks about the extrusion intrusion ratio as well as the proclination of the maxillary anteriors. Coming to the soft tissue analysis, according to Arnett, we have various sorry for that. We have various values that one can consider, including the total facial height, the upper facial height, the lower facial height, and the ratio of the lower facial height, that's the upper 1/3 and the lower 2/3. And when we look at all these values, the most important or the most standard indicator of your vertical growth is supposed to be your mandibular plane angle in comparison to the lower facial height to the total facial height ratio. And these are references for the postgraduates. Please take a picture of this. And any slide that has got a camera picture or a book picture, please take a photograph of that. Then the smile evaluation, whenever we are evaluating a case, it goes into a classification of malocclusion. We have to follow the Ackerman and Proffitt's classification, even though it is not a recent one, it's about 60 years old, but this is considered to be the most recent classification of malocclusion. The systematic evaluation of the facial and the dental appearance goes with David Sawer's method, that is your macro, mini and the micro aesthetics. There are various other methods or forms that are available to evaluate smile, including your New York College of Dentistry smile evaluation form. We have the 8 components of smile by Roy Sabri. We have the diagram of facial aesthetic reference by Alexander Kamara. We have the smile grid. One could create their own smile grid. We have to open up a PowerPoint slide, put an extensive smile picture and draw these lines and evaluate which particular portion of the smile is altered. We have a more systematic way of evaluating the smile. Whenever we look at smile and we have been classifying smile, we are just looking at the amount of gummy smile that is there, the amount of gum exposure that is there. But what particular factor is contributing to that? It could be because of a vertical maxillary excess or excessive maxilla, but it could also be an additional factor that is there because of the lip. So this is a newer classification method that is available. Makada and Rosso classified it into 3 categories, and this is the most recent way of assessing the gummy smile. This is an article by Dr. Elbrook, wherein first they measure the excessive gingival display in relation to each single tooth clinically. Second is to assess the upper lip length at rest, assess the upper lip mobility or the elevation of the upper lip, then assess the maxillary incisor length and the probing depth, followed by the 2 cephalometric values I explained earlier. Then it is tabulated together and we have to evaluate, we will come to know exactly what is the contributing factor to this gummy smile. Sorry for that. Okay. At this particular juncture, we plan something. We hope that everything happens according to our plan. But many a times during various stages of our treatment or various stages of our planning, we get confused or we get lost. To avoid this, there is a necessity to formulate a checklist. This formulation of a checklist is influenced by this great book written by Atul Gawande, who is an American Indian American Indian surgeon at the University of Harvard. So I have formulated a checklist for the etiology of gummy smile. It includes prognatic maxilla, vertical maxillary excess. It could be because of a short or anatomical short philtrum or an anatomically short lip, hypermobility or the hyperactivity of the upper lip musculature. It could be because of a short clinical crown. It could be because of retroclined anteriors. Severely proclined maxillary anteriors also contribute to a gummy smile. Canted occlusal planes, especially in the anterior posterior direction, or even there could be transverse gummy smiles with transverse cans. It could be also because of altered passive eruption, and it could be iatrogenically induced. However, once we know the etiology and we plan our treatment, we should always remember the envelope of discrepancy as my earlier speaker was talking of. Now let's look at few of the treatment modalities based on age and the etiology. Let's look at this young girl, 11 years old, female patient, prepubertal, with prognatic maxilla, vertical maxillary excess, hypermobile upper lip and proclined maxillary in anteriors. To make things worse, she has got a mandibular deficiency as well. The simple solution for this is we have to think of, we need to advance the mandible forwards, we need to restrict the maxilla, we use functional appliances. Is functional appliance alone enough? Well, in this case, at that time, we never knew that functional appliance alone can do all these things, but in that time, we used a headgear in combination with that, and this is what we achieved. Well, there are n number of studies these days which show that only the use of functional appliances, not just the activator, but any of the functional appliances, including your removable functional appliances as well as your fixed functional appliances, they have a restraining effect on the maxilla, and there is a remodeling of the point A that is seen. Now let's look at this young man. He is 14-year-old male patient. Accidentally, when he came to the department, people saw just the vertical excess, the mandibular deficiency, the huge overjet, and they send him for a radiograph, not only this lateral cephalogram and OPG, but also they send him for a hand rest. And the accidental finding in the hand rest was he was in the stage 2 of growth. That means there is lot of growth remaining. Well, whatever we want we can do. So we planned on giving him a headgear and a splint and this is what we achieved. Later on he came back and he disappeared after the correction was done, and later on he reappeared back after his 10th class, and then he underwent fixed appliance orthodontics, and fixed appliance orthodontics was finished almost 2 years later. He was again a irregular patient, and again out of inquisitiveness we repeated the hand rest and found that he was still in the 2nd stage of growth according to the hand rest, and the CVMI also showed the same. So the reliability of hand rest and CVMI staging is questionable. We should not completely blindly believe on it. Similar results can be achieved with a more complicated appliance, what is called as a J-hook headgear. Let's look at this girl. She is a 16-year-old female patient with short maxillary philtrum height and procline maxillary anteriors. So we can't push her into surgery at this age because the maxilla as well as the mandible will grow. So we used simultaneous intrusion and retraction mechanics using a 3-piece intrusion arch in this case. Similar results can be achieved either with a Connecticut intrusion arch or a K-loop retractor or even a utility arch. But there is a sign of caution for this. Okay, most important thing is every action has an equal and opposite reaction. Please don't forget this. So what happens here? Whenever you are applying an intrusive force in the anterior region, there is certainly an extrusive force on the posterior region. That means when you have a gummy smile both in the anterior as well as in the posterior, if you are going to use this mechanics, you're going to make things worse. If you're having a vertical growing patient and you use these sort of intrusion mechanics, you're going to make the situation more worse. You're going to increase the lower anterior facial height. So such mechanics should be used with caution. Now how to avoid this? You have to use an intrusion arch and you don't want the side effect of it. Then you can use some mechanics what Dr. Adit Venugopal in his International Journal of Orthodontics paper showed that he used TAD assisted intrusion arches. But very important thing, when you're using TADs here, you have to use counterclockwise and clockwise screws to get anchorage. Otherwise, every time you activate it, one of the screws will get loosened. Now let's look at this girl. This is a 16-year-old female patient. She had excessive display of gingiva. The parents had demanded that absolutely no surgical procedures can be done, not even gingivectomy. So intrusion retraction mechanics was used using TADs. We used 3 TADs in this case, 1 in the anterior and 2 in the posterior region, and this is what was achieved. Similarly, if you look at the previous case, please note that she has an anterior gummy smile, mainly an anterior gummy smile with proclination. Now let's look at this particular case. This girl, even though after we are almost completing the retraction after extracting the force, but there is excessive amount of display of gingiva both in the anterior as well as the posterior region, and to make things worse, she has got an anatomically short lip as well. The anatomically short lip, if she would have come much more earlier, we could have thought of doing some sort of lip exercise and things and tried to grow the lip, but this is quite late. So what we did, we placed 2 implants. Again, affordability of placing implants is very important. So we placed 2 implants in a region such that it does not affect the position of the roots. Neither we could use extra alveolar implants. So we placed 2 interradicular screws, and I tried preventing the gingival overgrowth as the implant is placed way up than the attached gingiva by putting a separator around. Possibly that was a bad idea because all the elastomerics accumulate more of pathogens as well as plaque. But fortunately in this case, I don't know, the patient was brushing well. There was no pathogen nor plaque accumulation, fortunate to me. So this is what we achieved. And please note that the hook direction is downwards so that I get a force which is almost parallel to the center of resistance of the maxillary dentition. So this is what we achieved, and hopefully that is a reasonably better result than what she was before. Now this is a 23-year-old female patient. Again, she has got maxillary prognathism, vertical maxillary excess, short lip, maxillary procline anteriors. To make things worse, she has got a mandibular deficiency. What is our treatment plan? Mandibular protraction, maxillary impaction. This is a surgical procedure what we can think of. But again, here the patient's demand is no surgery at all. So somehow we could convince her, and in fact I had that itch to try something new. So in this case we tried mini plates, the SAS plates. These plates initially were not available, so we had to modify our surgical plates and get them, and this was done, and this is what we achieved. The patient was also relatively happy, and once this maxilla got intruded, well, there was slight amount of autorotation of the mandible which actually masked away the mandibular deficiency. Similar results nowadays can be achieved using your infrazygomatic crustal implants and the buccal shelf implants. Please note, postgraduates, how do you place the buccal shelf and your IZCs? IZCs have to always go perpendicular to the bone, insert once you get a catch on the bone, and then you have to get it as parallel to the buccal surface of the teeth as possible, so that you will be one, away from the tooth roots. Second thing is you will be more in line with the occlusal plane. And this is how you place the screws, and if you see here, it is an interradicular screw which is avoiding the movement of the teeth in between, but here the extra-alveolar screws are placed more buccal so that the teeth can be moved easily. And coming to the failure rates of these extra-alveolar screws, the bone screws, it's very less, both with the stainless steel as well as the titanium, but stainless steel is preferred because it is more fracture resistant. This is a case of a medical student who was treated for, like, the third time I was doing orthodontic treatment for her. Extractions were not possible because previously she had got her extractions done. 8s were extracted, 4s were extracted, I had nothing else to extract. So the only option I was left with was intrusion retraction using as many number of microimplants as possible. We used 2 anterior screws and 2 posterior screws, and maximum whatever we could do has been done. And please remember, this patient, I took almost 4 years to complete the treatment because the patient used to appear once in 3 or 4 months. Let's not get left back. Now aligners. Aligners, when we listen to aligners, we think that this is the master tool today. Well, aligners are the worst sort of appliances. I'm not blaming the aligners, but the worst sort of appliances to produce intrusion, because intrusion requires very, very light force, which is very difficult for an aligner to apply. And even if we use aligners using our squeeze watermelon seed effect and things, single tooth intrusions are possible, but en masse intrusion of the anteriors or the posterior segment is, posteriors are relatively easier, but the anteriors are very difficult. Okay, so there are different modalities that these days we have used. Chris Chang has come up with placement of TADs in the anterior region, and to prevent this will produce intrusion, but to prevent flaring of the anteriors, as we see in our fixed appliance mechanotherapy, they place 2 bone screws posteriorly and intrude and retract. And if you have retruded anteriors, we have to place cuts in the anterior region. The screws, either place a midline screw or place 2 screws here and apply forces from this area, this particular area, place cuts in the aligners here and produce some sort of intrusion mechanics, and thus we can correct. Please note this, the easiest type of tooth movement that you can achieve with aligners is either flaring or retraction, mainly by tipping, and the most difficult one, as in case of a fixed appliance, is intrusion. Please remember, the reason why intrusion is the most difficult movement is because we have to apply extremely light force. It is a theoretical force, but practically when you are trying to apply 15 or 20 grams of force is very, very difficult. Now this is a challenging case. The challenge is not just the case. The patient per se is a challenge, because she is 38 years old, and the challenge she gave was she has to get married in 3 months. Is it possible to correct something like this in 3 months? And she has undergone orthodontic treatment for 3 times, and this is the 4th time she has come back. I don't know, out of some sort of pressure, somewhere she has found a boy and she has to get married now. 3 months is the limit. So what can we do? Instant aesthetics, instant Maggi noodles? Yes. We did surgery first approach in this case, and leaf out 1 impaction with AMO setback. A modified AMO was done with the extraction of 6s, and this is what we achieved. And then she came back 1 year after her marriage, got her fixed appliance, and this is what we achieved. This is what correction we have achieved. Okay, please note this case. This is another challenging case, 33-year-old male patient, a politician from northern part of Kerala. I hope all those people from Kerala can understand what this particularly implies to. And this guy came with almost like 10 millimeters of vertical display of the gum, prognathic maxilla, proclined anteriors, and vertical excess. We did conventional pre-surgical orthodontics, made the situation more worse. My situation is bad here. The patient and his gang will come and beat me up. Somehow we convinced him for surgery, and this surgery was done here. High Lefort-1 impaction with lip lengthening chirioplasty was done, and this is what is achieved. Surgeon extremely happy. Surgeons have got some sort of extreme skill to convince the patient whatever they do is the best thing they can do. I'm sorry, sir. Where is he? Sherry sir is not here. I'm saved. Fine. So surgeon happy, the patient happy, because patient is already convinced. But you know what? This khujli, itching. As orthodontists, what have we done? We have not done much. We made situation worse. And that is when something new is running around in the market, and that is running around in our head. We need to try this. So that is when this ad of Pepsi was coming up, and Dil Mange More, and then we use this botulinum toxin, and this is what we achieved. So he looks much better, much younger and much better. This is another case I treated during my postgraduate days. That is almost like 20 years back. So the challenge in this case was half the department was in the opinion that there is a maxillary prognathism, half was on the mandibular deficiency side, but this particular true vertical saved this mandibular deficiency group. So the boss is always right. The boss told no surgery in the maxilla, only the mandible will be advanced. So we extracted the force and retracted, intruded using a continuous intrusion arch, and then we went on for a mandibular advancement using a BSS advancement, and this is what we achieved. Relatively good result, even though the gummy smile or its lower incisor display and all increased, but he is far better than before. Well, where is gummy smile now? Here. This is a case the patient presented with no gummy smile. Absolutely no gummy smile. She had a relatively good smile except for severe crowding in the upper anteriors and the lower anteriors with instanding lateral incisors and so. So we did our orthodontic treatment. We opened up the space. Non-extraction was a challenge that was given to us. We used self-ligating appliance. We expanded the arch a little bit. We proclined the upper anteriors. Got the laterals in position. But now the new problem has come. The patient has got excessive display of gum, as well as the color of the gum has become bad. Now what to do? Yes, very simple. We went in for gingival recontouring, that is gingivectomy using lasers, as well as we did depigmentation, and told the patient after few months this depigmentation is going to come back. Well, now she's happy. This is another case with vertical maxillary excess, short philtrum, bimaxillary protrusion, and altered passive eruption and short clinical crown. This short clinical crown is induced by us. Iatrogenically caused, since the same mechanics as I used in one of the previous cases, wherein we placed the TAD lateral to the more distal to the canine, and we did some intrusion retraction mechanics, which resulted in short clinical crown. That is altered passive eruption, and gingivectomy was done, and this was achieved. That's fine. And conventional, no additional mechanics, no use of TADs, no intrusion arches, just used curvoscopy to produce intrusion retraction mechanics. Controlled intrusion and retraction was done in this case, and finally a little bit of gingivectomy was needed, and that was done in this case. Now what is this altered passive eruption? Altered passive eruption is nothing but there is an inability of the gingiva to move when the teeth erupts, or when the teeth is intruded, the gingiva remains right there. It is basically of two types, the wide gingival dimension type and the narrow gingival dimension type. The wide gingival dimension type, where the attached gingival length is normal with alteration in the position of the height of the gingiva or the exposure of the clinical crown. The second type is the narrow gingival dimension type, wherein the height of the gingiva, even though it is high, but the marginal gingiva, the height of your attached gingiva is short. This is further classified into type A and type B. In type A, please note, in either 2 or 1, it is the normal position of the bone and the altered position of the gum. But in type B's, both 2 and 1, the skeletal position is altered. So wherever there is a problem with the bone, cut the bone. And wherever there is a position with the gum alone, cut the gum. And this is a surgical procedure that was done using diode laser to increase the crown length. And there are various modalities of treating a short clinical crown. We have to identify where exactly the problem is and then treat it. If it is the problem with the gingiva, treat the gingiva, resect the gingiva, either do a normal gingivectomy or do a repositioning flap, either superior or inferior repositioning flap according to the need. If you find that the clinical crown is short, when you look at the crown root ratio on the radiograph as well as clinically, then one has to deal with either increasing the crown length using composite buildups, or you can go for crowns, laminates or veneers. This is not attempted by us, it's a more tedious procedure, but one of the Chinese group has published this. They have used a MAFT gun wherein they have auto-transplanted fat from various parts of the body onto the face. It's nothing but something very similar to fillers that we use, and this is what was achieved. Then this is the case, this article is pertaining to the use of hyaluronic acid fillers. They are considered to be more safer because hyaluronic acid is as such present in the human body. These are various other surgical procedures, non-surgical procedures that can be used to correct the gummy smile. Now this is a very interesting case. Why interesting case? There are so many interesting cases I showed, but this is specifically interesting because this patient was misdiagnosed and treated for over 25 years as a case of trigeminal neuralgia. Now where this trigeminal neuralgia came in this person, and why I am showing this identity of this patient is because the problem I am showing is in the mouth, but actually you look into her eyes, she is in pain, agony, she is in great agony. And what was causing this agony is this retrocline anterior. Why she came to me? It was basically referred from the prosthodontic department because she wanted some teeth in the lower arch. She went to the prostho department, they told, okay, you go to ortho department, ask them to push the incisor a little forward so that we can get some space. So the vertical dimension of this case was corrected. We used a night protraction arch, and once we proclined the anteriors, instantly the patient had pain relief. Where is trigeminal neuralgia gone? Where is the trigeminal nerve gone? Everything is there. Nothing has happened. It's just the mandible has got protracted forwards, and that's what is saving her. And there are many situations. Please remember that you don't have to correct all the gummy smiles. This is what is called aging gracefully, and most of you know this beautiful lady. She's one of the most beautiful ladies on earth. That is Benazir Bhutto. In her early 20s with excessive gummy smile, in her late 50s, just before she died, she had no gummy smile. And I had told you to watch some cases. Okay, please note these cases. 1, the vertical impaction of the maxilla surgically or using your dentoalveolar mechanics will disastrously age the patient's face. Look at the patient's face. We have spoiled the face, but the patient is happy because the gum is not seen. Let's give it 2 minutes. Next one, the dentoalveolar intrusion of the maxillary anteriors rotates the occlusal plane, especially with the continuous arch mechanics, and this will increase the open bite situation. Especially these sort of situations happen during COVID when the patients disappeared. The over retraction of maxillary anteriors will result in disastrously flattening of the face and destroying the whole smile, and additional caution should be taken to avoid iatrogenic damages to the roots when we are doing various surgical procedures. And this flowchart is for the postgraduates. Please take a picture of this. This is the gist of all that I presented. Done? And at the end of the day, what matters is smiling patients, happy patients. And in conclusion, one must plan their treatment, select their appliance, mechanotherapy based on the individual needs of the patient. It's not like some cookbook mechanics that you follow. The boss has told, put this wire, I'll put this wire. Next case which will come, sir had told in that case I should put 014, 016, 018, then we go back to 1622, 1725. No, you have to plan your mechanics based on the patient's need. Every patient is different. And well before you have started your treatment, you should plan your retention also. Including retention, you should plan well before. And you should get it accepted by both the patient and you should satisfy yourself also. That is the key for orthodontic success. Thank you very much. And there are situations wherein leaving a gummy smile alone is fine, when they can turn into Miss Italy and so. Thank you very much.
Thank you, sir. Over to the chairperson.
Dr. Vinith, that was a very interesting and comprehensive presentation on this gummy smile. I would ask the audience if they have any questions. Interested in asking any questions? Yeah, Dr. Hari Krishna is there. Please hand over the mic to.
Sir.
So put it on.
Dr. Vinith, I just joined few slides before, but I can understand the crux is like, I remember the 1999, I attended a lecture by Bjorn Zachrison in British Orthodontic Society meeting when I was working there in Glasgow. So what he was showing that as age advances, the upper lip sags down.
Yes,
that is the crux here. So we should not over intrude things so that after 20, 30 years, the beauty diminishes.
Yes, sir.
So that's the crux I can understand. And apart from that, fillers and other things, I don't know, we have to explain to the patient it's temporary. Yeah. We have to redo it again.
So fillers are relatively more permanent, relatively more long lasting than your Botox. Botox basically is for 6 to 7 months. And then you have to keep on repeating this. And most of the people who go for this are the people who are in the beauty industry, the superstars and models and so on. So they are all the time in the beauty salon other than when they are out for the shoot. So it is something like that. They will visit the cosmetologist, they will get these things done, they will smile around, and then once they feel that they have started getting aged, even before this diminishes away, they will go back for the repeat.
And surprisingly, the only surgeon I have seen in the world, Dr. Rodney Roy, plastic surgeon in Texas, he does a upper lip surgery where he says like the upper lip lengthens. Only it's a very small minor surgical like procedure, but only he does. Nobody else I have seen in the world does that surgery. And he says like gummy smile can be treated. And but he also mixes up with orthodontic case, so he doesn't have a good case series of showing the exclusive surgical outcome. Yeah. And another thing, like the sign of beauty, a little bit of gummy smile is a sign of beauty. You see that in many times in beauty pageants, contests. So we have to understand that as well, not to overdo things. Yeah. Thank you.
I think with this we'll conclude. Thank you, Dr. Vinith. That was a very nice presentation and you have nailed it. And I think that will be also very helpful for the PGs. Thank you.
Thank you, sir. I would request the chairperson to please felicitate Vinith, sir. I would request Dr. Puneet Batra, sir, to please come on stage and felicitate Ravindranath, sir. Next up, we have a panel discussion. It's just a short 2 minutes break. Next up, we have a panel discussion. The topic is risks and rewards of adult jaw expansion. The panelists are Dr. Sherry Peter, MBBS, BDS, MDS, Orthodontics, FDS, RCS, and FRCS. Currently, Sir is the lead consultant, Department of Craniofacial Surgery and Orthodontics, Aster MedCity, Kochi, Kerala. The second panelist is Dr. Pandurang Harikrishnan Sir, MDS, FDS, RCS England, FCR, Orth Taiwan, FIAO USA, MBA and PhD. Currently, Sir is the professor and head, JKK Nataraja Dental College, Tamil Nadu. The third panelist is Dr. Ejaz Anwar Sir. Sir is a consultant orthodontist, MDS, MFDS, MOrth, and RCPSG Glasgow. Sir is also a member of IOS and American Association of Orthodontists. Our fourth panelist is Dr. Venkat Ambekar Sir. Sir worked as an assistant professor for 4 years and later on shifted to full-time private practice. Sir has many national and international publications to his credit. The moderator of the panel discussion is Dr. Puneet Batra Sir. Sir is the principal, professor and HOD of orthodontics at the Manav Rachna Dental College, Faridabad, Haryana. He's a M.Orth from Royal Colleges of Surgeons, Edinburgh, and an FFD Orth from Royal Colleges of Surgeons in Ireland. He is presently the president-elect of the Indian Orthodontic Society. Over to Sir.
So I welcome all the panelists to come and join us on the stage. Dr. Sherry, Dr. Venkat, Dr. Ejaz, Dr. Hari Krishnan. So, the topic we call the great orthodontic gamble is basically the risks and rewards of adult jaw expansion. See, many of us, when we get an adult patient in our practice and we have, we see there is a constriction and we need expansion. Many companies say that their self-ligating brackets can do the magic expansion. You do not need to have any other aspect required. There are many people who have designed number of appliances. I talked yesterday about customized appliances. There are some cases which will do well with them. There are some cases which will go awfully wrong. So what's the current update? These are 4 panelists there today who are experts in their fields and who have delivered good care to their patients. So let's have a look at their aspects and their vision on what should be an ideal case for an expansion with a particular modality. And once we are able to have this discussion, we will come to some conclusions about how, as a postgraduate, we should be looking at a patient who presents to us, an adult patient, and what expansion modality should we consider in the best interest of the patient. So I welcome all the 4 panelists today. And it's not just one day work. There has been a lot of background work done by all of us to actually bring it forward to the audience. The controversy surrounding orthodontic expansion centers on the potential impact on facial aesthetics and also on long-term stability. What we look at the end of treatment should also remain stable. And there are evidence-based studies which will aid in reaching out to amicable conclusions to the latest controversies regarding the risks and the rewards of this expansion. However, these 4 people who are on the dais today are experts and they can tell us what pitfalls they have faced during their careers and what could be a guiding light for the Gen Next to understand about where you need to go from the realms of non-surgical methods to surgical methods of expansion. What are the criteria for case selection for surgical or non-surgical jaw expansion in adults? And we have a leading surgeon who's also an orthodontist who can give us an input into how he looks at cases when he has a referral for a surgical jaw expansion. So can I request each one of the panelists to kindly give your opinion on how you plan a case for an adult expansion? We can start with Dr. Venkat, please.
Hello.
Good afternoon for everyone. So whenever we take a case and when we check intraorally and when we see maxillary reconstruction, the first thing what we focus is what is his age of the patient. So that tells us like if the patient is young, then we generally don't go for any mini implant assisted or SARP. Rather, we directly go for the Hyrax. And then we put the screw and then we go for expansion of the maxillary arches. And most of the time, these class 3s are associated with maxillary deficiency and mandibular prognathism as well. So it's also like that when we are giving an expansion devices, we give reversible face mask also, so that the downward and forward growth of the maxilla is enhanced. But at the same time, in my experience, what I have noticed is for one girl, she was around 12 to 13 years of age, where we gave her an expansion appliance, and we had been working and we have been teaching her parents to screw open up the screw. For 3 months, there was not even 1 millimeter of expansion we noticed for that. At the same time, we have given an expansion device for a patient who was almost 20 years of age, and we had because of due to his medical legal issues like complications of his health, we had given an MRP during that period of time, and we had just given an RME Hyrax, and it expanded very well for that patient. So the note here is whether the suture has been closed or open. So we just have to make a note of it, whether, like if you, as I was talking with now, like out of 100 cases in adults, suppose we see, most of the time 80% of the patients, adult patients, the sutures may be fused. That time you may have to take the help of surgical assisted or mini implant assisted. Whereas the 20% of the cases, you can go with the Hyrax expansion as well. So when like the area where I'm going to work, like where it's a rural area, we don't have a CV city also. But that areas we have to do a trial and error method, but we have to inform to the patient prior only. If the expansion doesn't work, then we have to go for adjunct procedures and we have to take the help of a surgeon to give some cuts and corticotomies so that we get a good expansion in that area.
Yeah, thank you, Dr. Venkat. In fact, those of you who don't know, I call him the orthodontic factory. You know, the number of patients he finishes per week, I think many of them do not start in a year. And he serves in a rural belt of India where he is giving service to so many patients who don't have access to care. Like he said, there is no cone beam CT itself for diagnosis. Dr. Ijaz, would you like to?
Yeah, so what we are conventionally taught was that as the age advances, the mid-palatal suture fuses. So after a particular age, because the mid-palatal suture is fused, the only option for a patient to get skeletal expansion is SARPE. But recently in the 2000s and in 2010, there are many histological studies. The initial study, what we learned was Burton-Melson's classic study in AJDU, but many histological studies, they did cadaveric studies, and they found out that these sutures are not fused. Even in adults, the sutures are not fused. They are complexly interdigitated. So that was a finding they came up with. So it is technically wrong to call the suture fusion. If you see a histological study, even in an adult patient, a 50 or 60-year-old patient, the sutures are complexly interdigitated, interwoven. So this complex interdigitation radiologically gives us a picture that it is fused. So that idea came to a conclusion that we can, with the help of mini implants, we can transmit these forces directly to the bone and we can get skeletal expansion. So the many pioneers, including Dr. Von Moon, Dr. Ki Jun Lee, Dr. Benedict Williams from Germany, all these, they have many designs of Marpace. And first, they tried it on children. So the current where we are standing is that in a 15 or 16 or 20, 25-year-old patients, we can easily expand with Marpace. But as the age advances, it depends on the age of the patient, the mid-palatal suture maturation status, many other factors. But till 20, 25-year-old patients, we can easily expand, which was only possible with a SARPE. But with the advancement of customized Marpace and many other surgical assist protocols, even the age barrier is broken year by year. And now many adult patients are also treated with modified techniques using customized Marpace and all, which was only possible with SARPes.
Yeah. So, Dr. Venkat told you that 20% of the patients will not need any expansion modality. Just regular RME screw, which you are using, would be active in those patients. Some of them, just with, a bracket system itself, can get expansion just with wires. And like Dr. Ejaz mentioned, the age at which you can do, you can omit SARPE and still work on MARPE is going on increasing, and the sutures may not have fused at the age we've learnt in textbooks. So can we get the experience hand of Dr. Hari Krishnan to tell us more about it?
Good afternoon, folks. I think the topic was different in the brochure, and the title of the presentation, Dr. Puneet put it as Great Gambling. So welcome to Las Vegas of the convention. So it's really good. It's a really gamble. Yeah. When you see a patient, you want to try simple slow expansion or rapid maxillary expansion or SARPE or MARPE. There's a menu of treatment planning available, and you are now going to gamble which one you want to do in this patient. Yeah. So it's before there were no enough CBCT availability and other things, but now enough diagnostic procedures are available. We can see the main thing we concentrate on is the mid-palatal suture. Yeah. The mid-palatal suture, as my colleagues, other panelists said, we always think it fuses like 15 years of age, and after that you cannot expand the maxilla, yeah? Because the mid-palatal suture doesn't allow you to expand. But if you see the literature, 11 to 15 years, the fusion or maturation of the mid-palatal suture is only 60%. Yeah? And there's enough literature showing like 15 to 20 years, there is 50% of suture is still patent open. Yeah? And again, from 20 to 25 years, literature shows clearly 50 to 60% of the suture is still patent. So what this shows is we have an opportunity to do till the age of 25. Yeah. I've done many patients like adults 28 and 32 and all with not even hyraxia. I put, if the palatal vault is very deep, I try to give a removable expansion appliance itself, and I am able to get 5 or 6 millimeter transmolar with expansion here. So what we want to understand is the gambling is like orthodontic expansion. Literature shows there is an age limit up to 25 years, and then you have a CBCT maturation criteria to analyze. The article in 2011 by Fernando and Lorenzo Franchi and Jim McNamara's article clearly shows you can stage the MPS suture maturation, and accordingly you can do, even if the patient is adult, you can do only orthodontic expansion here. So we have to always keep in mind in this gamble, the orthodontic expansion possibility in adults is always vital here, and patent available for you. And it's quite easy, not scary for the patient, quite convenient for the clinician and for the patient as well. The compliance is very good. So we keep in mind in our agenda today, like orthodontic expansion possibility is well available. Yeah. Thank you.
Yeah. So he mentioned something very important, that he uses a slow expander, just an expansion screw also in some patients, which have a very deep palate. So can I come to Sherry, who is going to give us the surgeon's perspective and what he thinks would be his reason to do a SARPE rather than choose a patient for any other modality?
The older system was your choices were to do a surgical or an RP. Now that the implant-based expansion has come into the scene, you have another choice to the whole thing. Now, surgery-wise, when the other things fail and cannot be possible, you can think in terms of a surgical expansion, which is two types, either as SARPE, which is a surgically assisted. So you're going to put an expander in, cut the bones, and expand it. Alternatively, you can combine it with an orthodontic surgery or a procedure just for expansion. So you cut the maxilla into 2 or 3 pieces, or whichever you want to, depending upon what kind of expansion you need. And then you fix the maxilla, get the occlusion which you want, and plate the maxilla. So between the two, you are thinking of one single procedure finishing or two procedures. So if it is SARPE, you expand it, go back to orthodontics and come back for your orthodontic surgery if that is combined. Or it is one shot with orthodontic surgery, combine the expansion as well.
Yeah. So again, from the surgeon's perspective, it can be done on the table. In the OR, you can have a 3-piece maxilla, you can have 2-piece, and you can expand and bring the maxilla to where you desire. Or you can make the cuts and you can do the expansion subsequent to the surgery. So would you like to show your case?
As an example, I thought I'll just show one case of a 20-year-old male with a skeletal open bite, maxillary constriction, maxillary hyperplasia in class 3. So that's the intraoral picture of the open bite in a class 3. He had no midline issues, and that's the VTO we did on the cast. When we did that, the occlusion was not matching, so we split the cast in the middle and tried, and we got a reasonably good occlusion with a canine class 1 lock. So that's the kind of dimensions we are looking at. We are at the molar region, we are looking at a 4.5 mm difference, which is quite amenable for a surgical expansion, which should be reasonably stable in long time. And that's the moment which we predicted. Maxilla forward, expansion, surgical expansion, PN is up, and mandible auto-rotates into correct position. And that's the planning. Superposition, you can see that the upper incisors have come forward. Overjet is fine, PNS is moved up, and the maxilla is forward with the mandible auto-rotated. And there are a few things which needs to be looked at during stability-wise. So you can see that over in the other corner, you see the buttress is being plated, as well as the pyriform rim, which is standard in any left-forward osteotomy. But we have put a bone graft in, that's a piece of bone graft sitting there, which will prevent the maxilla from collapsing in, and the bone graft is separately fixed, so that it is independent, the bone graft fixation is independent of the whole thing. Over and top, you have a plate over here, which holds the maxilla in the midline, wherever expansion you want, but the posteriors are not fixed, so hence to allow the posterior to hold down, we put a TPA with a palatal button kind of thing, and that's wired on. On additional, there is an Eric arch bar as well. So this is an SFOA kind of approach. And over and all, you have a splint, and the splint is wired on. So you have the posterior expansion held back and retained, anterior expansion in the bony wise, as well as on the dental level to prevent relapse. Now these splints are going to be there for 8 weeks, and the TPA will remain for 12 weeks. And this is after the splint removal and the TPA removal. So that's a TPA still in place. And that's strapped up. At that point, the TPA, which is removable, has taken out, and a fixed one with extension has come in. And that's the settling phase. So you can see the transverse width is still being maintained. So it's important that you keep the transverse width and retain it as much as possible, because there's always can be a relapse. That's just pre and post.
Right. So, again, surgery has its, it needs to be, it isn't as simple as the master surgeon has shown. It again requires a very skilled hand to do the procedure and also plan retention. You see how meticulously the procedure was planned. Yeah, Dr. Ejaz.
So just to build up on it, so this is not SARPE. There is no expansion device given here. This is surgical expansion, and it is different from SARPE, which is surgical assisted rapid palatal expansion. So wherein we'll give a leaford one cut, and then we give a expander. So here no expander is given. So that's the difference. You might have noticed just for PGs to see.
Yeah.
Right?
So now, in non-surgical modalities, as a postgraduate, we have to link up with our interdisciplinary colleagues in surgery and plan for cases. But many times, if that access is not there, or there are cases where the patient does not want a surgical procedure, what do you do next? You want to try all the methods which are in your hands for non-surgical modalities, but again, there is reluctance to accept expansion in many situations. When the evidence-based literature demonstrates success in non-surgical trans-arch expansion, it's time for a paradigm shift. So, there are potential complications. Now, will the posterior teeth tip? When Sherry talked about an auto rotation, is it going to be the opposite way around? You're going to develop an open bite while you're expanding the maxilla? Will the bite open and the patient will become a more severe malocclusion than what it was before? to do it or not to do it are the questions. So, Dr. Hari Krishnan would like to add on how he plans expansion in his adult cases.
So, yeah. So as we are now moving towards evidence-based literature, yeah, but there's very less articles published on that. But whatever available, let us look at the literature. Yeah. So this is a publication in AJODO 2011 point counterpoint section by Dr. Chester Handelman from University of Chicago. So where he shows like they use Haas appliance expanders, like 29 cases of his own retrospective cases and 18 cases from Andrew Haas practice itself he has taken, totaling up of 47 cases with mean age of 37 years, 30 years, quite good adults, yeah, for expanders, yeah, normal orthodontic expanders, and a control of 52 adults. The patients had treatment of Haas expanders for 12 weeks, and then they want to see like what is the efficiency of the mechanism, what they have used. So what is the outcome of their whole analysis showed is like this very good expansion was present in the adult patients, and the average was 34.6 in first molar region and 4.7 in premolar region. And there's no big difference between the expansions, and the model contour analysis showed not a big buccal tipping of the posterior teeth, but it showed a bending of the dentoalveolar complex. Yeah, the palatal slope and the dentoalveolar bone bends because of the forces applied by the expander. So there is something called bone bending and bone drifting happens. Within that, there's a very minimal tilt of that. So one of the potential complications raised in orthodontic expansion is like teeth tilting out, fenestration, dehiscence, gingival recessions and all. So these things really, there's no literature showing much. Yeah. And when he examined the adult patients with displacement in children where there were like the comparative group 9.5 years of median age, who is a younger age, who had adult expansion, they had similar expansion, yeah, similar amount of adult expansion is achieved by Haas expanders. And mind you, like the younger patients, it's not like totally the mid-palatal suture opening. Even the age is lesser for mid-palatal suture. 50% of that only is through mid-palatal, and the remaining 50% is by the bone bending effect, the dentoalveolar complex bending itself, apart from tilting of the teeth. Yeah. Right. So the complications analysis, as you can see, like the maxillary molars tilt 3 degrees buccally each side, and the bone itself is tilted. The bone bending effect is tilted like 4 degrees. So there is a big amount of bone bending and then a very minimal amount of teeth tilting within the limits, not causing any potential complications on the buccal bone or gingival side. And also this has been proved by Garrett and Cartelian, because this is a quite old study, but still I think now we cannot do such study because of ethical issues. But they have taken bone biopsies on the buccal side in expansion case and shown bone apposition. And on the palatal side, they have taken biopsies and shown bone resorption. So really the whole bone complex is moving. The bone is drifting towards the buccal side, which is a good thing for us to look into adult expansion only by orthodontic means. And looking at gingival recession and crown lengthening, their data shows like very minimal, which is equivalent to the control or which is present in the normal population. So that is not a big claimed potential complication of doing only orthodontic expansion. Yeah. So the biological basis of this orthodontic expansion in adults, we would like to put it on, there's enough forces generated by expanders. And if you read the study of Chester Handelman, the methodology, he says like they started with quarter turn per day, the Haas expander. Then they shifted to alternate day quarter turn, yeah, which is like a very slow expansion which allows in the adult bone to form. This apposition and resorption can happen on slow expansion. So there is even no need to, even they have put a rapid expander, the expansion is done in a very slow manner. Yeah. So I have also faced this in adult patients with a normal removable maxillary expander itself. I was able to get like 4 to 5 mm in adult patients, which shows like a slow expansion really works in adult as well. Yeah. So the net expansion we contribute from orthodontics, pure orthodontics only due to bone drifting and bending without any complications. And the surgical expansion, as Dr. Sherry said, the outcome of only orthodontic expansion matches with surgical expansion, without expanders, surgical expansion. See, in 1994, I worked with Dr. David Frost from North Carolina, who is William Prophet's oral surgeon. We did many cases of mid-palatal split along with osteotomies, and we could expand like 3 to 4 millimeters. It's up to the limit how much your palatal mucosa can give leeway, which is an immediate transverse correction. And once you do it, the buccal occlusion improves and you get a very stable result. The relapse rate is very less. But the only cut we made different from what Dr. Sherry showed is we always did a paramedian split in the mid-palatal area to avoid the nasal septum sitting in the midline, and we don't want any nasal septal shift after the osteotomy of mid-palatal split. Yeah. Thank you.
So you know, the word of bone bending and bone drifting is something you should be aware of while you plan expansion in patients. You know, what's impregnated in our mind is that we are either having a dentoalveolar tip or the bone is moving parallelly. So there is a bone bending effect which is there. Now, what are the factors which influence adult expansion? You know, we would like the experts to talk on in light of whether, you know, surgically assisted rapid palatal expansion is done or MARPE is done. What are their desirable and undesirable effects, and what are the factors you consider important for expansion? So, can Dr. Ijaz start with his opinion?
So there is a recent study done by the Yonsei University. So they concluded that predictors of mid-palatal suture expansion in adults. So the main factors are, one is the gender of the patient. So it's quite different male and female patients. So male patients, it is quite difficult to expand after 25 years. So the failure rate is much more higher. But take in case the same case of 40-year-old female patient, the success rate is much high. So even a 60-year-old, a 65-year-old female patient, using a customized MARPE or any advanced MARPE procedures, the success rate is high. But in a male patient, it is quite low. A 25-year-old male patient, the success rate is low. The reason being said is that it can be due to the hormonal variations in male and female patients. So the second factor is the length of the palate, the thickness of the bone, and the mid-palatal suture fusion, as told, the Angelini classification. So these are the 4 criteria they found out, and age, gender and age being a very critical factor. Gender means male patients are difficult to expand than a female patient. As the age increases, that means you take a case of 20-year-old and a 30-year-old, a 20-year-old patient is more easy to expand. Then the total palate length, if the length of the palate is too long, difficult to expand. If the thickness of the bone is too high, it's difficult to expand. And if the mid-palatal suture is in the range of E, then it is also difficult to expand. In all these cases, you need you cannot expand with a conventional MSc or a MARPE. You need some modifications, you need some surgical assist, or you need some customizations.
So, you know, there are multiple factors which will influence your success. Can I request the factory I was talking about to quickly take you through 4 or 5 cases and his experience? Because, you know, we are short of time. So as quickly as you can, Dr. Venkat, I request you to showcase some cases.
Yeah. So this was an adult patient, a female patient. She was around 25 years of age. So like when she was like, when we see she was having an orthognathic profile. But once you check intraorally, there was a severe constriction of the maxilla and there was a lot of crowding in the upper arch. And she had a trauma earlier and she had got some root canal treatments there. So what we did was like, we So as I told earlier, like mine was a rural practice, then I have to come with some of the devices which I have to fabricate so to cut down the cost. So I took a regular Hyrex screw and then the plates which normally surgeons use for plating, and I did a modification of this in this fashion, and then we welded or soldered to that of the screw, and then we fixed into the patient's mouth like this, and then we slowly started expanding. So the expansion was good, but at what particular point of time in the second quadrant, the molar started tipping. So I had to cut the arm, and then we started the braces. And so that whenever you expand, according to my experience, 30% of your expansion will be lost. So definitely you have to have some over expansion correction. Suppose if you, so that has to be kept in mind. Over correction has to be needed when you are going to do whichever cases you are going for expansion. And then we gave some self-lightening brackets and slowly started the occlusion. And this is how we finished up the case. This was the OPG. And after 2 years of follow-up also, she was doing out well. It was not like the expansion was retained for her. This is another similar case where she was having a vertical growth phase and she was having a constricted maxilla. When you check intraorally, there was a posterior crossbite and very narrow maxilla. So I gave the same appliance and I modified the appliance. I have placed the implant supported RME, and we got a very good expansion for this case also. And she responded well. She was also in somewhere around 25 to 26 years of age. And then this is how we finished up the case. A good intercuspation also one of the key role so that it doesn't relapse back. So almost from a V-shaped arch to U-shaped arch. This patient, like the main complaint was asymmetry. She was having an asymmetric smile. So when we checked intraorally, she was having a unilateral crossbite, and there was a constriction of the maxillary arch. And the patient's age was 22 years of age. So we gave the expansion, implant-supported expansion appliance for this patient also. And then we slowly expanded the arch. The same thing happened here. The arm was impinging into the mucosa. So we strapped up the appliance and cut down the arms. And once the expansion was possible, so we can see the diastema also, it has been opened, the before and after. Now we can check the asymmetry has reduced. Now because of her marriage, she has got the braces removed. Now we have given aligners for her for settling the occlusion. Now this boy was having a severe crowding and a vertical growth pattern. Now what we did for this patient was, like, we were not able to place the screw for this boy. We were not able to fabricate an expansion appliance for this. So we thought of using an SLB appliance, relieve the crowding. After that, in the second phase of treatment, we thought of putting an expansion device for this patient. But to our surprise, we did some extractions in the upper arch, and the SLB was doing a wonderful job. Here we extracted 2 premolars and 6s because it was having a vertical growth pattern, and now we got a good expanded arch, and we didn't use any expansion appliance here. Only the self-ligating bracket itself has done the job of this patient. And he was also somewhere around 21 years of age. This is how we demounted the case, before and after. This is a post 6 months photograph. Yeah, this patient, he was having a unilateral crossbite, and the patient had some allergic to some drugs. So we thought of giving him an regular RME screw. He was 20 years of age. So we gave him a regular RME device for him. His age was 18 years at that time. He was having a vertical growth pattern. a regular RME device was like opening, then we inform the patient if we are not able to open up, then we have to take the help of a surgeon to give some cuts so that we can expand further. But luckily, the suture had splitted and we were able to have some dysthema, and then we bonded up the case. And this is how he is. Then this is immediate debond. Then we have a follow-up of 4 years for this patient. It is doing out well. So I have showed you some cases where I have taken the help of implants. The second case, like the category where a self-ligating bracket itself has helped out to have an expansion. And here is the adult patient where a regular Hyrax has helped us to achieve the expansion.
So you see that Dr. Venkat has gone through all the methodology where you can do expansion with either a self-ligating bracket itself, you can do it with your regular expanded wires with RME, as well as Marpe different designs which he has shown, which he is using in places where he doesn't have access to good laboratory support. So, what are the periodontal effects? Gingival recession, dehiscence, fenestration, alveolar bone thickness, and also the effects on the airway. You know, augmentation of the oral cavity, OSA related quality of life, oxygen saturation, and snoring. These are, things which I would request Dr. Sherry Peter to put some light on. Any idea, what is your opinion about the effects on the respiratory benefits versus periodontal issues in such patients?
Yeah, the airway, we have evidence to show that it definitely improves the airway to a larger extent, and it's been accepted now as one of the treatment modalities for OSA, provided the OSA related problem is in the transverse axis.
Right.
Because the expansion is not going to make any substantial difference in the AP or vertical way. So that's a limitation of an expansion. Other than that, in periodontal effect, I think there are divided opinions regarding how the periodontal effects happen based on the kind of expansion which you do versus that is between Marpe and Sarpe. Sarpe, it's a absolutely bony expansion. So the amount of movement which happens is purely bony. And if it is tooth-borne, yes, it could be having a periodontal effect. But if it is bone-borne, it will be extremely minimal if it is a Sarpe kind of situation.
Dr. Harikrishnan, would you like to add on to the periodontal effects?
Yeah, the periodontal effects, I think it's very important. Like in the pre-treatment situation, we have to be very careful while selecting the cases. The patient doesn't have any periodontal problems, and you have enough attached gingiva thickness and enough buccal cortical plate. So these things you have to check it with your CB. You don't start expanding orthodontically when there is a very thin buccal cortical plate, yeah, on the posterior region, yeah. So you have to be very careful. Look on the pre-treatment periodontal considerations, oral hygiene, gingival thickness, and buccal cortical plate, yeah. Regarding the airway, as Dr. Cherry said, the upper airway is only affected by this, yeah. You have a narrow constricted nostrils, you have the lateral wall, you have the inferior turbinate popping in, and there's enough airway obstruction. So you expand it, there are enough CT studies showing that there's enough volume increase in the upper airway nostrils, which improves the patient's breathing and reduces OSA when the problem is in the upper airway region. Yeah? So it's not that, and literature have shown like women have more gingival recession compared to men. Yeah? We don't know whether some hormonal effects are there, but as long as you don't select a bad periodontal patient and do expansion, you are in the safer zone. complications reduce so that you can successfully complete it.
Yeah. So can I go to Dr. Venkat and ask him, you do so many patients, what do you think about retention and relapse? Is there a need for overcorrection? What is the formula you use for overcorrection?
So normally, what we see whenever we open up the screw, we check when the lingual cusp sits on the lower buccal cusp, we stop it. But it's better always to at least to anyhow the relapse tendency will be more when we are expanding. Almost we say 20 to 30%. It's always better 10 to 20% overcorrection is better whenever you do the case because it is going to collapse. At least 20% is going to collapse.
Right. So it's very important to overexpand.
Yes.
Dr. Ijaz, we are talking about customized appliances, future aspects on the biological basis, and faster modalities. Can you show us some work on?
Yeah, sure. So customized appliances are the future of skeletal expansion. So as mentioned, as the age increases, the success rate of MARPE decreases. If it is a male patient, it drastically reduces. If the patient is having a very long palate, very thick bone, not only the zygomatic, but not only the mid-palate suture, the zygomatic buttress is a main resistance fracture. Then there is the lateral pterygoid plate. Then there is the piriform aperture. Then only the mid-palate suture comes into play. When you're taking the resistance structures, the first and foremost will be the zygomatic buttress. So if the zygomatic buttress is too thick, it will resist the expansion. If the lateral pterygoid plate disjunction is also very critical, then the piriform aperture, then only the mid-palate suture plays a role. So as it's generally agreed upon, as the age increases, the success rate of MARPE reduces. So let's take a case. So this is a 37-year-old male patient with a suture with E stage of mid-palate suture fusion. And here the palate length is too deep. There was thick zygomatic buttress, and all the factors as I mentioned, the gender, the age, and all were negative for successful MARPE. So the solution for these patients are customized MARPEs. So what are customized MARPEs? So customized MARPEs are we will first take an intraoral scan, and then we'll take a CBCT, and then we'll combine CBCT with the intraoral scan, and then we'll do a digital planning of where the implant should be placed. So as we know, for successful expansion, we need bicortical engagement. So in some patients, as we go posteriorly with a conventional MSC, the bone will be paper thin. The bone will be too thin to accommodate the expansion. So we'll utilize implants. So different patients will be having different bone morphologies, and different patients will be having different canting, different morphologies. Some patients will be having very thin bone. Some patients will be having very thick bone. So we'll prop for the bone and place the implants digitally where there is adequate bone. It is not for one patient, it is in 6 region, one patient is in 7 region. So if a particular patient has no bone in the posterior region, we are not placing an implant in the posterior region. We are only placing the implant where there is adequate bone for that patient. So that is where customization happens, and then we'll be 3D printing the appliance. So all these appliances are 3D printed except this screw. So this is a tiger screw, a power screw. So except the power screw, all these appliances is 3D printed, including the bands. So the appliance fit will be perfect. So this is after expansion. You can see that all these factors: male patient, 37-year-old male patient, mid-palate suture E, all these patients, we can get a successful split without surgical intervention. And this is and you can take the CBCT. You can see that the expansion we got was a parallel expansion. And another aspect with regard to SARPE is that when we are doing a SARPE, we are doing a Leaford 1 cut without disjunction. So a successful SARPE, the effects will be limited to the Leaford 1 cut. But if it is a successful MARPE, the effects will be till the frontonasal suture. That is what Von Moon study shows. So a successful MARPE is much better than a successful SARPE, provided that this MARPE should be successful, because the SARPE, the effects will be limited to the Leaford 1 cut. So all the airway benefits and all, the patients will be having a much more airway benefit, and the mid face coming forward will be better with a MARPE if it is successful and if it's successfully executed. And in this case, the same case, we can see that there is a parallel splitting of the suture. You can see anti-region into 7, 4, 5. So around 5 millimeter of expansion we got from the ANS to the PNS. And this is possible with customizations, and now customizations are the future, and the age limit is being breached year by year. And many of these adult patients can be treated in the future without SARPE or with minimal surgical assist.
So, is there a time for a paradigm shift? So, while the masters have shown how they think and how they plan their expansion cases and shown you some examples, I request questions from the audience to the masters while they are there. Anything which you think are doubts in your mind? Any questions from the audience? So no questions mean two things. It's all clear. So if we do not have questions, yeah, please. You wanted to ask? Yeah, please. Yeah. Mike, can you hand over a mic to him? वहाँ पर.
Hi, sir. I have one question. In your Prophet in chapter 14 and 6th edition, when you read it, they told that when you do MARPE, you get 8 millimeter, if you do 10 millimeter of expansion, if you do 8 millimeter, you get skeletal expansion and 2 millimeter of dental expansion initially. And after 4 months, it will become 5-5. So 5 millimeter is skeletal and 5 millimeter dental. So total is still remaining 10 millimeter of expansion. But how this is, sir? That I don't understand.
So, Dr. Harikrishan,
you talk about the bone bending phenomena.
You're on MARPE or?
MARPE, sir. It's written in Prophet, sir.
It's given in Prophet when they are using in Hierarchs or MRP?
MARPE.
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 Yeah. So when like in MARPE also there is two things, like there are two, like the screw is there and it is anchored only to the bone. Like they don't have any arms at all. So when you are having your only bone bond MARPE, then you will have more of a skeletal expansion.
No, my question was.
And no, I'm coming. And if an MRP is having an arms connected to that of the tooth, that is the tooth and bone arm, then you will have a mixture of you have some amount of dental expansion also and some amount of skeletal expansion also. And you cannot like make it as there is a lot of difference from patient to patient variation. Like as you said, the suture, if it splits very parallelly, then you will get more amount of expansion and the relapse tendency will be reduced. But if you are like, for example, if you place the screw more posteriorly and nearer to that of the vault, then you will get more amount of parallel expansion. In that cases, your relapse tendency will be less. On the contrary, if you are putting your screw little below the vault and more anteriorly, then you will have a V-shaped expansion and your chances of relapse tendency will be more. And then a question is like at the end of the day, you are telling we are going to get only 50% of the expansion, right?
My question was, initially we get 8 millimeter of expansion by skeletally expansion and 2 millimeter dental. But after 4 months, it's become 50-50. But still we maintain that 10 millimeter of expansion.
Okay, so skeletal reduces actually.
Yes, sir, that's what.
Yes, it reduces actually. Dental component, it's a mixture of, the expansion will be a mixture of dental component and also a skeletal. So,
sorry, I'm interrupting you. Can I ask Dr. Abhishek, who does a lot of cases, would he like to comment on this?
No, taking a bit of your time, okay. What happens is that when you expand, you do get a skeletal expansion initially, and there is a bit of dental component to the whole thing also. Because you have a dental component associated with, because your teeth are getting attached to the screws. But when the relapse happens, it happens very arbitrarily. Sometimes the teeth which are tipped tends to upright because you're not holding all the teeth to the MARPE. And then you need to reassess after 4 months of retention
how much amount of dental component and skeletal component is actually achieved. So there is a lot of things happening in between, because you have created now an unstable situation by expanding as to how much amount of net effect you are getting. I hope I have an answer to you. Yeah. I have a question also.
Yes.
Okay, please. It comes to Dr. Ijaz. I'm so proud of you that you have reached the stage in life, and I've seen you grown over the years. Congratulations to you. But I have a small question for you. I saw a beautiful case of yours about customized this one. Astonishing, excellent. I do believe that Tiger Screw comes from the European sectors of Ludwig and other people, okay, because you don't really get a proper lab doing a beautiful work like this in India. But how do you determine, because you showed a case of stage E in which there is complete fusion. How do you determine, okay, before, now you have a successful case, I know it's good for a retrospective thinking, but how do you decide the number of screws that you will use to expand that suture specifically before? Because you know bone biology is very unpredictable, and that appliance had something like 4 screws on the right side and 4 screws on the left side. We are more accustomed to maybe 2 on either side. So how did you determine beforehand that you need such an appliance what is your criteria of selection of the number of screws?
Thank you for the kind words. Much appreciated. So it's a very critical question. So this appliance was manufactured by a US laboratory called Partners Dental, and they specialize in this. And they have an in-house metal printing facility. So the criteria they follow is by Dr. Mariana Evans, Dr. Lipkin, and Dr. Ting, all these pioneers. So what they came up was that for male patients about 25 years, you need minimum 8 screws. So the concept is, there are 2 concepts. One is the concept by Benedict Willems from Germany. So he emphasizes that implants should be placed in the T-zone, that is distal to the last rugae. So that is where adequate amount of bone is there. And there is another school which is by Von Moon who proposes that you should place the implants more posteriorly along the zygomatic buttress, and it comes along the 6 and 5 region. So in many of the cases in that 6 and 5 region, the palatal bone will be very thin, and the advantage of keeping the implant in the T-zone is that we'll get a very predictable expansion and the implant will be very stable. But the expansion what we'll be getting will be a V-shaped expansion, not a parallel expansion. But the Von Moon group, when they place the implants more posteriorly, the biomechanics is in such a way that we'll get a parallel expansion, but the failure chances of the screws are much higher. So these guys have combined both these, keeping the implants in the T-zone as well as keeping the implants very posteriorly.
Are you looking at a tricortical anchorage?
Not tricortical. So these 2 screws will be anterior. So these 2 screws, the anterior screws will be bicortical in the T-zone. And here you can see I have used an implant of 17 millimeter. So it is around 17 millimeter to get a bicortical in this region. So it is not possible with the conventional MSE. So maybe because of these screws, and there is also a screw like a Haas appliance in the lateral region, just distal to 6, it is going interdentally. So it resists the lateral force also. So all these factors plays a major role in expanding, and I think this appliance design, a 25 to 30-year-old male, above 25-year-old male patients and above 30-year-old female patients, 8 screw expansion has now become a protocol according to some others, and I think I have done around 8 cases. It was all successful.
Right. I would love to see all of them.
Yes, sure.
We would all love
after this.
Anyways, I think we have run out of time. These panelists will be there at lunchtime. You can, learn more from them. Only last 1 minute, 15 seconds each, one take-home message for expansion. Let's start with Cherry. One message, only 15 seconds, about expansion in adult patients. It can be a single word or a small sentence.
Overexpansion and long retention.
Excellent. Dr. Ijaz?
Yeah, customization will be the future, I think.
The gambling is there, really you have to chip in your money and you select which part. The idea is you don't try your luck, you should have a protocol where to put your money.
Thank you.
So that gamble, we have shown enough treatment plans. So select that method and chip in and then get the best result. Thank you.
When everything fails, surgical. Uh, but,
So,
but, but with a word of caution, good retention, good fixation,
Right.
long retention, and make sure the airway is increased and the anterior and posterior is also thought of differentially because relapse can happen.
So on behalf of the Indian Orthodontic Society and the organizers, I'm really thankful to all four of you for your efforts in making this, interactive and very useful presentation. Thank you.
Thank you, sir. I would request Puneet Batra, sir, to please felicitate all our panelists. Dr. Venkat Ambedkar, sir. Dr. Pandurang Hari Krishnan, sir. Dr. Ejaz Anwar, sir. And Dr. Sherry Peters, sir. Yes. I would request Dr. Arun Nayak, sir, to please felicitate Puneet Batra, sir.
Come. Amazing. Thank you so much. Blake. Yeah. Why? It's okay. Doesn't matter. It's anyways a light thing. You have to introduce any chairperson.
Our next guest speaker for today is Dr. Arun Nayak, sir, Dr. Arun Nayak sir is a postgraduate from the Government Dental College Nagpur and has been practicing exclusive orthodontics for the past 19 years. He's the only orthodontist from India to be invited to the Align Top 300 Global Forum at Vancouver in 2023. He has mentored scores of orthodontists all over India through hugely popular and intensively scientific national study clubs. He's also a diplomat of the Indian Board of Orthodontics. So we welcome you here. The chairperson for this session is Dr. Simsa Jacob ma'am. She's a postgraduate from Bapuji Dental College and into clinical orthodontic practice for the past 2 decades. I would request ma'am to please. Over to you, sir.
Yeah. Thank you so much. I hope I'm audible right at the back. Okay, so there we go. So this morning, I actually woke up really early to run a half marathon. And you can find maybe 2 of your professors here also who were there. And they gave me company, so it was really good. And now I'm here to share my experiences with the clear aligners, the present and the future. So a disclaimer, I'm an Invisalign educator, or you can call me a KOL since 2015. And my practice is 50% limited to aligners and 50% braces, fixed orthodontics. And of aligners, I use practically only Invisalign. Okay, so so in these 40 minutes about aligners for you, because the time is tight and a lot of speakers are in a rush. So I'll be talking about what kind of malocclusions you can treat predictably. We'll be showing you some complex cases, only complex cases, which we have treated with Invisalign. Severe crowding, severe deep bites, severe cross bites, class 2s in growing children, extraction case, and maybe a TAD plus aligner case, okay? And of course, an overview of what lies in the future. So the experience with Invisalign since 2015 to 2024 has been a journey like this, okay? So it started, looks like fun, and then you start seeing complications, looks hard, but you still keep doing it, you know, learning, hearing people, attending conventions, hearing more people, and you think that you get it. And then you again have doubts whether some things which you're working earlier, maybe some things are not. And then you really feel that sometimes, you know, you don't know what you're doing, but you keep at it, it actually makes sense. And now the challenge is to try to treat every case of Invisalign as best as I could try to do with fixed appliances. So that's the challenge. And what it will be in future, 2024 to 20, who knows, till when I practice, it's going to be the same, okay? It's going to be the same journey. So where do we stand today, and what kind of cases do I treat with Invisalign? I think 80% of my cases can be treated, though I don't treat 80% of my cases because I always leave that option to the patient. You have to understand, over-promising with aligners will lead to a lot of problems. So non-extraction, proclination, dental expansions up to 2 to 3 millimeters, you can treat fairly well. Distalizations from end-on to class 1, end-on. Open bite closures up to 3 to 4 millimeters. Lower incisor extractions always successful. Midline corrections with IPR, you know how much it's a struggle to correct an upper midline with fixed braces, but this can be done fairly easily. Upper lower incisor intrusion up to 2 millimeter each, and lower maybe more. I'll be showing you some cases of deep bites where we've done more than that. 4 premolar extractions with the G6 protocol in well-selected cases. TMJ cases and periodontally weak dentitions, because you can really control the amount of force. I heard a speaker earlier saying that forces are heavy, but I mean, politely disagree with that. And you can actually treat periodontally weak dentitions. I've seen teeth which are almost ready to fall. You know, you blow the chip syringe and the teeth move, and even they haven't fallen in the aligner treatment. I think it's quite good. However, Invisalign still has weaknesses, okay? So those are posterior extrusion. If you want to extrude molars in a skeletal, deep skeletal pattern, it's tough. Severe palatal torquing of incisors, especially those with small crown size. Big crown size, yes, it may be possible to an extent. Skeletal deep bites which need true intrusion. Maybe a case like this. This is very hard to treat with aligners. I say no to such cases. skeletal anterior-posterior issues in adults where you're not performing surgery, severe premolar rotations, especially lowers, buccal root torque of the molars. However, we see some promise with the newer innovations. Scissors bites in class 2 and class 3, full arch intrusion in gummy smiles. We have champions here. We've seen a gummy smile presentation, and these are hard to treat with Invisalign. You'll just be fooling yourself trying to attempt it. Full arch distalization, it's very difficult. And of course, I mean, it's an evolving technology. What I'm saying is difficult may actually be reality in a couple of years down the line. Posterior protraction more than 3 millimeters. So these are real challenges, okay? So what's the real secret of success with Invisalign? The main thing is patient compliance. So you have to be very clear with your patients that if they don't wear aligners, they're wasting their time and money. It's better to go for fixed braces. The second secret is what I learned when I used to see Dr. Kenji Ojima's cases. If you want to follow aligner handles on Instagram, this is one handle which you should follow. And what I see in most of his cases, or rather all his cases, 99% cases, is that the crown size is really big. Okay, and that's, to me, is the second secret of success with Invisalign. So if you have good crown size, great patient compliance, go for it with Invisalign. I can't say the same with, I've not tried other aligners, but here you can go for it. So let's look at case number 1. It's going to be a very clinical presentation. I'm a full-time clinician. I don't have any academic affiliations. So please bear with me if it's not following your PG presentation protocol. Okay, so let's look at a severe crowding and a phase 2. Again, it's a very complex case where we did a 2-phase treatment for this 10-year-old girl. When we started her treatment, she barely used to smile. She really had a tough time smiling. She was very good girl, very sincere, but she really had a problem, you know, in school and everywhere because she couldn't smile. And if you see, she had incompetent lips and a hesitant. And this is how her teeth were. She had proclined upper incisors, end-on molars, there was hypocalcified enamel, there was a deep bite, there was severe contraction of the lower arch, there was a cross bite in the lateral incisor which possibly locked the mandible. And if you look at the upper and lower arch, severe in the lower arch, you also lost the C space. Still, there is crowding, okay? So you can imagine the extent of this. And these were x-rays. The only good thing we had was that we had growth in hand. She was on CBMI stage 2. She was a horizontal grower. So what we decided to do is use the strength of Invisalign, which is arch development, okay? So we decided to expand the arch. This is probably what you would do in any phase 1 case, okay? Maybe you would use a removable plate or a fixed RPE, or, you know, to expand the arch, or maybe Damon braces and expand the arch. So we decided to do the same thing with the Invisalign, okay? So this was the plan. You have to remember that with aligners for crossbite corrections, you don't need occlusal blocks to relieve occlusion because the 2 layers of plastic works well to disclude the teeth. Of course, COVID came, we followed her treatment, and I lived in Hyderabad. So we followed her treatment over there on video calls, and that's a new thing, okay? I mean, when you come out of your post-graduation and you start working in clinical practice, you'll see there's a huge difference in the way you consult today with your patients. And when they came down somewhere in the middle of COVID, and we saw that we had achieved all our goals. So we had actually achieved good arch expansion, we had achieved the crossbite correction, and we had regained the lost spaces. And if you look at the lower arch, you can actually see where the canine was erupting at its old place. Okay, and now even the D's and E's, they have expanded. Many people say, don't do that, okay? But I feel it's a great thing to do in delayed eruption cases where you feel that the primary teeth will exfoliate late. It actually helps in loosening them and makes their eruption pattern more normal. Okay, so she had that, so we decided to do that. Okay, and of course, then because she had hypocalcification, we didn't place retainers. We gave her a removable. We didn't want to give her a removable, but we had to give something for retention because it was a severe contraction, right? And 1.5 years later, she came. Okay, so you can see that the central incisors have moved forward again. But the good thing was the crossbite was still out of crossbite. The arch expansion actually held, and now all the premolars, the succedaneous teeth, they all erupted into its place. So now here again, we decided to expand because the arch was still narrow and the teeth were still inclined lingually. So we felt it was a good reason to correct this thing with arch development, and you can see that the benefits of regaining space actually allowed the trees to erupt. Here we also had to correct the curves P, and if you see, the lateral incisor again was the culprit in not allowing the mandible. So what did we do? Again, we decided to use the strengths of Invisalign, which is to expand and correct it. Okay, so this was the second phase of treatment where we gave the optimized expansion supports on the upper posterior teeth. We also gave her class 2 elastics to help in advancing the mandible. And we also opted for IPR because she had very big teeth. IPR may be a new concept, and I've been using Invisalign for 8 years now, and touch wood, there's been no problems because it's very, very technique sensitive. You have to master the technique of IPR if you have to do aligner treatments, because most cases don't want extraction. So that's one of the main ways. So you have to learn that technique and master it. Okay, so any case, so now she needed only 23 aligners to fix this, and along with IPR, and 18 months following the start of the phase 2, this is where she was. So we got great expansion, we got great alignment. She wore her aligners well, imagine. Okay, so that phase where we didn't give her a retainer actually worked in favor because that gave her a break in treatment. Because otherwise people will get just fed up, wearing aligners for 4 years, and it doesn't work then when you really need it. Okay, so this is where we landed up. This is now her smiling as much as she wants, and these are the changes that we got. These are the comparisons. So let's evaluate how much expansion we could get with the aligners. So we use the AutoCAD system, which is part of the Invisalign system. And this is the amount of expansion in the premolar region, 4 millimeters. Lower intercanine was 1.8 and intermolar was 0.5. Okay, so it was mainly in the anterior region, right? Now, we also need to remember that retention is important here because of the amount of expansion achieved. So we have now opted to give her Vivera retainers to hold the expansion. You can see here how beautifully the fan-shaped lower incisors have been corrected. And these are tough things to do. These are very tough. You really have to plan your treatment well to be able to get these things into, in your results. And these are the AutoCAD models further analyzed. You can now see the better intercuspation at the end of treatment. You can see how much overjet has been eliminated. And these are your takeaway points. Okay, arch development is excellent with Invisalign. Good crown size ensured better predictability. Teen compliance was remarkable. Okay. Second case. Okay, this is a severe deep bite case in an adult. I've treated around 100 deep bites. Okay, and finished them. Okay, but I choose to show you one of the most complex ones in an adult with a severe curve P. Okay, I heard the doctor saying that it's very difficult to intrude lower incisors, and yes, I mean, with experience we learned that it's even possible. Okay, so this patient had a canted occlusal plane, retrusive incisors, crowded incisors. He had a severe skeletal class 2. You can see it's parallel maxillomandibular plane and retroclined incisors. You look at the lower incisors. Look at the upper intrusion, upper over eruption and the lower eruption, okay? And of course, he had a strong elevator musculature, antigonial notch. These are really tough to treat with aligners. You heard it, right? Because the aligners are posterior intrusive and you are working against the strong musculature to get these changes. So let's see how we progressed. Okay, so these are the findings. I'm sure you can all see that. And this was the treatment plan. Upper lower incisor proclination first to be able to intrude the teeth, leveling of curve P, idealizing arch form, correcting class 2 with elastics. It needed 25 aligners, 250 days, 6 months. unbelievable, right? It is. I mean, I never promise my patients that your treatment will be done in 6 months. I'll be lying. Okay, so you have to be honest and you have to tell them that there may be a requirement for a further additional aligners. But still, at the end of the first round of aligners, we got good corrections. We got some opening of the bite, which can be attributed to a relative intrusion. There was some class 2 correction, but there were still some corrections to be done. So what were they? We wanted to torque the upper incisors. We wanted to intrude the upper incisors for cant correction. We want to idealize the arch forms further and maintain the class 2 correction. So we opted to keep the class 2 elastics, and again, it needed only 15 aligners, right? 15 plus 25, 40, right? And 10-day change. So that's nearly 1 year of treatment already. Okay, and simultaneous staging with retentive attachments on the premolars. This is where we got. Patient was happy, okay, but when you tell them that you got so far and we need to do some more corrections so that it becomes a complete correction, he was ready, okay. So again, it said 16. So 16 aligners more, doc, what is this? Earlier it was 13, now it's 16. So any case, we had to do what we had to do, and this is what we had planned in the second stage, 10 days change, so another 160 days of treatment. And this is where we landed, okay? In a skeletal class 2, strong musculature, severe curve, severely extruded incisors, this is where we landed. And these are the changes which we found. You can see the correction, and on the left side, you can actually see that even in an adult, we got unlocking of the mandible with class 2 elastics and could get the class 2 corrections. And these are the changes that we got in this. And this is what I'm talking about. This is reality. And this I've gotten around 100 cases. And when I conduct lectures, I show them multiple cases, okay, of how we get this done. And there was excellent predictability. And the good thing about intrusion with Invisalign is that it doesn't flare your teeth. You place braces, you'll flare your teeth. After that, you have to give torque and, to correct the flaring, which has happened. Here, you don't get flaring because it covers the labial surface very well, and it doesn't let the teeth flare, okay? And you can see this is reality. Look at the upper incisor torque. You look at the lower incisor intrusion. You look at the bite correction, okay? So that's where we got. Look at this, right? Okay, fine. So these are your takeaway points. Excellent cooperation by a busy adult. He's a CEO and he did its job well. I would invest in his company. Okay, so upper incisor intrusion to correct cant, upper incisor torque correction, effective leveling of curve speed. What I've also seen is for PGs, because you are into research, I would suggest this, okay? I'm tired of telling so many professors, please do this study, is that whenever we intrude teeth with Invisalign, this is another case where we actually used to do overcorrection trying to, get the right result. And we could get this in 10 months in a kid. And what we see is that when the teeth intrude, there is no bunching of the gums. When you place a utility arch to intrude, you instantly see the gums bunching up, right? And this is what I consistently see in all intrusion cases, that the gum doesn't bunch up despite the shorter correction. And this is around 4 millimeters. These days, I actually find it easier to intrude with aligners than with fixed braces. Earlier, we would probably place subapical tads and try to intrude, okay, but to do it, quickly. But this is how it is today. And these are the corrections that we got, okay? So now let's look at a severe crossbite, okay? Whenever you think aligners, don't think money, don't think convincing patients, don't think anything. Think orthodontics. Think orthodontics. Think long term. Okay, you want to be known as a good doctor who does good work and has something to show, and that should be your motto. Don't think, oh, I want to be gold, I want to be silver, and this is rubbish. Okay, so aligners cannot compensate for lack of education, experience, or illogical planning. If you have grand ideas to treat your case with aligners, don't, because it's really difficult, okay? It's not as easy peasy as some companies make it out to be. And Invisalign is an evolved system, but you still need to respect biological parameters. And when I was hearing the previous the group discussion, I realized that it's true. You have to respect biological parameters. So let's look at this maxillary deficiency case where we had a severe crossbite, okay, like on unilateral, severe crowding, and lower incisor crowding. And you know what? This girl is the is the niece of an orthodontist who's been trained by Ravindra Nanda, sir. So, and they hate Invisalign in US, okay? And here I'm treating their niece with Invisalign. So I had a lot of, pressure. Okay, so anyway, so what did I decide to do? I didn't say just wear Invisalign. I said we have to correct this in a more serious manner, and we decided to do an arch expansion with Marpe. Okay, so if you see, she had minor maxillary deficiency, normally inclined incisors, CVMI stage 5, and this was the amount of crowding we had to correct, 9 mm in upper, 7 mm in lower, and we used class 3 elastics to control proclination and also get. And this is the CAD-CAM customized that Dr. Ijaz was talking about, beautiful cases, but this is my work with it, okay, and this is what we got. Okay, within 1 month, we could get this much expansion. People asked, why did you use TADS in a 12-year-old? Okay, it was basically not for getting bodily expansion or rather split of the maxilla, but I actually wanted the expansion screw to be retained when I transit her to aligners so that it holds the expansion, okay? 8 months, we then scanned for the upper aligners also, okay? And this is how she was. Her crowding had reduced because we had done this expansion, and because in 8 months with weekly change of aligners, she was on the lower aligner 32, so the crowding was now lesser. And this is how we evaluated again with OrthoCAD. We got exactly 6 mm expansion in the posterior teeth, 33, 39, 43, 49, okay? With the increase in nasal volume, and the torque was maintained. That's the beauty of it. You don't get tipping of teeth. You get actually maintain the posterior torque when you expand it this phase. And then we moved her to upper arch Invisalign, and this is how the case progressed. Easy, okay, but not really easy, okay. And this is how we kept it for, I said you keep that expander for as long as it stays in your mouth. And she was studying in boarding school. She came back again at the end of 1 year, and it was still there, and that's when we removed it. So these are the post-treatment findings, okay. The SNA actually increased by 3 degrees, so it improved her mid-face deficiency. The upper incisors were actually upright, not proclined, what you get when you place fixed braces. The teeth just flare forwards. Lower incisors were proclined by 3 degrees because that's how we are gaining the space. And when you see the roots, they were all parallel. You can see the expander still in the mouth at the end of treatment. And this was the amount of mid-face improvement that we got. Huge, okay? And no fanned out appearance. And let's look at the gingival, health. The teeth were still in cancellous bone, and that's how she was. And this is 1 year follow-up. It's exactly the same. This is 1 year follow-up. That's when I removed the retainer. Though she had an inflammation there, but the screws were still tight. Okay, and this is 1 year follow-up. Yeah. So these are your takeaway points. Aligners are versatile with TADs and TAD-based appliances, but use it sensibly. Don't use every case with TADs. Don't mix your biomechanics of fixed with aligners. You'll mess up your cases with TADs. Aligner versatility plus orthodontic acumen is what helped us. Okay, you have to use your orthodontic head. Let's look at a next case, which is a class 2 correction in a kid. And I'll rush through this case because it's kind of similar, though we had a full cusp class 2 and we opted for distalization in the upper arch. And you can see this was the plan. So look at down here. That's where the distalization is happening, but don't expect miraculous distalization of 3 mm, 4 mm, 6 mm. No. Up to 2 mm is fine, okay? And upper arch was again proclination, and we wanted the class 2 elastic jump, which was around 3 mm was the class 2 advancement. The lower incisor intrusion, which is so important, bite before jet, is what I learned in PG. It applies here too. Invisalign doesn't change that, okay? But it helps you in getting that, right? And this is what we got at the end of 2 years. One more thing, never promise your patients that your treatment will be 6 months, 8 months. That's rubbish. You will be doing horrible practice, and it's always better that you understand that teeth work on biology, and if you want to finish your case well, it takes time, okay? So mentally prepare them for that. Otherwise, you'll never get finished cases. And this is where we landed up in a growing child. And again, let's look at the intrusion. We did 5.5 millimeters of intrusion because we did 2 rounds of aligners, okay, for this kid. And it's true intrusion because you're not flaring. The white and blue is pre-treatment, white is post-treatment. You can see that they are right on each other, and it's true intrusion. And this is the actual result. Intrusion can be done. And this is the proof of what I'm saying. And again, with no proclination, right? So when you don't have proclination, you can actually get better improvement of your mandibular advancement. This is the A and B correction of 3 degrees. And you can see there's barely any proclination of the lower incisors. And this is how the kid was. So these are your takeaway points. Mandibular advancement is favored due to excellent lower incisor intrusion and no proclination. And this permits more change in S and B. Let's look at an extraction case. Okay, extraction case is a dilemma for everybody who uses aligners. Okay, and frankly, I have not seen any peer-reviewed research on any other aligner system or plastic which people use in name of Invisalign. Okay, but we have treated a lot of extraction cases, and this is just one of them. I want to show you. It's a more complex case because there's no crowding. Whenever there's crowding, if there's severe crowding, it's a great case to treat with extraction because you just have to move the teeth into extraction spaces and your space is closed. There's no headache. But here, there's no crowding. It's pure Bimax. Let me call it Kerala Bimax, okay? And he's a guy from the south, okay? So this is how his teeth were. This is the amount of proclination he had. But if you see where the teeth are, okay? This is, use your diagnostic head. You see this? It's in cancellous bone, right? All you have to do is tip the tooth back and the root stays in cancellous bone. So in your planning, don't go over adventurous to try to keep bodily movement. You don't need to do that. So just look at where the roots are and plan your treatment. Okay, so we opted for 4 premolar extraction and G6 protocol, 42 aligners, 2-week change. I stick to 2-week change for extraction cases. And it was mainly tipping movements for incisors, bodily movement for canines. 2 millimeters of anchor loss was factored, and he needed 44 aligners. Okay, there was no bite deepening. So if you see in the plan, there was no bite deepening. Okay, but when you see no bite deepening, you should understand that when teeth are tipped back, the bite will automatically deepen. But if your ClinCheck is showing you no bite deepening, that means there is around that much intrusion which is happening, and this is serious intrusion, okay, to be done with aligners. And these are upper arch intrusion, okay? So let's see how the treatment tracked. So what I did is I opted for canine retraction up to 3 mm because I feel it helps in maintaining the bite opening. And it tracked beautifully. This patient never came. He just used to upload his photographs on virtual care, which is what we have when we use Invisalign. And he just used to ask for aligners, though he lived in Bombay. And I used to see it's tracking beautifully. So I used to always say, boss, please come, I need to take photos also, okay, and see whether it's real. And this is how the teeth were at the end of canine retraction. There was a slight molar tipping in the posterior teeth, which always, eats my head. But with aligner patients, you also have to understand you cannot be too demanding. And ideally, I would have corrected it with fixed braces. But here we said, forget it. So this is his post-treatment photograph. You won't believe it. He came for orthodontic treatment at this age because he thought it's part of his overall makeover. Okay, he lost 27 kilos in his treatment. And this is how he was at the end of treatment. And this is how he was at the end of treatment. He was absolutely thrilled. And these guys don't want braces. Many times people say, Arey, you should have done it with braces, you got better. I've seen worse cases treated with braces and poor occlusion. So I would be happy that at least some patient who would have otherwise never seek treatment is now seeking treatment because he has that ability. However, our aim should be try to get as good result as you can with fixed braces. It takes time, but it will happen eventually. And these are the changes which we saw, and this is the root parallelism that we could achieve with, and no refinements, okay, no refinements, right? Even I was shocked. I told him we can refine it further and correct the molars and all, but he was in no mood. He said, it's all good. And you have to respect that. So stage of retraction is crucial to prevent bite deepening, and Invisalign G6 protocol works efficiently in well-selected cases, and I stick to 14-day change. So let's look at a last case, which is the TAD plus aligner case. And here I want to explain you why you should use TADs in selected cases. Because aligners work by the push mechanism, not like braces which use a pull mechanism, okay? They work by push. So every surface on the tooth, be it the buccal, palatal, the cuspal slopes, or whatever you have, they are all push surfaces where the plastic exerts that force on the tooth to move it. So this is how it moves. So obviously, if you have a flat cusp, it's more difficult to move, or a small crown, more difficult to move. So if you're moving it purely distally, then this is your push surface. But if you move it distal and expanding at the same time, you're increasing your push surface. So you have to use these ideas in your head when you're using aligner cases. So aligners work by push mechanism, whereas TADs work by pull mechanism. So push plus pull leads to more efficiency is what I learned from Dr. Chris Chang. And they use TADs more rampantly, but our patients are too finicky. They don't listen. I mean, if you tell them TADs, they'll run away. They'll never do treatment. Okay, so what I did here is that I first treated his problem, which is crowding. His problem was not the crossbite behind. His problem was not the class 2 behind. His problem was not that. And if I think of distalizing right at the start, it will lead to many more aligners. And we don't know whether this patient is actually going to do treatment all the way, right? So I first said, we'll first treat your problem, and then we'll deal with my problem, okay? So here we decided to expand, and these were his x-rays before the treatment. Nothing exceptional in that, except that the third molars were missing. So that was a good thing. We had 40-day aligner change over here, and 40 aligners with 10-day change. So already 1 year of treatment just to get him happy. Okay, and we opted for IPR because whenever you decrowd teeth in adults, it leads to black triangles, and that tends to annoy them. They tell you, doctor, triangles aa gaye. ये पहले नहीं थे. Then you have to tell them, आपके दांत भी पहले सीधे नहीं थे, अभी सीधे हो गए. So now we have to do IPR to reduce that, okay? So that way. So this was the plan. And we didn't use crossbite elastics. We only gave him class 2 elastics at the end because we are not sure of his compliance. Nevertheless, he wore his aligners very well. And at aligner 36, he was very thrilled. But now was the time to pitch my agenda. Okay, so I said, see, you got great results. Let's do something more. Let's try to distalize. Let's try to get bite correction. Let's try to further expand. And he was very good. He said, no problems. Never said no for anything. So, I mean, you get some patients like this. But you have to tell these things in advance to patients. You cannot tell them halfway that, your treatment is going to be longer or that. You have to tell them that it will be longer, it will take time and all that. Only then they are mentally ready. Okay, and watch what we did here, because I didn't want to give sequential distalization, which is the norm, but I wanted to do en masse distalization. Okay, because I'd seen Dr. Harshvardhan, he's a brilliant practitioner in Bombay, and I'd seen one of his cases where he did that. So I said, fine, let's try that. Okay, so this is what we did. So if you saw that, we tried to distalize the 4 teeth together, and this time we decided to use TADs. Okay, why TADs? Because you need to reinforce your anchorage. Okay, so what happens is when you're pushing, aligners work with push. So when you're pushing these teeth behind with aligners, it's not the TAD which is pulling it back. The role of the TAD is only to counteract the reciprocal push force which comes when the teeth are being pushed back. Okay, you're trying to fit a longer aligner on a shorter arch. So there will be a push force on both directions. One is favorable, one is unfavorable. And you have to counter that with the right amount of force. So we used a reinforcement of 6 ounces elastics and reinforcement of 6 ounces elastics to help in this correction. Okay. And here we had already got the crossbite correction. Like I said, we didn't use crossbite elastics. The aligner is good enough to correct it because it has a lot of push surface on the palatal. So now it was this mechanism, and he did it very well. Okay, and this was the changes which happened. Here we had started the distalization. Here we were midway through distalization. You can see that the molars, premolars and molars are in class 1. And now in Jan, he is just about at the 43rd aligner, and he's nearing the end of his treatment. And I'm expecting a better, like a good case, you know, to completely finish. So that's how it is. And of course, that crown is going for a new crown, so it doesn't matter really. This is what we got, right? These are tough cases. But what I want to tell you is this is where we are today. This is where we are today, right? And it's not that aligners can treat only easy cases. In fact, if you read Profit, I wonder how many of you all read Profit, but if you all do, then please read it. Even Profit has admitted that you can treat complex cases with aligners, okay? So these are your takeaways from this case. As far as possible, avoid TADS with Invisalign because it tends to complicate mechanics, because the complication lies in our head because of our muddled thinking. And use sparingly in special cases with full patient consent before you order your aligners. You cannot say, oh, I forgot to tell you you're going to place TADS. He'll say, forget it, doc. You never told me this. You better don't use this now. So you have to tell them, you have to show them photographs, what it looks like, how it will be, everything. Only then you're on a safe ground. And TADS actually help only in countering the reactionary forces. It doesn't exert direct force by itself because the aligner itself is very rigid. So if you apply elastic to an aligner, nothing will happen, okay, if you think you're using it for pushing. Now let's see what lies in the future, okay? This is where we are. I've shown you only complex cases because I want to show you where we are with Invisalign or aligners today. And let's see what lies in future. All plastic is not Invisalign. Invisalign is the only peer-reviewed research. I see rampant use of aligners. I mean, I don't know. You know what's the bend bracket? If you buy a cheap bracket, you try to apply torque in it, the wire torques the bracket, not the tooth, because the wire bends the bracket, right? So the same thing happens with poorly researched plastics. So let's see what's coming in the future. Some good. Predictability of leading softwares will improve for even tough clinical situations, which I mentioned earlier. Direct printed aligners, shape memory aligners will be more common. Aligner materials that will allow aligner wear for less than 22 hours. It's hard to believe. I don't believe it, but let's see. In-house aligners will be rampant for limited treatments, which is good. And literature may actually expand, I hope, that to evaluate aligners other than Invisalign, so that we have some competition. You know, otherwise they will rule the roost and the prices will just keep going up. Some bad failed cases may lead to negative publicity for aligners. I already see it happening because people ask each other and they know that, oh, it doesn't work. I've heard from my friend. Then I have to sit them and completely change their thought process that no, it works. And I show them cases, and that's how it goes. Treatment costs may drastically reduce due to orthodontist undercutting. That happens everywhere. And some really ugly, okay? Thousands of mutilated cases to retreat. I don't know if Dr. Ghosh is here, but he's a champion of retreatment. You have to just see his cases and it's just wow. And the cycle of aligner treatment may actually end. We had cycles in orthodontics, right? Extraction, non-extraction, lingual orthodontics, tip edge, and this and that and everything. And it's probably just a cycle which will die its natural death. Okay, so that might happen. Or braces and biology may improve, and who knows what we'll see in the future. But one thing is for sure, braces will always exist. I had a PG student before joining orthodontic post-graduation. His mom is an orthodontist, and he asked me, sir, 5 saal ke baad braces rahega kya? So I said, 100% rahega. Don't have to worry, okay? So here, thank you all for your patient hearing. You can follow me on Instagram. I post cases, I can answer your questions there. Thank you so much.
Thank you, sir. Over to the chairperson.
40 minutes. Yeah, perfect.
Over to the chairperson. I would request the chairperson to felicitate sir.
Yeah, thank you so much, ma'am.
I would request Dr. Rohan Rai sir to please felicitate our chairperson.
I'll take it. Thanks, sir. Thanks so much.
Our next guest speaker for today is Dr. Rohan Rai sir. Sir is an iOS certified orthodontist, started his career as a clinician and as a teaching faculty in orthodontics with a total of 25 years of academic and clinical experience. He completed his BDS from Manipal College of Dental Sciences, Mangalore in 1992 and his MDS from Manipal Colleges of Dental Sciences, Mangalore. He is a former executive member of the Indian Orthodontic Society and also a member of IDA and MOSG. In the year 1996, he was the organizing committee member of the first PG convention held in Mangalore. He has authored publications in both national and international journals. He has efficiently guided postgraduate students and is also a recognized PhD guide under the Rajiv Gandhi University of Health Sciences, Bangalore. His mentored work, Twinkle R 3D Appliance, an innovative functional skeletal class 2 corrector, won the second best poster at the iOS PG Convention 2018. Also, in iOS PG Convention 2017, variation in lip and tongue pressure in orthodontic patients won the second best paper under his guidance. We welcome you here, sir. The chairpersons for this lecture are Dr. Digvijay Patil and Dr. Sheetal Patni. Dr. Digvijay Patil, sir, is a reader at MGV's Dental College, Nashik. Sir is also the head of Public Awareness Committee of iOS. Dr. Sheetal Patni, sir, is the head of IDA Local Branch, Nashik, and also is an EC member of iOS. At present, sir maintains a private practice. Over to you, sir.
Yeah, warm good evening. Thank you so much, iOS, Dr. Sailesh and his team for giving me this opportunity. In fact, second day, when you start presenting in the evening, in a way it's advantage, in a way it's a lot of disadvantage. The advantage is most of the topics have been spoken out. You know, we are like the last few batsmen remaining to play, and very few audience would like to hear them. But still, game is not done. I'm sure this couple of topics will keep you more interesting. So I'm here to share my experience over a period of these past 25 years, whatever has happened with me in my practice. And this is my timeline, how I have progressed. I was a PG like you all when the first convention was, planned. We had to coin a name. So the name was Pogonian. So I was a second year PG. Four of us coined this name. The point was to have the chin in front, okay, as an orthodontist. So registration fees of 350, and we had a participants, the entire PG curriculum, PGs were, numbers were around 150 all over India. So now we know where we have reached now. So I progressed after that, I should say progress, And this is how the timeline was. I started my clinic, Ira Dental, and initial days they were more of a, we were BEG practitioners, trained in BEG, and stage came where, we had to adopt to the newer things. MBT was just happening then. What was aligner and the newer TADs for you all today, the MBT was for us those days. I'm sure this front benchers, I mean, your seniors, our teachers would agree with this, right? Right, sir? Yeah. So I had to adopt or adapt between two things. One is I was doing BEG, and simultaneously I had to take up straight wire as well. And in between, I had to make a decision because I had to keep my inventory very, very less. Okay, so I took over or shifted to predominantly a own 8-slot practitioner for reasons. I'll as the discussion goes on, I'll tell you. Then came a point in life where in 2013 I shifted my practice. Rather, I had taken up few consultation abroad, and that was a turning point in certain ways because I started to reevaluate, be more critical about my cases because that place, the UAE, I had to deliver a lot of good results. Okay, the accountability became there were more stringent accountability. So when you're pushed to the wall, you have to deliver. Then of course, the self-ligating appliances had to be adopted in my practice. And lastly, now we are in an era where I had to make a change and also do a little bit of clear aligners and use of intraoral scanners. So having said that, so today's topic is treating the treated. Now, yesterday, Dr. Raj Ganesh, Dr. Ghosh, they spoke about all the mechanics, which RMM, spoke about the essentialities of what you need to have a good technical hands. Today, my friend Dr. Naik spoke about those the fantastic cases. So, when you come into practice, you are forced to take up all lot of cases. Let me tell you, it's a survival of the fittest now. Okay, competition is so high. So treating the treated, the current trends in treatment of relapse cases. Okay, now let me tell you, it's no different than the protocol set for yourself to treat the first-time cases. It is the same. However, there are certain important things which you need to keep in mind when you address the cases which are going to come back to your practice or those cases which are treated with elsewhere. Now, I always had a high esteem for this man. Now, life always gives a second chance, they say. So, the quote of Ratan Tata is definitely an inspiration for all of us to, try our hands in those cases which are giving us a second opportunity. So, introduction to this case, I mean today's discussion, I was trying to get some articles and I was not very successful. However, I could manage a few of them. Why do patients come back or why are they opting for a second time treatment? It is mainly unfavorable skeletal growth pattern during and after the treatment, relapse after the treatment, insufficient compliance resulting in discontinuation, and the last one is a very important thing. It's the increased self-perception of dental appearance. Rather, people are more and more currently focusing on their aesthetics. So prevalence, the most common age associated with removable appliance therapy was found to be the age group between 21 to 30%, 21 to 30, around 62%. Majority of the patients who reported were females when compared to the males, about 53.8. 50.7% of the study population had retreatment, due to relapse cases, while 49.3 were new patients. This was reported in Journal of Dental Sciences in 2022. So let's see the cycle of events. Okay, this is a typical scenario where you would encounter in your routine practice. Now here is a situation. We have done a small animation which represents, the cycle represents a malocclusion and the individual carrying the malocclusion. So this goes this way, okay? An individual would probably at a stage encounter a problem, and he would probably realize that probably this is a time he needs to undertake an orthodontic treatment. So he would move along. Okay. This is a scenario where currently we are in now. We would probably go to a closest orthodontic clinic. Probably he or she would have been recommended there. It would be a simple orthodontist. Probably years earlier, life was very easy for us to practice. You would approach there, meet the orthodontist and get himself sorted out. So orthodontic correction would have gone through. Over a period of time, when the patient is an adult, maybe he has encountered another problem, more of a serious nature. Okay, so now the patient is more of an adult. So then they start researching. They see, all of us now want to be seen in the social media. We advertise. We want, our cases to be shown. Definitely, marketing is the new thing now, which is happening more so in all of us. I'm sure you would agree with this. So then, current scene is he would meet an orthodontist who has advertised that he's the best. So all of us do that, right? We all want to showcase our cases and then, be seen as one of the best. So this patient walks into your clinic with a lot of expectations. Okay, he would have made a thorough study for what his case is about. Google would help him, and then he would come to you with all his clear cut problems with these options also. So definitely you have to be there very carefully, maneuver and motivate this patient or take up the case, because a lot of issues would also be propping up later if in case you're not careful. So once you have identified this, that this case is a retreatment case, then probably it's very easy to assess them. It's under this Jackson Strat. All of us know this. Functional deficiency, aesthetic disharmony, and structural imbalance. So functionally, you have TMJ deficiency, pain in airway, difficulty in speech, gummy smile, irregular smile, facial asymmetry, occlusal cant, and muscular pain. These are the very common thing which a retreatment case or the case would come back to you. Okay. So what are the diagnostic criteria we require to be undertaken? Now, it is no different from a regular case. However, see, when we start having our case histories, since I say it's more of an adult case, we need to have a thorough investigation or have a complete general health history, because currently a lot of health systemic issues are also to be seen in younger generations. So when you are taking up a case for the second time, you need to record them very carefully. Diabetes, osteoporosis, vitamin D deficiencies, because the kind of protocol you would undertake would warrant all this. So case history and study models, and now of course we adapt or adopt intraoral scans. Now, once we are okay with this, the next very important diagnostic record for me is essentially is an OPG. Now, reason is, once we take an OPG, we evaluate the third molar, because that would help you to give or help you to make a decision a little bit that whether the case can be, you have a little bit of leeway to maneuver the case. Third molar evaluation, bone loss, root inclination. These are definitely to be carefully monitored because it's a retreatment case. IOPAS, photographs, lateral septum. These are anyway go as a standard protocol. So what are the general considerations for treating a retreatment case? Now, space management. You are left with nothing actually. When a case comes to you second or third time, extraction would have been done. So even the proximal slicing would have been done. So you hardly have anything to play with. However, expansion, the list would, I would list it like this: expansion, distalization, IPR, extraction of third molars, extraction of lower incisors, and lastly, if you are lucky, extraction of first and second premolars, if you are left with these teeth. Okay. What else would you try? Of course, now jaw rotation, the factor of jaw rotation is in a big way. Okay, advent of TADs definitely have put this, made it life easier for us. So jaw rotations, and the third one is aesthetic fulfillment. Now, patients are aware of these things. Okay, so you have to be relevant in practice. You have to be discussing these things with the patient as well, whether, if the patient really requires. Okay, so that will give the patient that, a positive feeling that you are an updated orthodontist. Okay, so we do a little bit of veneers, bleaching, etc., part of smile designing, and of course augmentation, reduction, fillers we use, graft, etc. These are the things we have adopted in my practice. Now, retention protocol, it's a similar situation, whether it's a first timer or a second or third time. However, when the patient is coming for the second time, the patient would have had a history of retention or retainers. So they would be very choosy. However, you have to make very clear that if in case your retainers fall in line with the same as the earlier ones, you have to tell the patient that this is thing you are likely to have. Okay, so clarity in what you tell the patient is, it's a very important part of your treatment planning. Now, choice of appliances. We have done with this thing, you go to the choice of appliances. Now, customized prescriptions. Now, with so many things available now, you are given with an option of having a practice in a more systematic way. Okay, you have so many things in hand. You have customized prescription. Definitely, the mechanics have to be a low friction mechanics because it's a second time case. I always advocate a low friction mechanics. Application of TADs, light forces, use of elastics, use of aligners, prosthetic rehabilitation, soft tissue aesthetics, and retention appliances. Okay, so let's start with the first case. Now, all these cases are for my clinic, and it's treated by myself. So this is a case which reported to me. She's a 29-year-old. In fact, she's a doctor. She's a general med. She was transferred from a different establishment. Okay, all fours were extracted. Her complaint was a gummy smile, and this was a picture. This was a state I got the case. She traveled from Kerala, and with some brackets in the mouth. So we took some pictures. The next thing what I do is I remove those brackets. I immediately do an OPG. Okay. Now, if you see here, root inclinations are not favorable. We have a missing 1 4 1 4, all fours missing. 1 8 and 4 8 is missing. Root canal treated, crown. The most important thing is the condyles. You see those condyles there. Okay. So condyles are not in a favorable position. So throughout this retreatment cases discussion, my emphasis is on the condylar position. So facial form, oval facial form, non-consonant smile arc, and convex profile, typical class 2 case. So we remove all the appliances, right side end-on, molar and canine. We have a deep bite, British incisor class 2 relation. So all the 4s are extracted. Lateral ceph will show you an ANB of 5 degree, lower anterior facial height of 65, and of course, see, most of these cases are retracted, over-retracted or retracted. So incisor inclination definitely either will be retruded or very rarely you will get a proclined teeth. Upper and lower reasonably, upper is retroclined in fact, lower is fairly upright. So skeletal place class 2, underlying British incisor class 2 malocclusion with retroclined upper anteriors, deep bite and a horizontal growth pattern. So what are the objectives? Now, objectives for me will always be when an extracted case is there, a class 1 canine is mandatory. Okay, you have to get your class 1 canines, and if there is a possibility, achieve a class 1 molar. Okay, correction of gummy smile in this case, definitely a soft tissue profile which has to be corrected by propelling the lower jaw forward. My appliance is 018 slot, MBT. I use biturbos, elastics, implants, upper and lower fixed retainers, and soft tissue lasers. So plan is to extract 28 and 38, leveling and aligning, intrusion of anterior teeth, rotation of the jaw, long-term retention as usually the case in, and lastly gingivectomy in the upper anteriors. So wire sequencing for me in a 018 slot is usually I start with the 012, progress 014, 0 night eye and then SS, 016 night eye and then the working wires. Okay, so those what is marked in red is not used in this case. Okay, so always, I mean, whatever your cases are going to see, what is in the red were not used, because certain times we can cut short the treatment time. It is not mandatory you go through the wire sequencing as the protocol says. Okay, so these small adjustments you can get through. So what are the mechanics? It's no different from what is mentioned in the textbook. Definitely we introduce TADs. This Sung Jung by Dento's textbook will give this diagram for you. Rotations, clockwise or anti-clockwise rotation of the upper jaw, intrusion. So I follow what is in the textbooks, so nothing different. Again, if you see this, I use a lot of looped arch wires. Again, the mechanics are the same. Whatever is mentioned in the textbook is what is applied in these cases. So we are in the era where we call a lot of programmed brackets. Okay, so we have an advantage these days, unlike the earlier times. So we can standardize your prescriptions or make the adjustments. The canine bracket was changed here, -7 was used, and of course the tip and torque of the lower was maintained as what it is. Okay, so other options for you is Roth, or currently you have the self-ligating different systems. So I skip the wire sequence and directly come into a situation where I show you there is a TAD placed. We have an anterior 2 TADs. The curve P was exaggerated. Working wire is 1722. I used bite turbos. We use elastics. The first priority is to open the bite and put the canine into class 1. So always remember, your case or your retreatment case is always successful when you have your roots of your teeth aligned and the condyle which falls in place. So mid-treatment OPG, if you see the condyles, the right and the left slowly falling in place. The roots are getting slowly upright. In fact, it's almost upright. Over a period of between these appointments, a lot of repositioning of bracket would have been done. Okay, I have not mentioned how many, but we do a lot of repositioning of these brackets. Okay, so once we are satisfied with the OPG, I hardly have a lot of anything to do with the lateral ceph. It's the OPG which would really help me to finish my cases. So then I immediately debond the lower. Once I feel the lower is upright, because we cannot have a progressive, a long treatment time with this. These are retreatment cases. Your roots will not allow you to do that. So I do use a little bit of or more of frictionless mechanics to upright my upper canines. Okay, the cornerstone of any retentive case, retreatment case, is a upright canine or soundly seated in their cortical bone. Okay, so what I do is I debond the lower, fix the lower retainer, and hold the upper arch with a little bit of loops for some time. So this will allow the condyles. Yesterday, Dr. Raj Ganesh was discussing mutually protected occlusion. This is exactly what I try. Okay, because I've seen over a period of time this kind of mechanics, what I've tried, really helps. So I keep this upper arch for some time, and then finally the progress records will show us what actually we are attempting and how far have we succeeded. Gingivectomy in the upper arch to help her soft tissue fulfillment, and this is how the case finally finishes. Okay, a fairly good class 1 canine. Molars, I've tried to, that's the crown underneath. We have changed those crowns. Bite opening to a maximum extent was tried. Stable occlusion, upper and lower fixed retainers. Her smile is improved. Treatment duration was around 28 months. This itself is quite a long time. Okay. However, certain times we need to be more careful because there is no third time for these cases. Okay, because that cycle is going to come back to your clinic again. You saw the cycle, no? First it goes. If you don't do it correctly, the cycle is going to come back to your clinic. So OPG again, end of the treatment, is very important. You see the condyles now, they're fairly, absolutely stable and healthy. Okay, here is way where this is the time where you realize that case is fairly well treated. So all the boxes ticked, lateral ceph taken, A and B improved, lower antefacial height also considerably improved. Patients pre and post, intraoral, good class 1 molar. At least this canine, this was the best I could do. I still agree it's a little bit of endonish, but case is stable as of now. It's been 4 or 5 years now. So superimposition, and finally I have tried to fix or tick all the boxes which I have started with. Okay, class 1 canine, condyles in position, aesthetically fulfilled case. So this is how we plan for a retreatment case. Frictionless mechanics, it's mandatory that you know about it. As you know, students, don't overlook these things. This is what I always tell even my postgraduate students. So case 2, again, chief complaints of difficulty in chewing and improper bite. She has undergone extraction of first premolars, and I had a very difficult time to make her smile for a photograph. She hardly would smile. For probably they have gone. In fact, she has been treated twice. In between, the first part was some pedodontist gave her some trainers. The second part was extensive orthodontics. So I take an OPG. So first thing is, even before the records are taken, see, there are always cases. You have not always can you take up a case for the second time or third time. You have limitations. So most of the cases I decide once I take the OPG. If in case the bone loss is very severe and periodontally weak teeth, it's a risk you put your hands in this kind of. So OPG you take. So you see what you see here is, anyway, missing 1 8. All the other 8s are present. You see the cant here. This cant posteriorly, anterior cant, lower and upper. Condyles, you see the right and left, there is definitely a gross problem there, correct? So overfacial form, convex profile, this is our intraoral photograph, okay? Now, it's a disaster, right? Right side, you have a class 1 molar relation and a class 1 canine relation. Increased overjet, overbite. You see, in fact, this case I could feel the roots. It was so much close to the cortical plates. Okay. Left side class 2 molar end-on canine relation, and you see the occlusal cant. Okay. Now, these are the cases which invariably will walk into your practice expecting you to do a good job, because you have advertised, right? You have put yourself as one of the best, or those pictures pre and post. So you have to now do justice to when these kind of cases walk in. So you take a lateral ceph just to see where is your where is that you can maneuver few things. Now ANB of 3 degrees, lower anterior facial height of 64. Rest again, as I told you, upper incisors retracted anyway. You are hardly left with anything. 25. Lower incisors almost close to upright. So there is nothing, a lateral ceph, you will not be able to appreciate a lot of changes on the anterior posterior side in this view. So diagnosis, class 2 skeletal base, angle subdivision, proclined upper and lower anteriors a little bit, increased overjet, overbite, and an average growth pattern. So objectives, first thing is the cant correction, midline correction, overjet, overbite, class 1 canine, molar, and definitely a suitable condyle is what you want to achieve, and planning a soft tissue profile which is pleasing. Again, the same appliances, nothing different. I added an open coil spring here. This is what was an addition. Otherwise, more or less same, frictionless mechanics, emphasis on torque. Again, if you see here, the bracket, the canine bracket is what I had to manipulate a little bit. I use +7 here because for me, the canine is the one which, as I told you, gives a lot of stability. So again, you have other options for yourself. You have these inventories, I'm sure, in your clinic. Wire sequencing, in this case, I had to eliminate O12 and O16. Otherwise, more or less, the working wire is 1622 Hant and SS, 1725, 1725 SS, and finishing definitely with a lot of O14 SS. So I have skipped a couple of wire sequencing. I come to a point where leveling aligning, I am in the last working wire. I call this working wire because I do a lot of things here. Now in 1622 SS, I put an open coil. Okay, the midline gets corrected. Same time, you see my canine, it's slowly getting upright, right and left. So that is my first priority. Okay, upper canine, once they are upright, slowly you see the cant getting corrected. Of course, we had a lot of repositioning. It's not easy. We need a lot of repo in these cases. We give cross elastics, need to be advocated. You need to know the biomechanics of what happens with giving unilateral or bilateral elastics, definitely the whole arch. So Burstone book is again a cookbook for us. So I almost correct my midline and immediately take an OPG. Okay. Now you see the condyles again, the upper and the lower incisors. Okay, they're getting upright. In fact, the canine is nicely upright here, somewhere around mid treatment. Condyles are nicely shaping up. You see the condyle. I am almost happy with my leveling and aligning. Okay. Now comes a little bit of bite opening. We need to be very slow with the bite opening because these roots are already once manipulated. So the TADs are engaged. We engage a posterior, I engage a posterior TADs usually initially. Curve spin upper wire, manipulate the lever arm. The force vector is changed. Class 2 elastics are given there. The photograph anyway not taken, but slowly the case is getting in our control now. Now what I do here is I need to open the bite a little bit further. Okay, so I engage this loop. If there is a step which gives me a bite opening advantage anteriorly. If you see unilaterally, there is a little bit of modification. One arm is little longer and one arm is little shorter. It helps me to correct the cants. Okay. So canine slowly has fallen in place. Class 1 canine, right and left. Here is where I feel I am on course now. Okay. Midline is corrected. I engage a midline implant. It helps me to open the bite further faster. And slowly, class 1 canine, see, it's very important you engage all the teeth. All my cases, I make sure, I tell my PGs also, you engage the 7th. Especially, definitely in a retreatment case. Because unless all your teeth are aligned and upright, you will not finish the case. So again, we take a OPG to check how the case is progressing, because the condyle and the root inclinations. So now you see the roots. Upper and lower incisors are upright. You see the condyles right and left. They are almost, when you come to the last OPG after lower debonding, your condyles are happily seated. Okay, so you know now you are almost close to where you wanted them to go. Okay, so now she manages to smile. Okay, she's happy. I could show her the changes. See, these cases have no much aesthetic changes because the teeth are retracted. They have gone earlier somewhere. Somebody has taken a nice rasgulla and left all the leftovers for you. So they don't understand aesthetic corrections. Okay, but definitely when you show them where you have done the correction, they appreciate it. So this is how the case progressed. I could fulfill cant. I could fulfill the root inclination. Canines well seated, and finally I conclude the case. Okay. So pre-treatment, post-treatment, entire time duration was around 18 months. So this is how the case progressed. OPG is a testimonial for that. How the condyle and the roots, inclinations are all aligned. Once you fulfill them, you know you are a winner. So lateral ceph, as I told you, not much changes except for a lower anterior facialite. Marginally, we could manipulate. But all in all, a good case, stable result is what I would, because once you have done your records, you have done your best, you have done all the protocols ethically. So legally, you are on a safer side, right? You can always document, show the document that I have done all this. Beyond this, if there are changes beyond your reach, you can still have a reason to defend. So this is how the case finishes. So I've skipped quickly this case. This is a case, again, a retreatment. We had a tooth material issue here, upper arch. So you have to be very clear with the case, the patient. You have to recognize the problem, because these cases will not allow you to manipulate something which has not been informed. Okay, you tell the patient day one. So she had a premolar, way too small. So tooth material deficiency or discrepancy was there in upper arch. So we had to tell her, see, this is the case. You may require a little bit of tooth material increasing protocol, maybe a crown or something later. So we take a lateral ceph, not much of this thing anyway, it's 4-4 extracted case. Diagnosis class 2, class 1 with the angles, class 1 is spacing in the upper arch. Again, same 018, 022. We extracted 4-8, frictionless mechanics. Again, the canines, I kept it as a zero talk. Okay. The wire sequencing is this, not much of a change otherwise. So I skipped a few, I straight away come to, again, my canine going to be upright. Okay. So here we use, if you see her lower left and upper left second molar, morphologically it is, there is a cusp in the lingual. So it's very difficult to get your occlusion right. So you need to inform the patient about all this. So my first priority to keep the canines upright, I use these frictionless mechanics to keep the forces lighter and open the bite. Case progresses. See here, I do some changes. Moment the canine falls in, I debond the bracket. Okay, because I want my premolars to extrude. Once the canine I feel is stable, I immediately debond it and allow the premolar to extrude so that I get a good tooth fit. Okay. So finally, we conclude the case. Class 1 canine, class 1 molar. My objective of even the midline is corrected. Whatever she came for, I fairly could do it. Okay. So retention, again long-term retention, all retreatment cases I play safe. I give a lot of long-term retention. Profile improved. She had a better soft tissue adaptation. OPG again, you see, the condyles is where I emphasize on. We could fairly get a good stable result. Again, the lateral step was in not much changes anterior posterior, however, a little bit of A and B, anyway, maintained the same. Lower anterior facial height, again around 66. All other values were in our favor. So this is how the case finished, pre and post, upper and lower. So luckily, we could handle the tooth material problem. Okay. I also had given a little bit of tooth positioner for her, just to make sure the corrections are maintained. Superimposition, pre and post. Again, bite opening was the first criteria here with the class 1 canine. Root paralleling, and aesthetically you could finish the case. So this case is the last case for you. I have just on time. Yeah, last case. Now this requires a little bit of, it is a 52-year-old case, a lawyer by profession. He had all the entire list, whatever you've seen him. He had sensitivity, improper smile, bruxism, etc. History of early extraction of 2-3. OPG, again, luckily the condyle was stable. It was in our favor. Otherwise, there was a cant. This is how the upper and lower arches were. Crossbite right side, gross midline extracted, midline shift to the right, left, sorry, 2-3 extracted. Some orthodontist by mistake extracted is 2-3. Okay. So this was probably when he was 14. After that, he didn't go back to, undertake the case. Anyway, so this is what we did, and A and B of 2.7, class 3 skeletal base, class 3 molars, British incisor class 3, with missing 2-3, and upper midline shift and an average growth pattern. So here, he was very clear. He wanted a clear aligner treatment. Okay. So we had to plan the case in phase 1 and phase 2. So the plan was to undertake the case with Invisalign, and he wanted a similar looking teeth on his 2-3. So the day 1 I had to plan his Invisalign with an implant. Okay. So you need to be aware and clear with how you're going to keep the tooth as when you plan for the implant on the adjacent. So this planning is very critical for you. The premolar and the lateral roots have to be such a way they should take up the implant. The size of the implant has to be determined day 1. So planning of this is very important because you have promised a case. So we take the case with clear aligners, Invisalign, and I many of my cases, a few cases I treat, I give aesthetic pontics. So you have this option of building up a canine. As the space opens up, the canine is included in the aligner. So phase 1, I end up correcting my crossbite. That was the first protocol. Midlines are almost coming close. I regain space of 8 mm. The plan was to put a 4.5 mm implant, canine, right? So I progress through this. End of phase 1, we take the OPG. We see how the roots are getting parallel there. So I am in discussion with my prosthodontist whether sufficient space is opened up for the implants. So next comes the pre-prosthetic planning. Yeah. Yeah, done. Yeah. So this is the pre and post. Okay. And this is how the case concluded. Finally, the implants were in place. Pre and post. So this case was it's in the Align Global Gallery, and this is a summary of the entire discussion today. Give a lot of emphasis on the canine in retreatment cases. Curvus P, all others are standards anyway, and always keep a legal consent on your side. So conclude with emerging technology, it's therefore important to keep a strong biomechanical foundation in synergy with digital applications to expedite effective treatment outcome, keeping in mind the patient aesthetic as a priority. Okay, thank you very much. And before we finish, these are, I think the message should be, these two stalwarts, one is a cricketer, one is a Bollywood star. They are, one came by traditional methods, and the newer ones on the left side, the SKY and the Sharafs, they have come with a lot of flair and this thing. What we need to do is, I'm a huge RCB fan, so we need to club the both technique as well as the technical as the newer flashy approach. And of course, we are all fighters, orthodontists, so we also need to adapt the best of the two. The message today is, you all need to be versatile. So let's be versatile in our approach, in our practice to adopt new and old. Let's not forget the foundations which are required to be always, taken up. Biomechanics is what everybody discussed, and the newer things as well. Okay, thank you very much. Sorry, I've taken a little bit more time.
Thank you, Dr. Rai. It was a very nice presentation. Please, if you could collect your certificate.
I request the chairperson to felicitate Dr. Rohan Rai, sir.
Thank you.
I request Dr. Ankit Sikri, sir, to felicitate our chairperson for the session. Thank you, sir. Our next speaker for the day is Dr. Ankit Sikri, sir. He is professor and PG guide in Guru Nanak Dev Dental College, Sunam, Punjab. He has been the executive committee member of Indian Orthodontic Society and the founder, convener, and honorary secretary of Orthodontic Study Group of Panchkula, Haryana. He is the section editor of Indian Orthodontic Times newsletter since the year 2021. He has also been an ex-chairperson, International and National Student Exchange Program Committee, and the Joint Secretary and Chief Coordinator of the World Dentist Association. He is the Director of Confidential Multi-Speciality Dental and Orthodontic Clinic, Mohali, Punjab. He is currently doing his PhD from Baba Farid University of Health Science, Punjab. He did his international fellowship in clinical orthodontics from Chiang Mai University, Chiang Mai, Thailand, and he is also a fellow of World Federation of Orthodontists. To chair this session, we have Dr. Jaskirit Singh sir and Dr. Deena Dayalan sir. Dr. Jaskirit Singh sir is also the executive committee member of IOS and convener of OSGOP. Dr. Singh maintains his exclusive orthodontic practice in Karnal, Haryana. He has to his name numerous national and international publications. Sir maintains his exclusive orthodontic private practice in Karnal, Haryana. Next, we have Dr. Deena Dayalan sir. He is also a postgraduate guide and has guided so many students and also has got more than 40 Scopus Index journals, along with 6 books, published and attended more than 30 national and international conferences, and also elected as IOS AC member. We welcome you, sir. The stage is yours.
Am I audible? What a lovely evening yesterday we all had, and I know seeing the attendance, how beautifully that was conducted by the 27th iOS PG convention people. Hats off to you, all of you, for that. Now, if I split a word retain, it becomes retenir, to hold on to the occlusion which we have achieved after treating a certain kind of a malocclusion. To hold back or to hold secure is what retention is all about. Well, this is also one kind of a retention, but definitely we are not going to discuss in this academic podium of ours. Is it retention or is it retention? Definitely I would term, after 12 years of my experience as a clinician and academician, I would be sure that it is B, retention. Because there is a chance of relapse, because you have to do retreatment, because you have to ask the charges again to the patient, and then the patient will happily kick you for this and you will definitely regret. So this is what retention is all about. And Dr. Thomas Graeber once said, there is only one way to completely avoid relapse. At the end of the treatment, remove the braces, polish the teeth, make study models, take the photographs, take the patient out of the back door of the office and shoot him. This is what the best possibility he gave. That is on a lighter side. So first of all, good afternoon, respected chairpersons. It is the toughest lecture for me because the person sitting on the left is my BDS batchmate, Dr. Deena Dayalan, and we used to tease each other a lot in first year BDS. So I don't know how this lecture of mine will go, as he is the senior person sitting in front of me. And one is my very dear brother back from Karnal, Dr. Jaskirat Singh. So anyhow, good afternoon, both of you sirs. Good afternoon, audience, and good afternoon, everybody. First, I always start all my lectures with a token of gratitude, and I first and foremost thank Dr. Shailesh Shenava sir, organizing chairman, Prakash Mudaliar sir, organizing secretary, Robin, my very dear friend, scientific convener, Dr. Prachi, Dr. Ishwarya, Dr. Trupti. I tend to forget names. I hope Dr. Chinmay. So pardon me if I miss somebody, but hats off to all of you as a team. You have pulled off a great show. Thank you so much. I stand tall because of you giving me an opportunity. So I had a great Siddhi Vinayak Darshan yesterday. So I'm happy to be here in the Flamingo City and standing here at Terna Dental College. I'll not waste much more time in introducing myself. I belong to city of Mohali, and you know there is a cricket stadium there. This is the college where I am professor and PG guide right now. And this is a small clinic which I have just started on 13th February last year. So 2 days after this, 2 days after this lecture is the first anniversary of my clinic. So retention, a road less traveled in orthodontics. You might be wondering why am I running so fast. Actually, I have a furious ex-boss sitting in front of me, Dr. Sarabjit Singh. He was my HOD. And 1 year I was reader under him, and I know what all he is. He's a very cool and calm person, but whenever he takes tension, he becomes furious for sure. Now there is a gun on my head that you have to finish this lecture in 25 minutes. So my all things have been, is going haywire, and I have to talk 15 minutes less. And we have to catch a flight. Both of us are in the same flight. Wonder the flight will land in Chandigarh or not. Anyhow, so I'll be covering basically theoretical aspects. You can afford to yawn, you can afford to sleep. I'll not mind because the lunchtime is already over. And beautifully, the clinical things already have been discussed. So I'll just tune it a little bit into a literature part. So I hope you bear with me. The holding of teeth in ideal aesthetic and functional position, as termed by Graeber, is what all retention is. And Moyers actually beautifully defined this particular thing, which says maintaining newly moved teeth in position long enough to aid in stabilizing their correction. But the soul and the spine of retention does belong to this great personality, Sir Raleigh Williams. Without his mention, one cannot conclude his or her lecture on retention, because he beautifully gave us 6 treatment keys, a beautiful article which was given in JCO 1984. I'll just quickly go through because you all have done this in seminars and everything, so there is nothing new for me to ponder over it. So just to quickly rehearse it, key 1, he says incisal edge of lower incisor should be placed on anterior-posterior line or 1 mm in front of it. Second key, he says the lower incisors apices should be spread distal to the crowns. Third key, he says the apex of the lower cuspid apices should be positioned distal to the crowns. Fourth key, he says all 4 lower incisors apices must be in the same labiolingual plane. Sorry for this photograph. Fifth key, the lower cuspid root apex must be positioned slightly buccal to the crown apex. And the last key is you have to slenderize or you have to proximally slice whenever you want a lower occlusion to be stabilized. But there are some conventional retention modalities which we all use. Okay. First is Kiesling positioners. Okay. These are the tooth positioners which we use or we don't use. That depends on person to person. And a simple Hawley's retainer. Definitely when you finish your treatment in colleges, whenever you do, this is the best and the cheapest modality available. Essex retainers are the upcoming things, BioCrail retainers. The patients who don't have an acceptability to wires further after wires or further after the treatment things, they always go with Essex for sure. And this is one very cumbersome for me. I hate this somehow, but don't know it is the need of the hour, the LBRs, the lingual bonded retainers. And I hope you all know this is a floss technique where we, so I'll not go into the details because I still see my boss getting red. So I'll just, these are the interlegs, the fiber reinforced composites, okay. And these are the TADs which are actually retaining skeletal class 3, a growing patient. You can take TADs also as a mode of retention. So after these conventional modalities, we'll be talking at length about updates of what all orthodontic retainers are coming in. So basically these are the custom made devices. They are designed to maintain the corrected position of teeth following treatment and prescribed to prevent the teeth from shifting back to their original positions. And they also play a crucial role in ensuring the long-term stability of orthodontic results. There are 2 types. One is removable, one is fixed. Removable, obviously, they can be taken out according to the patient's will. And fixed, they are permanently attached to the lower teeth and they cannot be taken out by the patient. So the compliance factor goes with the fixed type of modalities. Now, retainers are mandatory in such factors. If you do a braces treatment, if you have post-surgical cases, you are done with the surgical case, you want to retain, you have done with the bite correction or the cross bites if you're doing, or the complex orthodontic cases, there the retainers, they become mandatory for sure. And retainers are not mandatory when you are done with some minor or local corrections. Age related factors are there if you are just performing an interceptive orthodontics and when the growth is ceased. So the chance of relapse becomes many folds down, so that the retainers aren't mandatory in that. But there are important considerations also. Individual variation is there, okay? It depends on the age, the genetics, and overall oral health. And according to the recommendation, because orthodontist A might have different opinion, orthodontist B might have different opinion, so that depends on opinion to opinion, okay? So the recent advancements in retainers are the clear retainers and aligners which are coming up. These are basically the devices which are made from transparent thermoplastic materials such as PETG, polyurethane or polyethylene terephthalate glycol material. They are thin, they are lightweight and virtually invisible. Okay. Now this is how they look, all right. And the advantages basically are aesthetically, because they have a discrete appearance, because they are barely noticeable, the individuals, they can maintain a professional or natural appearance during treatment. All those people who are working in MNCs or those who don't want to show a wire thing, they are going with these kind of retainers. Obviously, they are comfortable to wear and they fit to the teeth snugly, okay? And removability also is not an issue. The only thing you have to remember is, because they are too sophisticated, you cannot afford to have those spices and masalas which we have in India, especially in North India. Punjabis, we love masalas like anything. So you just cannot afford to have hot drinks or extreme temperature drinks all and along with all those spices along with this, because that tend to degrade this particular appliance. Then it is convenient on special occasions or activities where traditional braces might be less desirable. It is very convenient, okay? And it has a predictable treatment outline, okay? Because there is a digital planning process involved which allows orthodontists to visualize the entire treatment progression, and anticipation of the final results can be done before treatment itself. So this is one classic article on clear retainer. I've put some articles in between so that it becomes, if I'm not putting cases, I'm putting articles so that there is some take home message for you and take home content for you. So this is one of them. The considerations of such aligners or the clear retainers are, first is compliance, as I'm talking again and again. The thing to consider is that the removability of an appliance actually increases the dependency on a patient thing. So the patient compliance, it increases manifold. So you have to rely heavily on a patient compliance. Patient must wear the aligners for the recommended duration each day. And there is some complexity of treatment which was beautifully told by Arun Naik sir, so I'll not repeat all that. And obviously, the more sophisticated thing you wear, the cost increases many folds. So this is again one beautiful article on orthodontic retainers, a must read, a critical review where he has discussed a lot of things by Lyros et al. So this becomes another article. And a new type of clear orthodontic retainer incorporating multi-layer hybrid materials, KGO. This is one article again. Next one is the retainers which we get from 3D printing technologies. It is actually known as additive manufacturing, which involves building a three-dimensional object layer by layer based on a digital model. Basically, it depends on the scans and the digital impressions, and rest all what all is there. That key advantage is the ability to create highly customized retainers tailored to the unique anatomy of each patient. So this is how 3D printer works. I'll not go into the detail as the time is less for me. The only thing is I've summarized it like this. The workflow and materials look somewhere like this. There is a digital workflow where acquisition of digital data will be done by the intraoral scans, and there are specialized softwares to design the retainer. So the material options are mostly used are biocompatible resins, which are flexible, durable and transparent. The salient features, you can rapidly produce it, okay, and it has an iterative design, all right. Iterative means where you can do adjustment by adjustment by adjustment, okay, and the software, and then the printer will give you the productivity will be through printer only, and it will keep you giving the retainers like this. So this is one of the article regarding 3D printers in orthodontics. It's a very conventional article given in Turkish Journal of Orthodontics. Then we come to smart retainers. Yesterday, Dr. Puneet Batra in his lecture told in digital this thing that there are some Bluetooth and smart retainers coming up, obviously with some monitoring apps. These devices, they go beyond the traditional role of retainers by incorporating sensors and connectivity to enhance treatment monitoring and patient engagement. The salient features are it has a sensor which senses whenever you wear or whenever you remove a retainer, and there is a Bluetooth connectivity available, and the design, in spite of the sensor in the acrylic part, the design is still a comfortable design to be worn. And this is how it looks, okay, and this is where we keep the Bluetooth, and how these smart retainers work. They collect the data, the sensors will collect the data, especially the parameters, pressure exerted to the teeth, temperature variations, and till what time or for what span of time the patient has worn this retainer. Wireless transmission, where the collected data is wirelessly transmitted to a paired monitoring app over the patient's smartphone or a cloud-based platform. That same thing is on the doctor's, the app, same thing is on the doctor's smartphone too. So real time monitoring is but through that app access only, so the doctor can track the treatment progress and the compliance of the patient. So there is no room for cheating for the patient to be done to the orthodontist. The benefits, the continuous monitoring is there, real-time tracking is there, compliance is strong, immediate feedback comes, and the patient to the doctor engagement is superb. The future implications of such retainers are, there are personal treatment plans. Data collected could potentially contribute to the development of personalized orthodontic treatment plans. Teleorthodontics, as sir told yesterday only, we are good in telemedicine, but teledentistry and these things are still coming up. So teleorthodontics, integration of smart retainers with monitoring apps, they allow for remote consultations and monitoring, like what we did in COVID times. So it helps a lot. So this is one of the very interesting articles, applicative characteristics of new microelectronic sensors, smart retainer and theramon for measuring wear time. You just read it, and all the things which I have told is summarized in this. All right. Next, we come to biodegradable retainers. I'm sorry, I have to rush fast, but this is how I have to finish it. Biodegradable retainers, a forward-thinking approach in orthodontics, basically because we are just doing eco-green, eco things. So then we have to respect the environmental considerations in our branch also. It is basically to address the environmental concerns associated with traditional plastic retainers. Okay, the advantages, just the major advantage is to reduce the plastic use, that's it. So it has to be have a sustainable material, it should be environmental friendly, and there should be a reduced ecological footprint. But the challenge is, the more you take out plastic from this, the the stability decreases. So the material stability is definitely an issue, durability becomes another issue, and the regulatory body approval, you have to take lots and lots of approvals. Until and unless you don't get that you can be penalized for that also, so you have to take care. Then comes some vibratory devices, which emerged as a novel approach in orthodontics, aiming to enhance the retention phase and reduce the likelihood of tooth relapse. Basically, the purpose is it accelerates the retention. Basically, it is designed to accelerate this phase, and this phase is crucial to stabilize the corrected tooth positions. Basically, it is giving low frequency vibrations. These devices, they deliver low frequency vibrations to the teeth. They are gentle and have positive effect on bone remodeling. The more positive effects on bone remodeling, the more tooth stability will be there and retention will follow. So this is kind of vibratory devices which are available. The potential benefits, they have improved bone remodeling, reduced relapse, enhanced retention. The challenges, still we have to find out the evidence-based data in this. The studies are still going on. So it is very easy to say you use vibratory device, but you have to have a good evidence-based data with that. And then device design and comfort. It should be user-friendly, comfortable device, and the user-friendliness always adds up to the usage of the device. The last phase, and another 5 minutes I am left, just 6 or 7 articles which I have just kept it for you. First, this is what you have to read before you start reading retention. It has this article covers a lot about retention, a lot about relapse. That's what Raleigh Williams I started my presentation with. Then this is towards, Melrose has written a good article toward a perspective on orthodontic retention. Here he has done a broad critical analysis on how he has gone through his post-treatment stabilities and everything. So you can read this article also. One article which I have come through is outcome and long-term stability of an early orthodontic treatment strategy in public health care, and followed by all the cases, how the retention has gone. Then orthodontic retention protocols and evidence-based overview is another article which you can definitely have a read. So these are good 6 to 7 articles where you can read, and you I am so sure once you read them, you don't need to touch any textbook, or you don't need to go through any seminar as such. And this is one upcoming article, analysis of the effectiveness of FRCs, the lingual retainers, systematic review and meta-analysis, because FRCs, interlinks, they are upcoming. People don't even bear LBRs now, lingual bonded retainers. They don't want the wire show to be there. They don't want to see steel after the treatment is done. So it has to be, though interlink, I don't feel, I had interlink in my mouth for say 5 to 6 years post my orthodontic treatment. But the surface area is so broad that it attracts food particles, and it is a good friend of having calculus or things like that. So personally, I don't recommend interlinks as such, but the literature is such that you have to respect that also. Then mechanical and behavioral approaches to orthodontic retention. This is evidence review by Padraig S. Fleming. It's also a good article. And orthodontic retention protocols, a survey among Indian practitioners, a very basic article, and it is also written by my very good friend Divya Ruprai. So I hope I have done justice to what my boss has said, but I'm sorry if I ran like Vande Bharat Express today, but that was need of the hour because valedictory has to go through and we are running short of time. So I had to cut short my presentation. So time to say goodbye until we meet again. Okay. And I am a call away or a message away for anything and everything. Thank you so much listening to me. And thank you so much 27th iOS PG Convention for giving us a wonderful, wonderful show. Once again, hats off to you all. God bless you. Thank you so much.
Thank you, Ankit. Thank you for the short and this crisp presentation. I know you will say Flanley in Chandigarh. Sarabjit, sir? Yes? Thank you, sir.
I request the chairperson to felicitate Dr. Ankit Sikri, sir.
Sorry.
I request Dr. Sarabjit Singh sir to please felicitate Dr. Ankit Sikri sir, the chairperson for the session. Sorry. So, yeah, thank you. Moving on, we have our next speaker, Dr. Sarabjit Singh. He is an ex-Vice President, Indian Orthodontic Society, ex-Dean Bojia Dental College, Badi, ex-Professor and Head, Orthodontics Department. He has been esteemed with the Dr. P.P. Jacobs Award and National Dental Excellence Award for the year 2014, 15, 16 and 18. Sir is a beacon in Indian orthodontics over 3 decades. He has a brilliant academic record from KGMC Lucknow and PGI Chandigarh. He has a vast multidimensional experience and is a profile speaker at both national and international level. He has been executive committee member of IOS multiple times and is presently the convener of Chandigarh Orthodontic Society Circle. He served as the chief Indian doctor of the China-India Joint Medical Mission 3 in China in 2012. He has directed various courses and lectures all over the nation and has many national and international publications to his credit. To chair this session, we have Dr. Vijayadit sir. He is an executive committee member, Indian Orthodontic Society, Professor Head Priyadarshini Dental College, PhD at SRM Dental College with 16 years of teaching experience. We also have Dr. Neil Kedia sir, who is Professor, Department of Orthodontics, Buddha Institute of Dental Sciences. He is also member of American Association of Orthodontics, World Federation of Orthodontics, and Indian Orthodontic Society. We welcome you, sir.
Thank you. Am I audible? Yeah. So good afternoon, everybody. First of all, I want to thank the organizers for giving me this opportunity in such a esteemed platform. As very rightly said by the speaker just before you, Dr. Ankit Sikri, that we have a flight to catch, so I'll be rushing out things. My presentation was for 40 minutes. I'll try to, in this presentation today, I'm going to talk about very basics, just for you people, that is bonding in orthodontics. The various mantras, tips and everything, which I'll try to guide you how and what all you can use. One of the last slides which we saw, the braces are going to be there. You saw that? So in my experience of 30 years of practice now, I want to make all these young people very sure braces are going to be there. 100%. Whatever might be the situation, whatever might be the disappointing things you're going to hear, but the braces are going to be there. So you have to, and being an orthodontist, you can excel on things to make yourself survive and do the best. As simple as that. Okay. So the previous presentations were on retreatments, then retention. But our practice as an orthodontist, as you get senior, we mostly get retreatments. So that's our ray of hope. All the cases which get bad by the general dentists who are doing things, they come back to us. So if you want to be a good orthodontist, you need to know, you need to be very good in your bonding skills. That is what I'm going to share with you today. So this is where I practice. I was an academics dean, professor, head of department, but nowadays I'm in a full-time practice at this clinic. So one simple saying, your bonding with the patient will depend upon the bonding of your bracket with the teeth. Very simple line, conveys everything. You have done the full strap up, parents were very excited, next day they come back with half of the brackets broken, you will be the most bad position to be in. So you need to improve, you need to do your best in bonding. So this is the smile you can get if you do a nice case. One more photograph I want to share. This I got in my practice, must be 100% done by a general dentist, cannot be done by an orthodontist. He had all the braces in his kitty, he had put them. He has put a edgewise bracket at 90 degrees, he's put a Beggs bracket again in reverse. Somehow he passed the wire, added the magical E-chain, and this is the end result. So we can't be like this. So we need to be good in our bonding skills, whether it is metal, ceramic, diamond, self-ligation, or anything. To give clean bondings, we need to go like this. If this can play, yeah. I wanted to. So the volume is not there. This was just to show that the amount of happiness you get from the patients the day you debond your braces. So the foundation starts from the time when you had bonded them actually. So as we know, bracket positioning is very important for correct diagnosis, treatment planning, and all the other things. And especially when we are treating straight-wire techniques and pre-adjusted brackets, it become even more important. So the duration of treatment is definitely shorter when we are doing nice ones. Most of the PGs out here must be first, second year PGs, but just some more time you will get transferred cases from your seniors, don't worry, which will have all bracket positions, maybe at wrong positions. So that's a big issue when you get transferred cases. So don't let that situation come to your juniors. So please bond them properly. And they must be happy. Otherwise, this is what, as a junior, you're going to feel like, oh God, what has happened? All the 24, 28 brackets have to be rebonded. So good results, good bonding, good smiles. This is what we achieve. So coming to the basic process, see, I am talking very basic, but that works actually. One step which we miss as a postgraduate or in our clinical practice is the first step, which is prophylaxis. We miss that, believe it, we miss it. And we just start our etching process and all that as we get busier day by day. So one prophylaxis thing that really makes so much of difference, or whatever old grinded TC burrs or diamond burr, finishing burrs which you have, just clean the surface of the enamel and then go ahead with etching, sealing, bonding the selected bracket. Always remove flash and cure it. One thing, whenever you get a transferred case from somebody across the globe, across the country, if your bracket has a lot of flash, the first impression as an orthodontist goes, he has not done good job. He doesn't even know the basic. Always remove a flash. It takes few seconds. Always do that. Okay. So the basic process, as we know, is we have to have a clean adherent surface, good wettability, intimate adaptation, bonding, and ultimately good curing. So whether you are doing a mechanical, whether you are doing a metal brace, whether you're doing a ceramic brace, only thing is a mechanical locking. There is nothing called chemical bond. It is all mechanical bond. And coming to the basic systems which we use, all of us are aware there are 7 generations. As an orthodontist, we are mostly talking about 5th generation or maximum 7th generation. If you are working on enamel, you have to be on 5th generation. If you are working on more of dentine, it has to be on 7th generation. But still always preferred is the 5th generation, which all of us use, in which there is one bottle and one etch. There is a primer and one separate etch which you have to use, and that gives the best result. Every research has shown that when we etch with a orthophosphoric acid, it always gives a better etching as compared to 7th generation. So as I said, use prophylaxis, avoid oil-free mix. Try to make that old slurry, get that very easily pumice, mix in water and do it. Don't go get those marketed products. They have either incorporated oil or wax to make it into a slurry. Okay, those surfaces where brackets are to be bonded must be cleaned. While you are doing this, please avoid those used flared out polishing brushes. What they do is they will clean, but they will also lacerate your gingival tissue. And when you are bonding premolars, you should avoid gingival trauma because the bleeding is not going to stop and it's going to create problem for you. Okay, coming to the etching, we know only etch till the area where we need. Don't etch your whole surfaces throughout the tooth. Use good etching suction system. 15 seconds of etching is perfect. Different studies have been done in which they have tried to put etchant for 15, 30, 60, maybe for 2 minutes. But it has been seen that if you keep it for 15 seconds, that is the best. If you keep it for a longer time, those enamel tags are going to break on themselves and they are going to clog that etching area. So when your boss calls you, come, I have to see your case and you have already done etching everywhere, don't run away. Please request, I have to wash and let it, then only leave your patient. Okay? So when you come across certain specific cases, like in our North India, we have a lot of fluorosis cases. Plain phosphoric acid is not going to work. You have to use this magic instrument that is a hypo etcher, in which you use aluminum oxide. You just flare it and you will get a clean surface, and then use a phosphoric acid on top. So once we have done the etching, you have to get this chalky white enamel. If you are not getting it, don't be lazy. Again, clean it, again do it. Rather than your bracket breaking and patient coming back, it is always important to do it. We are all read this landmark article of Silverstone. He has given different patterns, type 1, type 2, type 3. Type 1 is the best which we need. It has a generalized roughening of enamel surface with a distinct hollowing of prism centers and intact peripheral margins. This is what we get at the center of a crown and more in the anterior tooth. In premolars and molars, we get more of type 2 and type 3 pattern, which is not very strong way of doing it. So have you observed that there are more bond failures happening in the premolar area? Why is it happening? There are 2-3 reasons. First reason is, as I said, enamel tags formed in the cervical region were shorter and less numerous than formed in the middle and incisal third enamel surfaces. So in middle and incisal type 1 pattern, as we go up, there is type 2, type 3 pattern, which is a weaker thing. So in premolars, since the cervico-incisal height is smaller and bracket is nearly same size, so our bracket is more in the gingival area. So that is why we often fail. And second, very important, crevicular fluid contamination. All these materials are hydrophilic. As you apply the bonding agent, it will just suck the crevicular fluid from the gingival crevice and contaminate that portion. So your bond is going to break there. And obviously in premolars, we have a lot of traumatic bites, so it can happen. So apply the bonding agent once you have done etching with the applicator tip. Just apply once, don't rub it. It is going to break the enamel tags. Once you have applied it, do not just wipe. Let it be there for a few seconds, maybe 10 seconds, and then you air dry it. So certain keys of success for light curing adhesive: light must be of proper intensity. Obviously, you have to check that. Divide the light curing into 2 increments. Always cure from back, front, and keep your light slightly away first and then touch the bracket. Don't be fear. Pehle thoda dur se, then get close and touch the bracket, because all research has shown even with few millimeters of best of light cures, the intensity drastically reduces. So you have to touch your light with the bracket and cure it from front, back. Keep checking your light with the light intensity meters you must be having in your departments or in your practice. Do it. Okay. So this is the light which I use, 3M Deep Cure light. I know as a student it is not possible, but in the future, spend on your armamentarium. Take good lights, the brackets will not break. This light is somewhere around 1 lakh rupees. I know it is expensive, but I have 3-4 of them and I use them very nicely. Now coming to how to select the proper bracket. Obviously, when you are picking from the kit, everything is well nicely fitted and everything, but you are often have to rebond. So certain clinical tips your teachers must have already told you. In a new kit, most of the time this dot is there. Then there is an indentation. In canine, the power arm is on the distal side. There is a small indentation also. So in your burned bracket, which you are rebonding, you can see that indentation, not the color. Obviously, for premolar bracket, this is in the mesial side. In the canines, it is always in the distal side. Okay. Always use a sandblaster to clean the mesh before you rebond your bracket. That after flaming the bracket, even putting in cold water, that black film of carbon is always going to be there. So if you have got a sandblaster, just blow it once, the mesh is going to be very clean and it will help. Okay. Don't apply composite on your bracket mesh in increments. Apply in one. If you are going to apply in increments, it will incorporate porosity and the bond failure is going to be weak. Okay, and one more thing, different thoughts, school of thoughts. I personally apply a layer of bonding agent on the back of the mesh also. That helps. Somehow it helps in my practice, but it is your own choice. Some people say don't apply a bonding agent on the back of the mesh, only apply on the enamel surface. But I personally do it on both sides, and it helps me. Okay, always remove flash. If you have not done it at the time of cure, before curing, always remove it. Don't feel lazy, yaar, agli bar kar dunga. Aisa na karo, because there will be lot of plaque deposition all around, and it gives a bad impression on your name of as an orthodontist. Okay, cure for 20 seconds for beginners. My first bonding, I am from PGI, first batch, and I'm talking 28 years back. My first bonding took me 7 days. Every day for 4 to 5 hours, I used to bond the same patient. After 7 days, my first trap up was over. So this was the amount of time I spent there, and all our colleagues were doing the same. So when you are only bonding 3-4 brackets, always tie them with figure of 8 and then send the patient. Otherwise, whatever hard work you have done, that will also go off. And the patient might ingest that bracket. Okay, always use the bracket from front, with a mirror from the down. Okay, I'm skipping a lot of things, time constraints. And mirror, as I said, put it below, check the position, as per the things, as per the system which you are using, where you have to place, you have to check that. Use height gauges properly. Okay, in incisor, gauge is placed at 90 degree to the label surface. In molars, it is placed parallel to the occlusal plane with a mirror, and check all that, and you will feel you are able to place the brackets better. Now coming to molars, since around 20 years back, I have totally shifted to bonding. I don't make any more bands until unless specifically required for a cause. So molar, give special attention to molar bonding. Remove the tongue guard, very important, because if the tongue guard is there, you won't be able to see the molar from, because you can't take a mirror back. So whenever you are bonding a molar tube, remove the tongue guard, give special time for molars, and as we know, notch of the tube must fit into the mesiobuccal groove of molars. Okay, this was the thing which I feel every person should have in his practice, either for removing that smoke layer from the back. Sandblaster, or also known as micro etcher, it uses 90 micrometer tan aluminum oxide at a certain pressure, which you can just apply or fix into your chair itself. It can be used for rebonding loose brackets. It can be used for bonding directly on the deciduous teeth. You don't need to etch those teeth. And it increases the retentive area inside the molar bands also. Okay. Now coming to bonding on porcelain. Nowadays, with my practice, 90% is adult orthodontics. I have patients who have already got veneers, who have already got ceramic crowns, who have already got zirconia crowns. So how to etch them? Very often people tell, no, our bracket breaks after some time, but I have observed they stay for a year or more if you do it properly. So always isolate the working area. You have to deglaze the surface. If you are putting on a ceramic crown, you have to deglaze again with a 50 micrometer aluminum oxide. Instead of phosphoric acid, you have to use 9.6% hydrofluoric acid. That is the difference in acid, which is a very corrosive acid. You have to handle it very carefully, preferably by putting 2 gloves at that time. Okay, carefully remove the gel. If you're etching, same thing, you should remove that gel gradually with a cotton rather than washing and subjecting the whole tissue to such a corrosive acid. Okay, for molar tube, if you are putting it, you can use this dam or any old composite, you can fix it, and that will allow the check of hydrofluoric acid in this area. Okay, like this case, full case of crowns, there was midline diastema, 65 years old lady. What we did all the same procedure. One thing, after hydrofluoric acid, wash it thoroughly for 20 to 30 seconds. Immediately after that, always use a ceramic primer, that is silane coupling agent. You have to use this. A lot of people miss this step. You have to use a silane coupling agent, which is a self-cure material. You don't have to light cure that material. After that, after 30 seconds, you have to use a 7th generation bonding agent, not a 5th generation bonding agent. See, these are the mistakes people do. You need to use a 7th generation bonding agent after ceramic primer has been used. And then you put your brackets and move teeth. Okay? Sometimes you have large composite restorations in which you have to bond. There is a very big composite restoration on the buccal pit, and you have to bond a tube or a bracket. So always resurface the composite before you put a bracket. Old composite layer has to be removed, a bonding agent has to be applied, and always do like this. Okay? And use an intermediate primer or a 7th generation bonding agent here. Now, as I said, since aligners are so much into it, just few slides. If you are not going to bond right attachments, that is also a type of bonding, you are going to fall from top down. Your aligners is not going to work because your attachments will keep on breaking every second day. Your attachment should stay for whole treatment. So you should avoid that. So the system which I use, one of the aligners here, and this is how the attachments look on visually. So basic clinical tip, this is the 2 armamentarium which we require for bonding attachments. And etch, see the treatment sheet. Again, don't etch the whole of crown because that flash is very difficult to remove. In that attachment template, you can see the exact position of attachments. Only etch that area. Only etch those specific areas. Sometimes it is in incisal, sometimes in middle. So do those, clean, then apply the composite on the back of this into those attachment points on the template and keep it inside a dark space, one of your drawers as you've done it. And like this is for lower. And then you need 2 light cures. You clean the area, you use a normal bonding agent for the upper where you have done etching, and then you light cure it. This is plastic. Don't light cure the attachment for 30-40 seconds. Always, sometimes try putting a light cure on top of your nail and see how it hurts after 5 seconds. It gives a pain. So imagine about your tooth. So one more tip which I just learned 2 years back, keep moving your tip across the tooth or the aligner. Never keep your curing tip at one point. Keep moving it so that it doesn't heat up the enamel. And here that plastic is going to melt and it's going to stick to your composite. So just 10 seconds, take it out, peel it off like this. As you see, the upper one has come out, lower one has come out here. And now we've got nice attachments, which sometime if there is extra material, you just remove it with a burr. And this is Invisalign which I gave. And one small tip, again, I'm giving more of clinical things. So you have done everything. After 6 months, the patient comes. There was a midline patient you have treated. Small space has come. Now what to do? You can't put the braces back. It can't be closed with a retainer. So one very simple system. Etch on top cervical areas, etch those specific areas, put a module there and put flowable composite on top and cure it. As you're going to do it, it is going to become, and after that with a sharp explorer, remove these modules. They will appear like lingual buttons, not visible, nothing. And within 2 to 3 weeks, you can put E-chain and close up all the space. If required, you can tie them with figure of it with a ceramic module, and all the spaces have closed. In 1 month, you are back with the patient and giving retainers. Like this patient, he had ceramic veneers on top. So we did the same technique. We started with a lower one because he said, I don't want braces in upper immediately. So we closed up the space. Obviously, no hull in orthodontics have come for turmeric. I hope somebody works and gets some remedy for the Indian turmeric, which is a big enemy of aesthetic orthodontist. So for the bonded retainers, it has been already very nicely been told by Dr. Ankit Sikri, but always clean the lingual surface. Ye nahi ki your treatment has finished, you have not resurfaced and directly done the bonding. Otherwise, your retainer will break. As simple. Keep it, depending upon the case, if it is a deep bite case, keep it above cingulum so that it prevents the eruption back. Okay, this is the retainer which we use in PGI. I have patients with even follow-up of 20 years having this retainer. It is only bonded at 2 portions, very hygienic retainer, very clean, doesn't get too much of deposits. And one more tip here I want to give for surgical exposures. See, at times in your practice, you will come across this situation. You have called an oral surgeon, you specially come after showing lot of tantrums, I don't have time and all that, and you have opened up everything, and now you want to bond lingual button or whatever you want to bond. See, you have closed the flap, surgeon has gone back. As you have pulled the ligature or the E-chain, your lingual button has also come with it. So in such situations, as I said, these materials are hydrophilic. They are going to pull the blood out as you're going to apply the bonding agent on the exposed tooth. So in such situation, one tip is on the lingual button, apply the composite, apply bonding agent excessive amount on the lingual button composite itself. Press it, immediately cure it. That means all that liquid is below the lingual button. It's not going to pull the material. So in that case, this will last for long time. You will see that once the tooth comes out, all these cases, we always try to do it like this. Regarding bite blocks, which you have always used, again, give few minutes, clean the surface, do etching, then apply the blue block you must be using. One tip I use in my practice, lot of the patients, they say we get a lot of pain hitting on those bite blocks. So I have bonded this modules on top. While before curing, put a module, press the centric point is now on the module. So it takes some pressure, so it helps. Okay. So coming to some troubleshootings, since we have already told most of the things, and there are 3 types of failures. So type 1 bond failure, in which the majority of the adhesive is on bracket. Always introspect yourself. Patient has come with a broken bracket. See, check where the bracket is. It is on the tooth, where the adhesive is, on the tooth or on the back of your bracket. If the majority of adhesive is left on the bracket, what are your faults? You have not done the prophylaxis. Subse important point. You have not done it. That is why everything was sticking with the tooth, with the bracket. Nothing was there on the tooth. Rubbing, etching. You have already rubbed that etchant. Too short curing, incomplete rinsing of enamel. All those points which I have said before. Excessive primer, limited primer. Okay, insufficient drying, moving bracket during adhesive gel preparation, hard, fluorose, calcified teeth. You have not used hydrofluoric acid, it is going to happen. Other situation is when the majority of adhesive is on the enamel surface. This is what you need at the time of debonding. You must have read that. When adhesive stays on the tooth, it doesn't give micro cracks to the teeth. This is what you need. But if that is the reason, that means you have pasted too much of composite and a lot of porosity was there, so the mesh ke upar composite has not flown. And for ceramic brackets, you have not done silination. And if it is a cohesive failure, patient abuse, improper light cure and all that. So I have still a lot of presentation, but sorry, I'll stop here. I thought these were the main tips I should share with you. Rest a lot of clinical cases, lot of things, how to debond, how to reposition. I'm always there, a call away. You can always contact me for presentation. Thank you.
It's always an honor to listen to you, sir.
I couldn't finish, sorry.
Especially getting the best seat in the house to listen to you, just really an honor. Thank you so much, sir.
Thanks a lot, sir. As you said, it's an honor.
I request the chairperson to please felicitate Dr. Sarabjit Singh, sir. I request Dr. Prashant Dole, sir, to please felicitate the chairperson for this session. Our next speaker for today is Dr. Prashant Dole sir. Dr. Prashant Dole sir is diplomate, Indian Board of Orthodontics. He is a postgraduate from GDCH Ahmedabad. He has his exclusive orthodontic practice in Mumbai for the last 25 years and has been invited as a speaker in many national and international conferences. He also has his publications in national and international journals like JCO, EPOS, Trends, and JIOS. He has been the convener of Bombay Orthodontic Study Group from 2008 to 2013 and organizing secretary of accredited international conferences like Interdisciplinary Orthodontic Symposium Mumbai 2018. He has also been an active member of Indian Orthodontic Society, AAO, Fellow World Federation, Indian Society for Cleft Lip and Palate Anomalies. To chair this session, we have Dr. Vinay Kumar sir, Associate Professor in the Unit of Orthodontics, Oral Health Sciences Center, PGIMR, Chandigarh. He has been granted patent for his design of intraoral distraction splint. His key area of interest are functional jaw orthopedics, cleft care, craniofacial symmetry and syndrome. We welcome you, sir.
This being one of the last presentations of today, I feel I need to wake your senses up. Okay, now everybody, till now has been talking about teeth, okay, the jaws, or which is below, I would say, below nasal line. I'm going to talk something which is above nasal line, that is TM joint disorders. And to wake your senses up, what I would do, I would want you to raise your hands up. Raise your hands up, everybody. Take it down, raise it up, and clap. 1, 2, 3. Good afternoon, everybody. So my name is Dr. Prashant Dole. I'm going to talk on TM joint pain disorders, and I thank the PG Convention, 27th PG Convention, for giving me an opportunity for this. I thank Dr. Shailesh Shenwa. I thank Dr. Prakash Mudliar to be allowing me to be a part of it. So starting with my presentation, I would like to start with explaining or stating about what are TM joint disorders. Okay, as orthodontists, we treat teeth, we treat jaws, we do lots of growth modulation. But what we many times forget, that something which is connected to everything what we do are the TM joints. And if at all, considering orthodontic part of it, if we are not aware about it, if we fail in our ability to understand TMJ, probably we may land up into TM joint disorders. So temporomandibular disorders are a group of conditions that cause pain and dysfunction in the jaw joints and the muscles that control the jaw movements. So that is basically the jaw joints as well as the muscles which control the jaw movements. So understanding about TMD or TM joint disorders, we orthodontists are probably the best people today in the medical fraternity to understand and treat TMD, because we understand growth and development, we understand form and function. Everything is related to function. Everything, the modulation, the shape of the jaw, the ability of any function to happen, that is that we understand better in terms of stomatognathic system, and we understand the dynamics of stomatognathic system. That's what I'm telling you. So as an orthodontist, I would do changes in occlusion, I do changes in the skeletal proportions, and I do changes in the functional environment as well. So I need to be adept with it. I need to be aware about this. So role of an orthodontist in TMD: identification of undiagnosed or developing TMD in patients coming for orthodontic treatment. So patients would come to you for orthodontic treatment, for correction of crowding, correction of proclination, so many things. But if you are going to be missing out on diagnosing TMD, and if the patient develops TMD during the treatment or after the treatment, you are going to be blamed for that. So it is important for us to understand TMD, and we should be probably looking at the temporomandibular aspect in terms of diagnosing our orthodontic patients. What measures we should take to prevent our TMD during and after orthodontic treatment, and providing correct treatment to the symptomatic patient. So if the patient comes to us with complaint of TMD, are we trained enough, are we knowledgeable enough to understand and to be able to deliver good quality treatment to the patients who come to us? So we should always look at TMJ or TMJ functioning or TMD when we are addressing patients as a whole. Audio? Okay, now this is a typical patient. I think audio is not visible. Audio is not audible. Is it audible? Okay. So these are the clicks. These are the clicks which a normal, patient who will come to us with a TMD will exhibit. And the patient who is already undergoing orthodontic treatment, you can see how the jaw is deviating on the right side. That means the left side of the TM joint is being affected. So these are the typical cases which you could see who are having TMD problems. So understanding TMD, to be able to understand TMD, we should know anatomy of TMJ. We should know how TMJ normally would function. We should understand what abnormal function of TMD, and we should understand the signs and symptoms of TMD. Then how to arrive at a correct diagnosis. It is important to diagnose TMD in the correct perspective if we are able to treat it correctly. Then again, phase 1 treatment, when the patients who come directly for TM joint disorder, generally, most of the patients are given the splint or a night guard or whatever, but is it enough? Okay, although the literature says that the treatment which should be recommended for TMD should be conservative and reversible, but is it enough? Okay, by doing conservative treatment, you are giving a palliative treatment. Okay, palliative treatment is something which is given, but is it enough? A good clinician or a good doctor is somebody who knows that the palliative treatment can be given, but when there is a definitive cure, if it exists, and if he is not able to give that to his patients, I think he is failing in his duty. So phased elimination of the etiology of the TMD after we get the symptoms corrected, and phase 2 treatment. I'm going to emphasize on that. Phase 2 treatment to correct the effects of TMD which has happened in the somatognathic system. So that is important. So coming to the anatomy, I'll quickly go through this. Let us understand the bony anatomy, articular fossa on the cranial base, the mandible condyle, and the articular eminence in the front, and the tympanic part of the temporal bone behind. So these, the mandibular condyle is basically covered with fibrocartilage. So is the articular fossa. So fibrocartilage is important because it helps to redistribute the force, and in addition to the articular disc and the synovial fluid,